All right, looks like we are live and we've got a great audience and a great chat already. Some great people in the live chat. Thanks for being here. This is definitely going to be a debate to remember. I'm extremely excited for this one. And real quick, actually, before I bring in Jackson Roe, as well as Praise I Am, who is going to be moderating this debate, I want to go over the topic as well as the format for the evening. So we are going to be debating human evolution versus independent origins, created or evolved. Now, the format for the night is pretty similar to the debate I had with Bill Tavis last week, which was definitely one to remember. We are going to be having 14-minute opening statements. I should say up to 14. So 14 will be the max that we can take for our openings, and whatever we don't take, we can just throw into the audience Q&A. Followed by 10-minute uninterrupted rebuttals, followed by another rebuttal round of 5 minutes, and then a 25-minute open discussion where we can ask each other questions pertaining to the topic of the debate, ending with 2-minute rebuttals to close out the discussion and a 5-minute closing statement. And then, of course, roughly a 25-minute Q&A with the audience. So please, everybody in the chat, make sure you are tagging Praise I Am and not myself, because I won't see it. I will be debating. I know you guys are probably used to me moderating, but today I'm debating, and that is why we've got Praise here, the #1 moderator, as well as Jackson Roe. Gentlemen, thanks so much for being here. Let me unmute you guys. Praise, thanks for being here for moderating, and Jackson, thanks for being here for this important debate.
Thanks for having me.
Looking forward to it. Hopefully we'll keep this a cordial, respectful, and entertaining debate tonight.
Awesome. I'm pumped. Well, why don't we, since I just went over the format, why don't we hand it over to Jackson Roe for a brief introduction to kind of break the ice. Jackson, who you are, what's going on over at your channel. For example, if people were to go check it out, what are they going to find, and so on and so forth. So thanks for being here, Jackson, and go ahead. The floor is yours for just a brief introduction as to who you are.
All right. Well, thanks for having me. Yeah, my channel's called Jackson Roe Science Show, which is kind of like Bill and I, the science guy. That's why I chose it. It kind of rhymes. And I started like probably just a month ago. Doesn't have many subscribers, but I think I've got some decent content. Not great production value, and I'm kind of monotone, so I don't have that going for me either. But yeah, I live in Arkansas. I'm 27 years old, and I've been into science since I was a little kid. So that's really about, well, I'm into animals. I have a lot of exotic pets. That's about it. If you want to, they probably know all about you, but if you want to talk about your intro.
I appreciate that. Yeah, real quick, I'm definitely no stranger to this channel since, you know, I do run the channel. So I'm Standing For Truth. I've had roughly between 80 to 100 debates. I've been doing this for almost 3 years. It's been a lot of fun. It's been quite the adventure. This is definitely one of my favorite topics to debate. I've debated all sorts of topics, but I like this one because it focuses on the bigger picture. You know, whether we're created or evolved, independent origins, human evolution. If independent origins is true, then obviously that kind of takes out abiogenesis and also takes out universal ancestry with it. So it's a good debate. It's an exciting topic because we're focusing on the bigger picture. So I've also written a bunch of books on this topic. A number of them are behind me. So anybody wants to check those out, I've written about 5, as well as co-authored a large number as well with Ramat. So he also runs this channel with me. So that's about it from me. And looks like we got a super sticker flying in as well from Jeremy Martin. Thank you so much, Jeremy. I appreciate it. And we've got a good chat. So introductions are out of the way. Jackson, thanks for doing this debate.
Thanks.
I'm excited. And we're going to hand it over to you.
Okay.
Praise. Looks like we are going into our 14-minute opening statement. So Praise, how do you want to work it? Are you going to give us a 1-minute warning or? Oh, Praise, you're on mute, brother. There.
Yeah. So, yeah, how I'm going to do this is when you guys get near the end, I'm going to say, go ahead and wrap it up. I'm going to give you about a minute to finish up and conclude your statement, and then we're going to move on to the next interlocutor.
All right. All right. So I'm going to
That's
jump in.
Yes. Thank you, Praise.
All right.
And Jackson, whenever you're ready, let us
Okay.
know, and Praise will start your timer.
All right, you can go ahead. I timed it out. It's about 10 minutes probably, so I probably won't need all the time. All right, my position is that humans evolved from previous forms of primates. Humans and other great apes share a common ancestor. I'll present fossil, genetic, anatomical, and behavioral evidence to that effect. I believe that this evidence doesn't necessarily preclude a God, even the Christian God. Millions of Christians accept evolution, including most Catholics and Methodists. In fact, the majority of people who accept evolution are religious, by one poll I saw anyway. While I personally don't believe in a God, I don't think modern science and God are completely incompatible. But back to the subject matter, I'll be presenting evidence about Australopithecine bipedalism, which I saw you give a talk, or Dr. Biddle give a talk on that on your channel. Modern ape anatomical, genetic, and behavioral similarities to humans, and probably some miscellaneous stuff thrown in. So first, I'll just touch on that, the Australopithecine stuff. Most creationists seem to only be aware of the Lucy fossil, which is Afarensis, but there are actually many species and many fossil specimens, not only of other species, but of Afarensis as well, including the what's called the Big Man fossil, which shows a partial pelvis as well, and the Salem child, which has most of its spine and skull. There are also about 400 individual fossils of Afarensis, though most of those are just singular bones or a couple of bones. But from those bones, we can tell that, for example, the Afarensis foot was built for bipedalism. Its heel was robust, unlike a chimpanzee and more like a human. Its fourth metatarsal, which has been found, indicates there was a stiff midfoot, unlike a chimp, and also an arch, like a human, an adaptation for walking. Same thing with the Africanus foot, Australopithecus africanus. Now, the Australopithecine spine was more S-shaped than the chimp spine, which is more arch-shaped. That's an adaptation for bipedalism. They can tell that from different spacings and different components of the vertebra. So also the foramen magnum, which is the hole in the underside of the skull, is intermediate between a chimp-like ape and a human-like ape. So it likely held its head similar to ours as it was upright. Its femur is angled the same way as ours, as opposed to a chimp's. And the pelvis has been kind of contentious with Lucy. I know some people have argued that it was filed down or faked, but there have been several Australopithecine pelvises found, including 2 Sediba pelvises, 2 Africanus pelvises, and 2 Afarensis pelvises, including Lucy's. Also Prometheus, which may actually just be an early Africanus, but some say it should be its own species. Now, where was I? The ankle joint of the Australopithecines are square-shaped, the ankle bone, where chimp is trapezoid-shaped, which allows for more flexibility for climbing, which the Australopithecines apparently didn't have, except maybe for Sediba. The rib cage was originally thought to be more cone-shaped, like a chimp, when Lucy was first discovered, but further finds have indicated that it's actually more barrel-shaped, like ours, so they would have had more of a waist than great apes have. The previously mentioned Prometheus species, which is known from the Littlefoot fossil, which is the most complete ever found of an Australopithecine, shows that the arms were in fact shorter than the legs, which is unlike gorillas, orangutans, chimpanzees, and a very human-like feature. The brain capacity was about 450 cc's, where a chimp's about 400, and early Homo species are in the 500 range, so it's intermediate between there. I'm not saying we evolved from chimpanzees, I'm saying we share a common ancestor. I'm just using chimps as an example. In fact, the line between Australopithecus and Homo isn't so clear-cut. Homo habilis, there's some debate about whether it should be classified as Australopithecus or not. Most scientists say it should be Homo as it is. There are a few that say it should be Australopithecus. And even Homo erectus, in its earlier stages, where it's called Homo ergaster, which is no longer recognized as a species, but actually just an early Homo erectus, had a very small brain capacity, had some more primitive traits. Same with Homo georgicus, which is also just an early Homo erectus. And Homo erectus is the most known from the most complete hominid skeleton ever found, the Turkana boy. I forget when that was found. I should have written that down. But anyway, the Neanderthal genome has been sequenced, and it shows that creationists often argue that Neanderthals were the same species as us, Homo sapiens, but it shows that we differ by 202 base pairs, where by comparison chimps differ by 1,400 base pairs. So they were very close to us, but they weren't quite human. Now moving on to modern great apes. While it's true that 99% of the regions that code in our DNA and chimps are the same, it's been argued that it's not 99% all the way around, which is true, but it's not dishonest to say that they're the closest living relatives we have. No other animal, plant, fungi genome comes close to ours the way a chimps does. In fact, chimps are more closely related to humans than gorillas are to chimps. And molecular clock data, which is mutation rate between generations, indicates that our common ancestors split off from chimpanzees 6 to 13 million years ago. Now, one of the best evidences for common descent in the genome is the endogenous retroviruses, which is when a virus infects the gamete cell and gets passed on to the future generation. And that's important because we share 99% of ERVs with chimpanzees. And that could really only be true if we shared a common ancestor with them. Now, behaviorally, there are some real similarities. Kanzi the bonobo has been taught to use hundreds of lexigram symbols to form novel sentences and understand novel sentences. And while that's not unique in the animal kingdom, it seems unlikely that something that wasn't our closest relative would know. Same for Coco the gorilla. She knew 1,000 sign language signs, understood thousands of spoken words, displayed human-like emotion. Chimpanzees are the only other animal known to have ever fought in an actual war, the Gombe Chimpanzee War, which took place in the early 70s. Over the course of 4 years, one tribe of chimpanzees totally wiped out the other tribe. So it's just a unique thing among the animal kingdom that we share with chimpanzees. Anatomically speaking, we should have the same bone number as the other great apes. We have the same number of teeth. We have the same teeth layout, the same number of incisors, canines, premolars, and molars. We have the same trichromatic vision. We have the same ABO blood type system. We have dermal ridges on our fingers, which is very rare in the animal kingdom. I think only the koala has those, other than the great apes. I may be wrong on that, but also our fingernails are very similar and unique in the animal kingdom. Now, some arguments against the Adam and Eve argument. primarily inbreeding. Now, even if Adam and Eve had the perfect genome, as has been suggested, after the fall, they would have started to acquire mutations from just mutagenic factors like UV radiation, and they would have passed that on to their children, and then their children would have acquired more mutations through the same factors, and that would have been amplified when the children had to inevitably reproduce with each other. And from there, it gets just really, really bad. They just more inbreeding brings recessive traits, harmful mutations to the surface, and pretty quick you get all sorts of negative mutations that are passed on and not selected out. There's a good example in the animal world, the animal breeder world, like ball pythons, axolotls. When you inbreed those, you get these color morphs: albino, leucistic, piebald. It's hard for me to read my handwriting. Hang on. Why were they even given the option to eat the apple? I think it's because the story is an allegory. Why were they made from dirt and then a rib? I find that hard to believe, and again, it goes back to being allegorical. And also the talking snake speaks of allegory as well. And why would God, being omniscient, not be able to see the future of Adam and Eve eating the apple in the first place? You would think he would, being omniscient, see that outcome. And why would all humans be punished for the mistake of two? The concept of original sin doesn't really make any sense to me, but that's kind of a different debate. Also, mitochondrial DNA doesn't really support the claim of Adam and Eve. Y chromosome Adam and mitochondrial Eve didn't live at the same time. Mitochondrial Eve, our last female common ancestor, lived about over 100,000 years ago, and evidently Y chromosome Adam lived tens of thousands of years apart. And nuclear DNA shows that the population of humans was never below about 10,000. So that kind of precludes the idea of only two human ancestors. That's pretty much my opening statement. I'm not sure I did it right. It's my first debate, but not sure how much time I used. But Standing for Truth, if you want to take it from there.
So you had about a minute and a half to spare, so we can add that in the question and answer period, or if you want to add that to your finishing comment later on. So SFT, you're next in for your opening statement, and whenever you're ready, then I'll start the timer.
Okay, awesome. Praise. I'm going to share screen right now, and then let me know if you can see it. Just 1 second.
Yes, I see it.
And then I'm just going to mute you, Praise, because I hear like a little beeping noise, which is fine. So just when you give me that 1 minute warning or 2 minute warning, just make sure you unmute yourself for that. And I'll let you know when to start the timer. Just 1 second. All right, Praise. As long as the audio video is good, maybe I'll get the A-OK from the audience as well. We can. Audio good, video good, or audio good, slides are showing?
Yeah, I can see it.
Awesome. Okay, here we go. So, Praise, you can start the timer right now. Tonight's debate is human evolution versus independent origins. Are humans specially created and made in the image of God? Or did humans evolve slowly over time from ape-like ancestors? Are humans related to chimpanzees and the other great apes? I love this topic because the evidence for independent origins also demolishes the so-called evidence for human evolution. I am going to waste no time and just get right into the data. My opening will actually rebut many of Jackson's arguments that he put forth in his opening statement, and then I'll save the rest for my rebuttal round. So here we go. To start, we have the major unsolvable problem of mutation accumulation and genetic degeneration. Mutations are accumulating far too quickly for natural selection to filter out the damage. What type of selection can Jackson invoke that will counterbalance the accumulating damage? DNA is a very fragile molecule. From one generation to the next, many mutations are passed on. Roughly 100 new mutations per person per generation. We are consistently more mutant than our parents. As a matter of fact, we are all multiply mutant, which is why selection is so limited. This is 100 typographical errors every generation that our parents did not have. Natural selection must remove or filter out these mutations, or else we will go extinct. The problem for Jackson here is that these nearly neutral mutations are only slightly deleterious and only slightly harmful to the point where they do not really affect reproduction. Selection cannot see these, which means species are going downhill. And Jackson does not have millions of years of evolutionary advancement. Low impact deleterious mutation accumulation puts shelf lives on genomes, meaning Jackson does not have the deep time he needs to evolve an ape-like creature into a human. We also see a perfect biological decay curve that matches the biblical model of longer lives closer to the creation event, while it drops off after the flood. This data has almost no chance at being coincidence. This is an amazing correlation between what we know about mutation accumulation, genetic degeneration, and biblical longevity. It makes no sense that the biblical authors would ever make this up. Also, fabricating this biological decay curve would require exceptional knowledge in statistics and mathematics. The systematic degeneration of man as documented in the Bible is extremely consistent with genetic entropy and deleterious mutation accumulation. The Genesis biological decay curve reflects real biology. Also, many of the so-called icons of human evolution are actually not evidence for ape-to-man evolution. They are evidence for genetic degeneration in the past. I will go over just a couple of these for now, and this also addresses some of the icons that Jackson brought up in his opening statement. So we've got evidence that Homo neanderthalensis was highly inbred, had a very high genetic load, 40% less fit than modern humans. This severe genetic degeneration probably contributed to the disappearance of that population. Also, the hobbits, Homo floresiensis, was also inbred and apparently suffered from a special type of genetic degeneration called reductive evolution. This results in reduced body size, reduced brain volume, and various pathologies. The hobbit-like human species may turn out to have simply been the remains of a human suffering from genetic illnesses. And this is exactly what we would expect given the post-Babel model, where you've got these people groups isolating themselves after migrating from Babel. What you get is founder effects and rapid genetic degeneration due to inbreeding. The anomalous features that the evolutionists like Jackson like to point out are transitional are actually due to pathology and degeneration. Homo naledi, same thing, likewise inbred and suffered from reductive evolution. Results in the same types of anomalous features: reduced body size, reduced brain volume, and various pathologies. Homo erectus, which Jackson brought up, same issues. Was inbred, suffered from reductive evolution, all consistent with the post-Babel model of isolation and inbreeding. So let's get into the next portion of this opening here. Jackson also talked about molecular clocks. So based on the Genesis creation account, we can make some extremely specific predictions and retrodictions. These have all been confirmed. We see low genetic diversity in humans, which is a direct expectation if we really did originate from just two people thousands of years ago. Starting from just two people automatically restricts genetic diversity. If we have been evolving for millions of years, we would have populations with a lot of mutations floating around, which would suggest we have high levels of genetic diversity today. We do not see this. We also have strong evidence for one mitochondrial DNA ancestor and one Y chromosomal ancestor of all people on the planet. The mitochondrial DNA compartment that we only get from our mother picks up a mutation about every other generation. The mitochondrial DNA mutates far faster than evolutionists have ever predicted. When we build these phylogenetic trees or family trees, we can look at mitochondrial DNA diversity worldwide. We can also see how far apart people are. And with the fast mutation rate, Eve, the mother of us all, existed only a few thousand years ago. There are only a few DNA differences separating any two people on the planet. How does Jackson deal with this data? Paper after paper confirming this. Oh, just 1 second here. The Y chromosome as well is also fast, picks up about 1 mutation every generation. There are only a few hundred mutations separating people worldwide. And our last Y chromosomal ancestor, being Noah, existed just 4,500 years ago. This is all inconsistent with universal common ancestry. I want to go to a phylogenetic tree here because I've pointed this out in the past. Dr. Robert Carter, he took all the Y chromosomes and made a Y chromosome family tree. This tree is incredible because what we see is exactly what we would expect if independent origins and biblical creation were true. What we see visually is an explosion from a central point. This is an explosion essentially from Noah, the last Y chromosomal ancestor of us all. This is basically all a reflection of DNA differences, a reflection of mutations over time. This reflects far too few mutations if deep time evolutionary history were true. We are not looking at tens of thousands of mutations here. We are looking at just a few hundred mutations, as would be expected if the Bible's account of independent origins were true. The data, this data here, did not have to be true, and yet it is. Evolutionists are constantly having to retrofit the data into their evolutionary story. They did not predict low genetic diversity in humans today. They did not predict the one Y chromosomal line, nor did they predict the one mitochondrial DNA line. One last thing to consider is the fact that the length of some of these branches indicate that some people can pick up more mutations in the same amount of time. It's difficult to look at the branch lengths and conclude a definitive amount of time has passed. This is why the pattern and the overall data is so important. It turns out this is what we see for the mitochondrial DNA as well. This is a mitochondrial DNA phylogenetic tree. This is only a reflection of a few hundred generations. Remember, God tells us that everything was created very good. When we look at the modern scientific data, we see a reflection of this. Everything must have started very good right from the beginning. The incredibly complex information storage system of life could not have been built up from inadequacy to precision. If we did not have a highly functioning decoding system, the entire system would come crashing down. Origin of life research has been a massive failure and faces numerous unsolvable chicken and egg problems. Also, evolutionists have no way to introduce functional information into the first life. How do you introduce functional information into random chemicals? If you manage to get the necessary building blocks for life, how is information introduced? This is a major problem for abiogenesis and naturalists such as Jackson here. If I were to spill ink on a paper, I would not at the same time introduce information. A book is more than just ink on paper. A book contains specified information. Also, these next few slides are going to address some of the arguments that Jackson brought up in his opening statement, such as endogenous retroviruses. We just touched on his arguments pertaining to molecular clocks. Let's deal with this data, because this brings me to the fact that we now know the entire junk DNA paradigm has been overturned. I want to point out that the design diversity model, the model that suggests Adam and Eve would have been front-loaded with DNA diversity, which would also apply to the created kinds, would predict that most of the genome and most of these DNA elements found within the genome, including pseudogenes, endogenous retroviruses, ALU sequences, and many other types of variation-inducing genetic elements would be highly functional and active, and this is exactly what we find. And once again, paper after paper after paper. How does Jackson deal with this data? Even the chromosome 2 fusion has been completely overturned. Universal ancestry requires lots of junk and evolutionary leftovers, and we just do not see this. We also have overwhelming evidence for forward thinking in genetics. For example, we have our genes, right, which make physical parts of our cells. Well, we also have what is called epigenetics, which is what controls how the genes make the physical parts of our cells. Our epigenome turns genes on and off, as compared to changing the actual genetic code itself. Our genes are not all turned on at the same time. This would not be good. These are non-random changes. Good designs means we have some type of control, a type of control that makes sure we have the right things being turned on and the right things being turned off. This is all forward thinking, which points us back to the forward thinker. Okay, I want to ask Jackson a few questions here for his rebuttal, and we could also discuss it in the discussion portion. So we know that ERVs, which he brought up, and other classes of retrotransposons accomplish many crucial functions in regulating gene expression, differentiation, development, and even cell stress responses. Does Jackson have any empirical evidence, maybe a technical paper, that actually demonstrates a non-functional DNA element such as an ERV, pseudogene, ALU sequence, going from non-functional to something critically functional in the genome? Praise, how much time do I have, brother? Praise, you are muted. Praise, how much time?
2 minutes. 2 minutes.
Okay. Here's another question I have for Jackson. Human chromosomes are made up of relatively large linkage blocks, and analyses of these original text strings tells us that the genome is young. Why? Because the genome has only been partially scrambled. Crossovers in gene conversion should scramble all linkage blocks quite quickly in an evolutionary time frame. This is a major problem for evolutionary theory. How does Jackson explain this data? A near fully functional genome turns the nested hierarchies and the similarities that Jackson pointed to in his opening statement to evidence for design hierarchies as compared to descent with modification. Therefore, evolutionists have to fight the evidence for genome function, including the results of ENCODE, which suggests that over 80% of our genome is indeed active and functional. If most of the genome really is junk, and these ERVs are nothing but junk, why would the cell bother with so many and so much evolutionary leftovers? Such an activity would be wasteful of energy and resources and would have been eliminated by natural selection long ago. Also, since Jackson liked to bring up the molecular clocks, how does he explain the mitochondrial DNA and the Y chromosome data that, based on the empirical method, tells us we came not from just a small founding population, but a population of just two, Adam and Eve, 6,000 years ago? These are all questions that must be answered for Jackson to have any chance at making a case for human evolution. Why? Because these are just some of the lines of evidence that can differentiate between the models, human evolution and independent origins. That concludes my opening statement.
Perfect. You got right under the time there, SFT. Awesome, man. So now we're going to move into the 10-minute rebuttal session, and I'm going to give you a minute warning beforehand to wrap it up, and then you can have that time to wrap it up. So whenever you begin, I'll begin the timer on whenever you want to start your beginning. Go ahead.
All right, you can go ahead. You mentioned genetic entropy. That's more of a creationist term. I think the real term is error catastrophe, which is when too many mutations accumulate. They've done this in the lab, but in some instances, accelerated mutation rates don't necessarily mean the death of the population. Sometimes it actually includes improvements. I was reading one paper today. I don't have a link to it. But you also mentioned Homo floresiensis and its degeneration. You mentioned insular dwarfism. Well, that's not necessarily a negative mutation, or this is something that happens to a population confined to an island. They tend to get smaller. Sometimes they get larger. In the case of some reptiles, but with mammals, it's usually the other way. They get smaller. Now, for the Y chromosome and the mitochondrial DNA being the same time apart, I couldn't find the original paper, but I found some pieces of it that said that's basically a misunderstanding of some of the original data. Now, low genetic diversity in humans. Some populations have low genetic diversity, but for example, the African population has incredibly high genetic diversity. Abiogenesis, it could be disproven tomorrow, and evolution could still be true. personally, I think life came about through natural processes. I don't know how, and no one really does. They have some hypotheses, but there's no one that's latched onto. Now, ERV function, they don't necessarily have to have a function, or they could have a function, and they can still be leftovers of viruses that indicate common descent. And you mentioned that chromosome 2 was overturned. That's not entirely true. The DNA sequences on the two chimpanzee genomes of two chimpanzee chromosomes and human chromosome 2 are essentially identical. And there's a kind of a degenerated centromere. Now, you said, let's see, non-functioning ERVs. ERVs aren't necessarily junk. And the MT and Y chromosome, they honestly don't link up to 6,000 years ago in two individuals. As I've said in my opening statement, from everything I've read, and I couldn't find the paper that said what you're saying. It says over 100,000 years for mitochondrial Eve and tens of thousands of years later for Y chromosome Adam. Now that's probably a lot less than 10 minutes, but those are the points you touched on that I wrote down. If you have anything else for me to address, I don't know if that's the format or not, but I can. But yeah, that's pretty much it.
Awesome. Thank you for your rebuttal. You have about 7 minutes left over. You can put that in the question and answer period if you want, or your final concluding remark. So SFT, your rebuttal is next. Whenever you want us, I'll start the timer whenever you get going.
Awesome. Thank you. Just 1 second. I will share screen in, probably about a minute or two. So just 1 second. Praise. And I'm just writing things down like crazy, Jackson. Okay. Why don't we start my timer here, Praise. So Jackson talked about afarensis quite a bit. This would be Lucy. He talked about some of the bones that were found. I will touch on this as well. He also talked about Africanus, another Australopithecine. He mentioned Australopithecus sediba, habilis. He mentioned we are related to the chimpanzee and we share a common ancestor with them. He discussed erectus as well. So lots of fun topics here to discuss. He discussed Neanderthals and the Neanderthal genome, which is one of my favorite topics. Humans and Neanderthals are the same species based on the biological definition of species, okay? Since we interbred with them. We literally have their genetics. He also brought up molecular clocks, endogenous retroviruses, and genetic similarities with the great apes. I touched on a lot of that in my opening statement, and I will touch on that a little bit more here. But I want to debunk his fossils first, because Lucy herself was found in hundreds of pieces over a large area. And when they say they have hundreds of specimens of Lucy's species, what they actually mean is 400 pieces of bones. Many of these bones even turn out to be teeth. What we find in terms of paleoanthropology is deliberate exaggeration a lot of the times, okay? Because even Lee Berger, a famous paleoanthropologist, has pointed out that there are more people working in the field than there are actual objects to study. He points out most of the record pertaining to human evolution is bone fragments, bits and pieces of post-cranial bones and teeth, lots of teeth. You'll often, actually, I hear a bit of feedback, Jackson, if you could mute yourself real quick, my good man. You'll oftentimes see these skulls, okay, propped up as, say, Lucy's skull, okay, Australopithecus afarensis, when in fact, most of what you have are a few skull fragments, with most of the reconstructed skull being inference-based. He discussed similarities in bones, similarities in blood types, anatomy, physiology, morphology. Well, as we know, similarity does not equate ancestry, as similarity can also mean designer. It is the differences we need to focus on. And the differences cannot be accounted for by the evolutionists. For example, we've got massive differences in gene content and architecture when it comes to the chimpanzee Y chromosome versus the human Y chromosome. Okay, we also have differences when it comes to epigenetics, genomic networks, and gene expression in the same sequences that the evolutionists say are similar between humans and chimpanzees, and yet massive differences even in those similar genetic sequences. Okay, what about orphan genes, these taxonomically restricted genes that have no evidence for any type of ancestry? They pop up, and evolutionists have no real empirical evidence for these taxonomically and essential genes. How does Jackson deal with these lines of evidence, for example? Okay, we can't just look at the similarities, we got to look at the differences. Now, when it comes to the genetic similarities anyway, scientists, what they did, and you can read about this in the original chimpanzee paper, they ignored huge gaps, size differences, copy number variations. And when we consider both genomes, including size differences and overall architecture, we are looking at something like 80% similar between chimpanzee and humans. Okay, this is hundreds of millions of DNA differences that Jackson here needs to be able to explain as to how they can become fixed. Fixed just means get stuck in place. How can Jackson explain? We got this huge waiting time problem about fixing hundreds of millions of DNA differences in such a small amount of time. Okay, I'd like to point out that if we had the exact same type of blood as Jackson indicated, we should be able to get a blood transfusion with an ape, and yet we know this would result in death. So Lucy, okay, afarensis has been proven to be nothing but an extinct form of ape. She resembles many modern day forms of chimpanzees in arm length, height, skull shape, mandible size, structure, among many other details as well. She, her skull, inner ears, locking wrists, curved fingers, and short legs demonstrate that Lucy was definitely an ape. And as a matter of fact, when you look at the fossil record in general, we see an abrupt appearance. It doesn't help Jackson. We see an abrupt appearance with unique differences in humans. Okay, Erectus has very human-like qualities and unique differences, guess what? Not seen in prior hominins. And this is from its first appearance. Humans have always been humans in the fossil record with no gradual transition from more ape-like creatures to humans. He also discussed Homo habilis, okay? Well, Ian Tattersall calls habilis a wastebasket taxon, little more than a convenient recipient for a motley assortment of hominin fossils. And he believes more than one hominid species is represented in habilis. And this is because Homo habilis is extremely fragmentary and low quality. The existence of artificial species is a real problem for the fossil record. He brought up Sediba. Sediba is the same thing. What we see are artificial species, chimeric species. Habilis is made up of bone fragments, partial skulls, and low quality, low confidence evidence. The defining specimen, for example, of habilis is nothing but a bunch of pathetic fragments. The stone tools that are often found with these bones obviously belong to humans, for one. They have also discovered a human-made shelter alongside habilis bones, okay? Humans coexisted with ape types. We see significant overlap, and oftentimes their bones are intermingled. Considering what was found outside the stone hut when you look into habilis and inside the stone hut, it is clear that what we're looking at is the fact that humans were living in this area, and so humans made the tools. Nomadic people today actually build the same types of stone huts that were discovered around habilis. And the evidence suggests Homo erectus and the Australopithecines lived at the same time. We see significant overlap, as I indicated. We can find evidence that what we are looking at is tribes of humans setting up camps where they made specialized tools and also butchered and ate animals, some of those animals being apes. Humans have always done this, especially in survival situations. And the evidence pertaining to Floresiensis, Neanderthalensis, Erectus, Luzonensis, Naledi, okay, I can go on and on. Floresiensis demonstrates accelerated genetic degeneration due to founder effects and inbreeding, exactly what we would expect post-Babel. Praise, if you want to pause my timer because I want to share screen real quick. So pause my timer, praise. And I'm going to, how much time do I have, praise?
You got 2.5 minutes.
Okay. So what we see here are massive differences when it comes to for one, we've discussed the genetics, but we also have massive differences when it comes to the physiology, anatomy, okay, the bone structure when it comes to humans and chimpanzees. For example, we as humans grow extremely long hair just on the top of our heads, and primates cannot. We have melanin in our skin while apes do not. Okay. We have a subcutaneous layer of fat under our skin, and no ape does. Humans have the same number of vertebra and ribs, and only humans have a forehead, nose bone, and parabolic jaw with toes that align. There are many features that separate us from apes. Okay. In afarensis, here's here's Ardipithecus ramidus. We see that Ardi consisted of scattered fragmentary bone pieces. Okay, a lot of these are due to reconstructions, digital reconstructions. Ardi, when you consider all the evidence, looks exactly very close to a bonobo. And you can see quote after quote from these paleoanthropologists that point out the fact that human evolution is confusing, and all new discoveries make things more confusing. It's a mess. And all of your so-called icons, okay, when you consider their best so-called transitionals, you can see here Ardi, Lucy, Sadiba, Habilis, all very, very incomplete, highly fragmented, okay? But when we actually have the data, like we do with Neanderthalensis, Erectus, okay, we can clearly see that what we are looking at is humans, okay? It is safe to consider all the lines of evidence that Jackson point to. What we're looking at, we can easily categorize it in what? Either fully human, degenerated human, fully ape, degenerated ape. Okay, we have Homo erectus here who had evidence for being a sailor, purposeful navigation, was able to speak. This speaks of intelligence. Even today, we see a ton of variation in humans. Okay, you can see the story changing over and over again as the years go on, and the more data that they find. For example, in this chart, you see a bush. It's a bushy mess with no clear transition from ape-like creature to man. Okay, and as you can see, it's low quality. I mean, with Lucy, afarensis, they find this, and they make this. Okay, you've got Lucy with a kid.
You can wrap it up so we can get into our second round of rebuttals.
How much time do I have, Praise?
You can wrap it up.
Okay, so this is the last thing I'll say, and the next rebuttal I'll cover some more stuff. As you can see here, the fingers of Lucy's kind were curved for tree life, and they also discovered a vertebra that belonged to an extinct baboon, and it took them over 40 years to discover this. This is why the fossil record is extremely low quality and low confidence science. Genetics is key to answering this question of ancestry. I yield.
Awesome. So that concludes our first round of rebuttals. Now we're going to go to the second round. It's 5 minutes. It's going to be a little quicker. So if you're prepared to have a little quicker rebuttal, that'll be awesome. So whenever you're ready, Jackson, I'm going to start the timer for your 5-minute rebuttal.
All right, I'll go ahead. You mentioned Neanderthals by definition should be the same species because they interbreed, but that's not necessarily true. That is usually the definition of a species, but there are instances of fertile hybrids from 2 different species. Now, from Lucy and her species Afarensis, I did say that most of the fossils are known from fragments, just even individual bones. But there are at least 3 that I know of that are semi-complete that show a good amount of detail between the 3 of them. Now, as for how hard it is to find fossils of these hominids, it took one site, it took them 10 years to find one piece of jawbone. 10 years. So it's not easy to put this stuff together. And you mentioned that the Lucy's skull is fragmentary, which it is, but they've found a male Afarensis skull, which is mostly complete, that shows great detail of what the skull looks like. Now, how do I explain orphan genes? I think was the question. Well, it's not really a problem. De novo genes can arise and be explained just as between generations and getting passed on. And you said that the chimp genome is 80%, which I alluded to that it's not really 99%, but the regions that code for proteins are 99%, so it's not really dishonest to say that we're 99% similar. And Australopithecine arms, you're right, they had curved fingers, they had locking wrists, they had robust shoulders, but their skulls were very ape-like, but pretty much from the neck down, they were very human-like, and I think that's the perfect definition of a transition, to have traits of both lineages. And what came before Lucy, her species Africanus, was Anamensis, which isn't as well known, but and you mentioned that there are some artificial species, like they're not sure about habilis, or habilis rather. They think Rudolfensis may be the same as habilis. They're, again, not 100% sure. Most scientists think they're two separate species. And you're right, it is confusing to make sense of these species. The more they find, the more confusing it seems to get. Zediva and Naledi are good examples. But overlap doesn't necessarily mean they aren't ancestors. Like two different populations of the same species, one can progress and live on, one can kind of go another way and exist at the same time. That makes sense. Now, you showed a picture of Lucy as a knuckle walker. Her hips, her femurs, her knees, her feet, everything about it. Well, she didn't have feet, but her species feet. Everything about it says that it walked upright. It wasn't a knuckle walker. Now, the erectus brains, you mentioned they were small, which was true in the beginning for when they lived in Africa and briefly ventured into Europe, or when they first ventured into Europe. But as they progressed eastward, their brains developed into 1,300 cc's, which is very close to the modern size. Now, Zediva, you said, was very fragmentary, but it's known from two pretty well complete skeletons, and Prometheus is known from a very complete skeleton, exceptionally complete. Let me see. And there are differences in apes, and that would be expected. We're not going to be exactly like them. But the toes of the Australopithecines, or most of them anyway, seem to have aligned with the big toes seem to have aligned with the smaller toes. And that's just more evidence of a transition, I think. An ape-like creature having human-like feet. But I'll agree with you, it is a very confusing lineage and hard to make sense of. It's not really the best transitional fossil line that I would choose, but it is evidence nonetheless. So with that, I yield back.
Praise.
Oops, I'm sorry. Yeah, on mute. But yeah, perfect. You were 15 seconds to spare, Jackson. Great job.
Right.
So now we'll hand it over to SFT for his 5-minute rebuttal, second round. And then we're going to go into 25-minute open discussion. Yeah. So whenever you're ready, SFT, I'll start the timer.
All right. Yeah, you can start it now. So many things to address. I could probably go on for 10 hours. I'll start here because Jackson's spending a lot of time on Lucy. Okay, afarensis. Well, even admitted by Lee Berger. Okay, Lee Berger has admitted that Australopithecus afarensis is a chimeric species. It is a jumble of bones from multiple species. Even Donald Johanson has reported at one point that at least 2 different species from 2 different genera were represented in his bones. Some fossils even looking exactly like human fossils. I'll tell you what happened, okay? And this is a fact. Due to the extensive range of differences seen in the bones attributed to the Australopithecines, including Afarensis and Africanus, what they invoked was called sexual dimorphism. This is simply the physical differences in males and females of the same species, okay? So paleoanthropologists claim that the larger human-looking bones belong to larger male Australopithecines, while the smaller ape-looking bones belong to females that lived in the trees. And this is why Jackson can focus on those bones, which, by the way, we don't have any genetics. Fossils and bones are not inherited sperm and egg. Geography is not inherited sperm and egg. It's the genetics, the genetic data that confirms independent origins. For example, when it comes to genetic degeneration, when it comes to Y chromosomal data, mitochondrial DNA data, but we'll get more into that a little bit later, okay? So people like Jackson, they can look to the larger bones or the bones that are actually human, since Lee Berger himself admits that Afarensis is a chimeric species, and point to those bones and say, see, Lucy had some human-like qualities, when in fact, what they're looking at are human bones, okay? Everything in human evolution is contested, and he agreed with this. And even this whole sexual dimorphism that is invoked is not a 100% agreed upon claim. Now, many proponents of human evolution have conveyed disagreement with pretty much everything in terms of human origins, okay? And the original assessment that Afarensis was a jumble of mixed bones, including human and ape bones, is clearly more accurate, okay? To put it simple, arboreals like to emphasize the bones that have been called Lucy's kind that look very apish, while others will emphasize more of the isolated bones, because remember, the fossil record is extremely fragmentary, and you find these isolated bones that look human and say this isolated bone is more human-like, and therefore Lucy walked upright like modern humans, when in fact, this is all better explained by the chimeric nature of Afarensis, these artificial species. Christopher Root, for example, co-author of Contested Bones, points out that we have never seen a case of sexual dimorphism where the male locomotes so much differently than the females. This is the nature of chimeric species and the mixing of these bones. I'm not saying it's done deliberate, okay? This is oftentimes accidental. Now, I want to share screen real quick and go over a couple more slides. If you can just stop my timer real quick. And right here. Okay, so when it comes to Sediba, okay, we have many paleoanthropologists. Here's a paper here. We have paleoanthropologists that have pointed out the fact that Sediba may be an artificial species, meaning the accidental mixture of human and non-human bones. Sediba and habilis being artificial constructs because they consist of a loose collection of human and ape bones. Ella Bin, for example, and Yoel Rack hold this view based on the lumbar vertebra and jaw bones that are derived from two different genera. Okay, and yes, you do often find Sediba with some articulated bones, but this is the point, because Sediba has been described as anatomical contradictory. For example, upper limb and shoulder anatomy are just like arboreal dwelling orangutans, and then they've got hands indistinguishable from modern humans. You see a paper here, human missing link fossils may be jumble of species. So this is what we see. For example, we also have evidence for human footprints. Okay, we got the Laetoli footprints. Well, years ago, Mary Leakey discovered footprints that are indistinguishable from modern human footprints. As a matter of fact, if these footprints were discovered today, nobody would question these footprints. They would all concede. They would all conclude they were made by barefooted human beings. Evolutionists have dated these footprints to roughly 3 or 4 million years old, and therefore, because of their circular reasoning, they cannot conclude these were made by humans. What's that, Praise?
Go ahead and wrap it up.
Okay. We also see the same thing in Crete recently, anatomically modern looking human footprints. And last thing I'll say, a recent finding here that 3 different early humans, Australopithecus, Paranthropus, and the earliest known Homo erectus, appear to have lived at the same time in the same place. The point is, overlap is what we would expect from a biblical model. And that's exactly what we find. I yield.
Great job. So now we're going to go into the 25-minute open discussion. You guys can just bounce off that if you wanted to, and just SFT's last point, Jackson. It's up to you guys how you want to do that. But whenever you guys, I'll start the timer.
All right. So you say Afarensis is a chimeric species, but I think that doesn't really work, given that there are three that I know of, three partial skeletons found at the same different sites, and all together, mostly together. Lucy was found in a 50 square foot area, I believe it was. I mean, it may be meters, but something like that. But the other two were found much closer together, that's from what I've read. So,
right. So,
what do you think of that?
yeah. So good points. You would agree that Lucy herself was found in hundreds of pieces, though, over this area, right? Very fragmented.
Mm-hmm.
And Lee Berger, I'm not sure if you're familiar with this, but even Lee Berger has pointed out that Australopithecus afarensis may indeed be a chimeric species, as in it is a jumble of bones from multiple species. And Donald Johanson, at one point, has made the claim that we are looking at least two different species from two different genera. Okay, and he was pointing this out in the bone collection that he had, with some of those fossils even looking exactly like human fossils. For example, I'll point to I'll share screen real quick. The Laetoli footprints, which I'd be curious as to your thoughts, where we've got footprints here in Laetoli that are indistinguishable from human footprints. So what is your position on this? And why not conclude that, since, as you can see here, here's a paper by Donald Johanson verifying what I was saying here, this collection suggests that Homo and Australopithecus coexisted as early as 3 million years ago. So why is this not a human footprint, since that's exactly what it looks like? Yeah, what are your thoughts on that, Jackson?
Well, I think radiometric dating kind of shows that it couldn't be a human. I know you guys don't really give a lot of credence to radiometric dating, but also the foot structure of what we know to be afarensis, not from Lucy, obviously, but the fourth metatarsal, the heel, shows that it probably didn't have a divergent big toe, and it had an arch and a striking heel for bipedal walking. So I think the Laetoli footprints may have been afarensis. It may have been another species, but I think they weren't human.
But do you find that circular, in that we have paleoanthropologists that are pointing out, for example, here's the footprint, here's a human footprint. We see how obviously divergent it is from a chimpanzee footprint. We don't really know what Lucy's feet look like, because what we have found is very fragmentary. So therefore, it seems to me that this argument is circular, Jackson, because you pointed to dating methods, which I'll go to, which we know are based on many assumptions, many of them unprovable assumptions. And I've got a slide here that goes over numerous sources from rocks of known age, as in we know when the volcano erupted, the samples were collected, and we get dates of millions and millions of years old, even though these rocks were just formed. So the point is, why would we trust dates for rocks of known age, right, or unknown age, when we have rocks of known age that are giving us inaccurate dates? I know this isn't a debate about dating methods, but my point is, your reasoning seems to be circular, because you're pointing to, well, it's dated far too old to actually be humans, instead of concluding the obvious. Yeah, go ahead.
Well, I know that the ICR did a test, the rate test, the rate study, and they used potassium argon dating to date Mount St. Helens, which was the incorrect method for that. It even said that Geochron Labs they use to date the sediment says anything below 2 million years, it's going to give you wild answers. And they did get multiple different answer ages back. And they think that's probably from, what's it called? Ion noise, I think it's called, from the mass spectrometer. So I think you can dismiss the ICR's research on that. And I know there are instances of multiple dating methods aligning on one sample and coming to within a few percent of the same age, which is very unlikely if it didn't actually work.
So what's nice about the rate project and the rate scientists is they took samples from the same rock layers and subjected them to the dating methods all at once. And what they found were many discordant dates, even with the isochron method. And these were repeatable. Okay. And they also did this with C14 carbon dating. And what they found was carbon 14 in diamonds, in fossilized wood, in dinosaur fossils, in coal, which means these samples cannot be millions of years old or billions of years old in terms of diamonds, because obviously we're finding C14 in these samples when given the half-life of carbon 14 and the fact that carbon decays so quickly, then obviously these rocks and fossils, okay, that the uniformitarians and evolutionists are pointing to as millions of years old, that kind of goes back to your argument here, which I believe is circular based. These samples clearly cannot be millions to hundreds of millions of years old because they had radiocarbon in them. And you can comment on that, but I did want to ask you this question. I'll get this question out first because I kind of want to bring us back to more of the topic, but I also don't want to leave that hanging. So you can definitely address that. But I wanted to ask you the question in terms of genetics and the fact that we have massive differences, which I discussed in the opening statement and my rebuttal, but a huge difference has to do with the Y chromosome, where we've got, I'm not sure if you're familiar with this paper, chimpanzee and human Y chromosomes are remarkably divergent in structure and gene content. When you consider overall size differences and architecture, Jackson, we're looking at a Y chromosome that is less than 35% dissimilar between humans and chimpanzees. So if they are our closest cousin and we share a common ancestor with the chimpanzee, how do you explain those huge differences? And take your time, take equal time, Jackson, go ahead.
Well, the differences in the Y chromosome, I know the Y chromosome mutates a bit faster than the other chromosomes, I believe. And as for carbon 14, again, it was likely due to contamination because uranium and thorium decay and can produce carbon 14 in the surroundings. So the fact that it was there doesn't necessarily mean it was ancient. So that's pretty much my comment on that.
Yeah, so what I would say is the rate scientists, what's good is they went into the rate project, okay? They went into it making predictions. And what they discovered were fulfillment of those predictions. For example, they even found helium and zircon crystals. We know helium is incredibly slippery, and yet we find helium and zircon crystals that evolutionists say are billions of years old. There should be no helium left. Same with the radiocarbon. So you point to contamination, but what's good is the rate scientists were diligent, and they subjected these samples to harsh treatments that would get rid of any of these contaminants, and therefore what they're looking at are blank samples. Okay, so they've looked at contamination, they've looked at other reasons that the evolutionists might point to in a way to rescue uniformitarianism in deep time. And these results are repeated. These are repeatable. They're testable. We're finding radiocarbon oftentimes 100 times above the limit the detectable limit that should be there. When it comes to the Y chromosome, the problem is in, let's say, 6 million years, you've got such a waiting time problem to account for massive differences in the Y chromosome. The Y chromosome is uniparentally non-recombining DNA, okay? It is inherited from our fathers. The problem is it should be relatively more stable due to the fact that it's non-recombining. But as you pointed out, the Y chromosome also mutates fast. So here's a problem. If you are going to look to that argument to explain the dissimilarity between human and chimpanzee Y chromosome, well, we have a lot of papers telling us now that since the Y chromosome mutates faster than ever predicted, there are only a few mutations in the Y chromosome separating any two people on the planet. When it comes to men, every single one of our Y chromosomes are nearly identical. So how do you have a rapidly changing Y chromosome with incredibly low variation in humans today? Every single human, every single male Y chromosome is nearly identical, and they can be traced back to one Y chromosomal ancestor just 4,500 years ago, as you can see here. So how would you address that if you're going to look to the rapidly changing Y chromosome? Why do we see very low Y chromosomal variation in the male Y chromosome, Jackson?
Well, I would say it's because humans are a relatively young species, no more than 200,000 or 300,000 years old. So not really much time relative to every other animal to acquire the sufficient mutations to look as different as you would expect, I would think.
Well, here's the person. Oh yeah. I'm sorry. Go ahead, Jack.
As for the helium and the zircons, they tested a sample that was taken out in the 70s, I believe, and it was taken out from a volcanic area. And from what I've read, the compression of the zircon can hold in helium for long periods of time, and the system isn't well understood in the first place to really assess the age from that, based on what I've read. And now some form of helium will decay past that point, but some forms of helium, like helium-3, are stable over long periods of time. Now, what was your next point?
So my point would be, and we can touch on that in a second, okay?
Okay.
My point here would be, here's a Y chromosomal family tree, okay? And this is based on worldwide Y chromosomal diversity. We're not looking at hundreds of thousands of years here, okay? We're not looking at tens of thousands of mutations to take your Y chromosomal Adam ancestor into the deep time that you need it. No, all we're seeing is a few mutations in the Y chromosome that separates any two people. Okay, let me ask you this question, okay, just to kind of steer this into relatively something new, okay? We don't only have low variation in the Y chromosome and the mitochondrial DNA. We have low genetic diversity in general, okay? Let me ask you, Jackson, what happens when you introduce a new mutation into the genome? What does that add?
Usually it has no effect. Most mutations are neutral. Some are harmful and some are beneficial.
Well, we'll get into we will get into that, because I've got a ton of quotes where population geneticists that are evolutionists admit that no mutation can truly be neutral. Okay, they refer to these as effectively neutral because they have only very subtle effects on fitness. They're only slightly deleterious, kind of like rust on a car, okay, that they build up over time, but they're still unselectable, and therefore they're subject to genetic drift. So they accumulate and degenerate. But that's separate, that we can get into in a bit. But just in general, mutations. When you introduce mutations, what do they add?
Well, it depends
especially
on the
according
mutation,
to the evolutionary model. What's that?
I guess it would depend on the mutation. Like there's a de novo mutation in cod that allowed for them to live in colder water for their blood not to freeze.
Well, how do evolutionists explain the origin of genetic variation and genetic diversity?
Well, what do you mean? Like from the
Like,
beginning
what
of
are the
time?
mechanisms? Like for example, we as creationists, we look to what's called the created heterozygosity hypothesis, the design diversity hypothesis, that suggests that the vast majority of nuclear DNA differences are the result of created heterozygosity. Heterozygosity just means a state of DNA diversity, while the evolutionary community explains the vast majority of DNA differences as mutations, right?
Mm-hmm.
Okay.
Yeah.
So when you have a mutation, according to the evolutionary model, what are you adding?
Well, oftentimes new information. I mean, there are mutations that de novo mutations, duplication mutations that add material or new information to the genome.
So in other words, when you add a mutation, you're adding genetic diversity to the population.
Well, not necessarily diversity, but information, yes.
Well, the evolutionists explain the vast majority of DNA differences, if not all DNA differences, and the origin of genetic diversity as a result of mutations over time. Okay, so a mutation adds something because it is adding something that was not there before. So I'm just going to go right to my point then. Okay. If you have the Australopithecines evolving for millions of years and you got Homo habilis, okay, and then you've got what I find interesting is I've got a couple articles that I can show you later where Australopithecus sediba has been ousted out of the human line due to some contradictions that they discovered in 2019. So let's just throw away sediba for now. We've got habilis, and then we've got erectus. And then you got erectus living on the planet, covering part of the globe, Africa, Eurasia, for millions of years, where they are also accumulating mutations. So we should have today in humans high levels of genetic diversity. But what we find is low levels of genetic diversity. Why is that the case?
Well, I think there's higher genetic diversity than you're letting on. But there was a genetic bottleneck in the past, about several thousand individuals, as evidenced by mitochondrial and nuclear DNA. So I think they can explain a lot of the low genetic diversity.
So here's the problem with that. And I'm not going to screen share again. We'll just have a free-flowing dialogue. But I do have papers in front of me that go over the fact that we do have extremely low genetic variation. As in every single human being, we're all 99.99% similar.
Yeah, that's true.
So that was a problem for the evolutionary community. And you gave the correct answer, according to the story, is in the past, there was a genetic bottleneck that reduced the population at the time to roughly between 2 and 10,000. How many years ago would you say this was, according to your model?
I believe it was about 80,000 years ago.
Okay, so say you've got...
That was the case.
Okay, so say you've got, because here's the thing, there's differing opinions as to when this bottleneck took place. Some people say, a couple 100,000 years ago, maybe 70,000 years ago with the Toba extinction, or some even say millions of years ago with erectus. But let's go with 80,000, okay? So here's the problem with that, is you've got this bottleneck that drops the human population to between, let's say, 2 and 10,000 years. Well, you would need thousands of years. You would need more than one generation to reduce those levels of heterozygosity and increase levels of homozygosity, okay? Therefore, you have generations and generations of what, Jackson? Of inbreeding. And is inbreeding a good thing?
Well, no, but by your model, inbreeding is the only option also. I mean, inbreeding from a point of a few thousand is better than inbreeding from a point of 2.
But here's a question I'm going to ask you. In light of what we've been talking about, what is the major difference in terms of the origin of genetic diversity between the evolution model and the creation model? What's the difference?
Well, I think the creation model says all the genetic diversity was put in at once, in Adam and Eve, and the evolution model would say new genes can arise.
So the creationists would say that the origin of genetic variation for the most part is due to design diversity, while the evolutionists would look to mutations as the source for genetic variation. So here's the problem. We don't explain every single DNA difference as being the result of mutations over time. So Adam and Eve would have had pre-existing functional heterozygosity with no mutations. And then at the flood, you might have some mutations built up, but still what you're looking at is mostly created variation, okay? It's the mutations that are in recessive form that are manifested during inbreeding that lead to accelerated genetic degeneration. It is the evolutionist that proposes mutations as the source for genetic variation and genetic diversity. So you've got this bottleneck roughly 80,000 years ago where you've got all of these mutations, these deleterious mutations that are now coming to the forefront, leading to rapid genetic degeneration. Because it's not one generation, it's not two generations, it's many generations of this inbreeding in order to reduce levels of genetic diversity. Are you familiar with the bottleneck today that is seen in cheetahs?
Yeah, cheetahs and Tasmanian devils and a few others.
So we see in cheetahs how disastrous this is. There's about 7,000 cheetahs today. Birth defects are increasing, and the cheetahs are essentially going extinct. Conservationists are extremely worried about them. So what we see with the cheetahs means that this hypothetical out of Africa bottleneck would have also been disastrous. So my question to you is, how is that bottleneck that's employed to explain the low levels of genetic diversity, which, by the way, is what we would expect starting from Adam and Eve, that automatically would restrict diversity. How is that hypothetical bottleneck associated with massive problems with inbreeding? How is that feasible, would you say?
Well, biologists say, or zoologists say, that the minimum population for optimum genetic diversity is about 100 individuals. So we would have had a few thousand to work with, so not catastrophic. But I also want to say, what you said, Adam and Eve wouldn't have been subjected to mutations. Is that right?
At creation, they wouldn't have had mutations, right?
Yeah, but after the fall, would they have?
Well, the fall would have introduced mutations because mutations are essentially copying errors.
Yeah.
So due to mutation accumulation, we inherit roughly 100 new mutations per person per individual. Over time, Adam and Eve's descendants would have had mutations. Yes.
Yeah, I think that's a way less workable model, just because you're starting from 2 and you're just inbreeding immediately from their children. And any gene is going to get expressed and passed on, and it's just going to be kind of a total mess.
Well, let me tell you why. So it wouldn't be a problem from our model, because according to the independent origins model, inbreeding would have been a non-issue since there would have been no deleterious mutations. And since mutations can be compared to typographical errors in a text, and during instances of inbreeding, those mutations come to the forefront, leading to disease and damage. But if there are no mutations to come to the forefront to lead to that type of disease and damage, then we wouldn't have an issue. But that brings me to the point you made on neutral mutations. And you said that a mutation can not be deleterious. I'm trying to find, I got 1,000 slides here, even more, 1,000,000 slides. So here's a source, okay, from, because you said it was a young earth creationist invention or talking point. But we have population geneticists here that point out, for example, right here, Higgins and Lynch, we find that the accumulation of new mildly deleterious mutations, okay, so mildly deleterious, and this one here, Crow's concern. The typical mutation is very mild. It usually has no overt effect, but shows up as a small decrease in viability and fertility. And the last one I'll show you is right here with Kondrashov. He points to very slightly deleterious mutations. There's also another quote that I can show you where a population geneticist points out the fact that any mutation must have at least some effect on genotype. So how do you explain if those mutations are only mildly deleterious and they build up, Jackson, which means they're unselectable? What
All
type
right,
of
guys.
selection can remove?
We got to start wrapping it up. We just finished the 25-minute mark for the open discussion. So we're going to go into the speed rounds and then your conclusions. So that was a amicable and enthralling discussion. Good job, guys.
Why don't we?
Yeah, I think it was good.
Yeah, I want to give Jackson the floor here, though, just to answer that. I don't want to end it there. So, Jackson, so the question would be, what type of selection can you put forth that would remove so many low impact deleterious mutations? And then take your time responding.
Many of the neutral or slightly harmful, as you would call it, mutations would build up. But the harmful ones, the actual harmful ones would be selected out by natural selection. And I want to bring up the Tuatara, not quite a lizard, but it is lizard-like from New Zealand. It currently has the highest mutation rate of any animal, acquiring new information, as well as having some of the neutral mutations. Fastest evolving animal on the planet at the moment. So it shows that the genome can kind of grow like that and not really have negative consequences. Also, you mentioned cheetahs. I didn't go into that, but cheetahs, it was thought that their population was only down to a few individuals at one point. That's why they're having such problems now. But humans had a few thousand individuals, so much more to work with. Now, starting from Adam and Eve, again, that would have been pretty problematic for genetics. And with that, we can go to the next subject if you want.
Okay, I appreciate that. Praise, are we doing okay? So we're going to do 2-minute responses now, Praise, to wrap up the discussion. And then we're going to go into 5-minute concluding statements. Since we did hit the hour-and-a-half mark, we agreed beforehand. We're going to have a 2-minute intermission right before the 2-minute intervals and then 5-minute intervals, and give you a chance to gather any questions that are coming through. I know we've got a big chat. And here we go, Praise. 2-minute break.
Are we on break?
Yes.
Okay. Can the audience hear us or no?
Yep. So you have to go hit the bathroom. Go do it now.
Okay. I'm good. I'll just sit here.
Okay, it looks like we are good. Praise, we're going into a 2-minute response. Who starts, Jackson or myself?
So it would be you because you opened up, or I mean Jackson opened up, so it'll be you in the speed round.
Okay, so I start the 2 minutes?
Yes, I'll start the 2 minutes on your first word.
Okay. Just 1 second. I want to share screen. And okay. So here's the problem. To kind of give some final thoughts on the open discussion is when it comes to the evolutionary model, they explain the origin of genetic variation as being the result of mutations. Since we know mutations are destruction, okay, they lead to disease. And as we can see from many population geneticists, they're acknowledging that most mutations are effectively neutral. Okay, they are unselectable as they build up in populations over time, 100 new mutations per person per generation, and they lead to genetic damage. So that bottleneck that the evolutionists look to as a rescue device to explain why humans today have such low genetic diversity, consistent with the Adam and Eve starting point, is not even remotely feasible, because it would lead to rapid and accelerated genetic degeneration. We see that in the cheetahs. We're not going to suddenly expect these 7,000 highly degenerated cheetahs today to spread out into all parts of the globe, seizing dominion over the planet. Of course not. But the evolutionists want us to believe that after this bottleneck that would have been highly damaging, humans spread out in all parts of the world, seizing dominion over the planet. It just wouldn't happen. They would be highly degenerated. It wouldn't be possible. So it is a fact. Here's a source. Mutations are the source of genetic variation for the evolutionists. But with the icons that Jackson points to, we find this problem of mutation accumulation explaining a lot of the anomalous features that evolutionists like to point to as transitional or anomalous. For example, Homo floresiensis, Neanderthalensis erectus, Naledi. Okay, this is what we find in the fossil record, is where we find inbreeding, we find pathology and disease and reductive evolution. Last thing I'll say, this is consistent with the post-Babel dispersal, and it's inconsistent with human evolution, ape to man evolution. Go ahead.
Go ahead, Jackson. We'll start your 2-minute timer, and I'll give you a 20-second warning on your first word. Go ahead.
Well, I think the genetic bottleneck argument is far worse for the creationist point of view. Not only do you start with Adam and Eve, but then 2,000 years later, you have Noah and his family. 8 individuals, 3 breeding pairs. Or if did Noah have a kid after the flood? I don't know. But anyway, let's say 4 breeding pairs. So it's reduced again. So you're inbreeding yet again after Adam and Eve. And not only that, but all animals on Earth would have been highly inbred. It's not just cheetahs or Tasmanian devils that would have shown evidence of this. Every animal should be showing signs of having reproductive issues. I think I read that 90% of cheetah cubs don't make it to adulthood. That should be happening to all animals if that were the case. Now, so I think the creation model for genetic diversity just doesn't really work, and I think an evolutionary model is definitely more workable. And with that, you can respond.
Actually, you have your conclusion, your concluding remark. It's set for 5 minutes, but you guys did go under your expected time, so you can add a little bit in there. Maybe 5 to. I think it's set for. Let's see here.
Yeah, maybe
5 minutes. You can go 5 to 7 minutes is fine.
Okay, so since Jackson started the debate, Jackson, take the floor. You have between 5 and 7 minutes for your concluding statement.
All right. Well, I think we both did pretty good in this debate. I think it went really well. I know I enjoyed it. We should do it again sometime. But I think that fossils, genetics are really on my side when it comes to this. And I don't know if I need the full 5 to 7 minutes, but yeah, I just really want to say that I've enjoyed being on here, and it was fun. And you made some good points, standing for truth. You're very competent, you're knowledgeable. So I have a lot of respect for you. Just want to say that. And honestly, that's all I have to say as a closing statement.
Awesome. I appreciate that, Jackson. Okay, Praise, I guess you can start my timer in 1 second here. And I'll share screen in a second because I do want to mention one thing. So you can start my timer now, Praise. Jackson, yes, thanks so much for doing this debate. I
Yeah,
respect
you're
you as
welcome.
well. This was very cordial, and
Yeah.
time has flown by. I can't believe it's already been an hour and a half. It's been fast paced,
Yeah, that's fine
and
by.
there's just so many topics to discuss and so many lines of evidence we could discuss that let's definitely have another one of these in the future. I know we're working on setting you up with debates with Ken Hovind, the Film Free. So this should be fun. So in terms of my concluding statement, then I'll just address a couple things that I think we're left hanging, because when it comes to the fossil record in general, okay, when we look at humans in the fossil record, we see no evidence of hundreds of thousands of years of human existence, okay? And given the evolutionary model, we can assume there were about 1 million. Let's look to Homo erectus, the Australopithecines, Homo habilis, Homo erectus, who lived apparently across Eurasia and Africa, okay? Essentially, they handled a massive distribution of the earth. So if we put some numbers on it, let's say Homo erectus's average generation time is 20 years, let's say, okay? That means about every 20 years, you have another 1 million dead bodies. And so after 1 million years, we have over 20 billion dead people, okay? Point is, maybe it's not quite that high, but we've got a lot of dead people, a lot of dead erectus. And the point is, this is millions to billions of people of which we have no evidence ever existed. Even the proponents of evolution would agree that erectus had primitive technology. They would have had campsites, they would have had villages, they would have had fire. We have a ton of evidence for their existence, and yet every single erectus fossil that we have can be fit in the trunk of a car. So where's the evidence that human evolution even occurred? Millions of years of human evolution. I want to share screen real quick here as well. Let me know when we reach the 5-minute mark, so then I can kind of wrap up from there. Okay, so when we look to because we discussed dating methods. See, here's the thing. Time is always looked to by the evolutionists as the hero of the plot for the story. But what we know about mutation accumulation and genetic degeneration, okay, time is not the hero of the plot, because the more time you have, the more extinction and degeneration you have. So they can have the time. But what we see here is hundreds of physical processes that set limits on the age of the earth. And we discussed just a few, the C14, we discussed helium, we discussed the discordant dates in dating methods, as well as in isochron dating, which they like to look to. We've also seen a lot of discussion pertaining to Lucy, and what we can see here based on her skull, based on the hundreds of pieces that were scattered across this 3-meter area of a hillside, the fact that what we're looking at is highly fragmentary data means you can have all sorts of interpretations and stories with these bones, okay? Bones found in the dirt, fossils, I always point out, they can oftentimes deceive us, okay? Because they don't come with genetics oftentimes. And the problem is, we know that today, many species have more variation within themselves than between species. So when we're looking at a fossil without genetics, without genetic data, then it's oftentimes difficult to conclude, is this the result of homology or convergent evolution? Convergent evolution is the independent origin, apparently, or evolution of highly sophisticated and complex traits that we have no evidence could evolve in the first place. But yet evolutionists say that something like the eye has evolved dozens and dozens of times. Okay, anytime convergent evolution is invoked, it is being invoked, and it is an admittance of the lack of uniqueness in the phylogenetic tree, okay? So we've got Lucy here, semicircular canals, better suited for walking on all fours. I pointed out the fact that they found a baboon bone, okay? It took 40 years. Homo habilis, evidence that Homo habilis is a chimeric species. And the problem with the taxonomically restricted genes, the orphan genes, they're highly essential, and you would need an extraordinarily amount of mutations that are beneficial, okay, in sequence to evolve something so essential. But yet what we see...
You're at 5 minutes, SFT.
Thank you. I'll start winding it down. What we see with the presence of these genetic sequences that are functional and essential is no apparent relatives, as in it's exactly what we would expect to find according to the independent origins model. And the last thing I want to cover, so I want to make sure I cover everything, is the retrotransposons, which he spent some time talking about. So the question is, when it comes to these retrotransposons, is are they really the ancient remnants of viral infections, or are they created units of DNA function? Because Jackson made the argument in his rebuttal that we still share more ERVs with the chimpanzee and the humans. Humans and chimpanzees share a lot of these viral-like elements than with other species. But that's exactly what we'd expect to find, is if these were created, okay, if these variation-inducing genetic elements are there for a reason, and that's what we find, paper after paper, retroviral promoters in the human genome, then of course we're going to share more, because we predict function, because we predict that these similarities are there for functional purposes. And that's what we see in the design world. Engineers build in homologous patterns. Engineers build in nested hierarchical patterns. Engineers even build transitional-like forms. For example, an amphibious assault vehicle and some of these crossover SUVs. So what we predict is function. What we find is function. Scientists identify new beneficial function of endogenous retroviruses in immune response. The last thing that I'll touch on, I don't want to leave hanging, was the chromosome 2 fusion that he talked about. Well, what we find is that the chromosome 2 fusion, the so-called site, is overlapped by a highly functional and essential gene. So Jackson, the problem is you're not going to get a highly functional gene by slamming together two chromosomes. The site is very degenerate, and there's lack of evidence for the cryptic centromeric site, as well as a satellite DNA problem that maybe we can get into in a future show. This would be a paper I'd highly recommend: The Evolution of African Great Apes, Subtelomeric Heterochromatin and the Fusion of Human Chromosome 2. Chimpanzee and gorilla chromosomes differ from human chromosomes by the presence of large blocks of subterminal heterochromatin thought to be comprised primarily of arrays of tandem satellite sequence. Why are these not found? Why is the evidence seem to suggest there was no fusion event to begin with? And great debate, lots of fun, great chat today, and I hope we can do this again in the future. Jackson, thanks so much. I conclude my statement.
Yeah.
Awesome, guys. That was a very respectful and stimulating debate. Great job. Both sides made great points, and yeah, it was impressive. But we're going to head into the question and answer period with the super chats. We got quite a few, so we're just going to jump right into them. So the way we format the Q&As and super chats is whoever was addressed to, they get to finish the final comment on that comment, whatever question or comment, whatever it is. So this is from Cheryl for $5. The comment, she says, "This is in the name of Raymond or Ramen Goiza Dingo."
Ramen, the super sticker master.
"Who could not be here. We are thinking of you!" Exclamation, exclamation mark. And I actually lost that super chat, so I appreciate Cheryl giving it back. So thank you, Cheryl.
I appreciate that super chat, Cheryl.
So we'll go to a super chat from One Cow Stampede for $10, and thank you for your super chat. And then we'll go to the next super chat from Landon Freeman from $2. This is addressed to you, Jackson. What evidence would change your mind?
About evolution or about God, or what specifically?
Either. Yeah, he didn't specify it, but probably the debate, probably evolution. Yeah.
Evolution, yeah. Well, if you found, say, a bunny in the Precambrian dated rock, that would convince me pretty quick that something's wrong. Or if you could find another animal that had a closer DNA coding than a chimp to a human, like say a pine tree had a closer genetic match to humans than the chimps did, that would change my mind pretty quick also. So it's evidence like that that would totally turn evolution on its head.
Gotcha. So SFT, do you want to say something to add to that, or you want to go to the next? This is the next super chat.
Yeah, can you repeat the question, brother?
What evidence would change your mind? So if it's applied to you, what would change your mind about creation or intelligent design?
Just like I write in my books, there's two predictions, okay? Evolutionists predict, if this is all just a long history of descent with modification and universal common ancestry, then we should find a lot of junk. We should find evolutionary leftovers. We should find genomic fossils. We should find ancient viral infections littering our genome. And yet, what we are now finding is that the vast majority of DNA elements, DNA sequences, and DNA in general is active, functional, and essential. Okay, so what would help to change my mind if that was not what we seen? If we actually did see the vast majority of DNA was junk and therefore reflective of universal common descent, then that would be convincing that banana plants and whales are related. It's just not what we see. And the last thing I'll say is this. This is why evolutionists struggle to make predictions on DNA function, while creationists expect function. So we can look to, say, the endogenous retroviruses and predict function. We can predict function in determining cell types, in gene regulation, in immune response, cell stress response, and so on and so forth. Same thing with the pseudogenes. And that's it.
So you can finish that, Jackson, if you want to respond to that really quick, a pithy statement if you want to respond to that.
Well, with the ERVs, a lot of them are highly fragmented and evidence of a long time of deletion mutations and inactivating mutations throughout time, and apparently are ancient from everything I've read on it.
But if I could ask you a question, Praise, before we move on, it's just going to be a question. We won't debate it. Have you seen a paper? Have you, for example, because what we're seeing in terms of the amazing functionality of these retrotransposons, are you familiar with any empirical evidence or any technical paper that actually shows a viral-like element, Jackson, going from non-functional to functional? Have we seen this in real time? Is this empirical?
I'm not aware of a paper like that. I haven't really researched that aspect of it. I'll be honest with you on that.
And I think that's the problem, because I've debated a biologist in the past who also looked at ERVs. And there's a number of critics that I've debated where I've constantly sought a paper like that, because it would be incredibly difficult to have a viral-like element, if it was truly negative to begin with, evolve something so essential to the genome. Because if I were to have somebody blindfolded and tell them to randomly put some brush strokes on a painting, well, they're probably going to mess up the painting as compared to improve it. And that's what we see with these viral-like sequences. If these really are insertion events over millions of years, they should corrupt as compared to make things better, especially make things better in the placenta and in embryological development. So until we see an actual paper or technical data that can show, a non-functional retroviral-like element going from non-functional to functional, then my point would be it's just not science. And ERVs shouldn't be looked to anymore by the evolutionists as evidence for common descent. And Jackson, you can have the last word on that. Go ahead.
Well, endogenous retroviruses, I would assume they're like mutations in the sense that most of them aren't going to have any effect. Some of them might have a negative effect, and some of them might have a positive effect. But other than that, I don't have much to say on that aspect of the ERVs.
Very good. So we have Jerome Super Chat SFT with a Popeye flexing emoticon. Yeah, $5, Jerome. Thank you for your support.
Thank you, Jerome. Thanks for the support, brother. I appreciate it.
Now we got two. So we got a super chat and a super sticker from Jamie. Went for $1.49 and a $5 super chat. He says, he asks, is common ancestry falsifiable, genetic similarity as is? If yes, how? If no, why is that a problem or not?
That's for me?
It's for both of you. Whoever wants to, address that. I think it's addressed to both of you guys.
Well, yeah, common ancestry would be falsifiable. Again, if you found that the genome didn't match close to chimps, it matched, say, to pigs, closer to pigs than chimps, that would turn common ancestry upside down. It would, just really anything like that would disprove it. So it is falsifiable.
for my response, I would just say it's not falsifiable because we have so many lines of evidence that have overturned universal descent, okay, universal common ancestry, especially in the fossil record where we find modern human bones, modern human footprints in areas where they should not be. Because Jackson said earlier, and it's a good point, I agree. For example, we can live at the same time as our parents and grandparents, okay? A domestic dog, although ultimately came from a wolf, can still live with wolves, right? We have wolves and we have the domestic dog living together, okay? The problem is you can't have a domestic dog living before the wolf. We can't have me living before my great-grandparents. And that's oftentimes what we find with this overlap in the fossil record when it comes to modern human-looking footprints, when it comes to the overlap we see with the Australopithecines, with Homo erectus, okay, with all of these so-called hominids. And it just better supports the post-Babel model, where you've got these people groups that migrate to different parts of the world, and we see similar anomalous features in Naledi, in the hobbits, right, Floresiensis, also in Luzonensis, where they experience island dwarfism and reductive evolution. Reductive evolution seems to be the rule, and reductive evolution is not going to take a single-celled like ancestor into a whale. So my whole point to wrap this up is I don't believe evolution is falsified, because every time a line of evidence comes about that poses a major problem to evolutionary theory, what they do is they change the story. Oh, well, we've got low genetic diversity in humans today, so there must have been some type of hypothetical out of Africa bottleneck, even though that bottleneck would have been extremely damaging genetically. So it's not feasible. So I just think it's circular. They're always going to force fit the data somewhere. They're going to adapt the story. And no, I don't think it's falsifiable. That's my answer, Praise.
Awesome, SFT. Do you want to make a quick response to that, Jackson? Go ahead.
Yeah, I don't think the young Earth creation model is really falsifiable, because, I mean, just look at the ICR and AIG statements of faith that I'm not equating you with them. I mean, I know you probably have more integrity than they do, but they say in their statement of faith that no amount of evidence would ever change their mind. And evolutionary theory does change with new evidence. And if evidence came about that totally ruined it, I think everyone would accept that it was done. But I don't think that's going to happen. There's a lot of evidence in support of it.
Great. Appreciate your answer. So we got to go to our next super chat. Steadfast and easy for $5. Thank you for your super chat. He says, or he or she says, "Ha! Bet you thought you lost me. Hee hee. Hi, fellas. God bless."
God bless you as well, brother, and thank you so much for all the amazing support from the chat. This debate's been a lot of fun, and we've had a very lively chat and a lot of good people. And Jackson, you've been a lot of fun to debate. So thanks for the super chat, guys.
Yeah, it's been fun.
Yes, it's been pleasurable to moderate as well. So here now we're into the Q&As, and I mean the questions instead of the super chats. Now we're just in the general questions. Now Landon Freeman, he says, for a question for you, Jackson. How do organisms acquire the intelligence needed to know how to use newly evolved features?
Well, I would say the brains evolve with the bodies. It's a gradual process. I'm not a biologist. I can't really answer it in detail. But the more neurologic function you have, the more your neurons are going to develop. And again, not a biologist, but that's basically my answer.
SOT, if you want to respond to that, go ahead.
Yes. Can you repeat the question real quick?
How do organisms acquire the intelligence needed to know how to use newly evolved features?
Yeah, I would say to begin with, what we see in terms of the forward-thinking mechanisms in our genome, right? Epigenetics, these amazing control systems, okay, and the genomes of living organisms. We literally have millions and millions of switches just waiting to be turned on via environment. Okay, rapid adaptation is what we see. So here's the thing, is evolution doesn't have a mind. Natural selection sees the short term, okay? Natural selection wants the organism to survive till tomorrow. It doesn't worry about getting cancer 50 years from now, okay? So natural selection doesn't have a mind. So what type of evolutionary processes can build that which requires a mind? And if evolutionists want to say epigenetics, for example, is the result of evolutionary mechanisms, then they are applying a mind to evolution, a mind to nature. And also what we have is evidence for interdependent and obviously engineered, simultaneously assembled body parts that could not come together without intelligence. And these are some slides from Genesis Apologetics, as Dr. Dan Biddle's been such a blessing, and he's given me permission to go through a lot of his slides because he does some great work. And he points out here the 5 different parts of our hearing systems work in coordination and how incredible it is. Sound waves are captured by the outer ear, which in turn wiggle 3 tiny little bones called the hammer, anvil, and stirrup, which press on the cochlea, turning into the mechanical process into a hydraulic one. Then you can just go through all of this and how all of this has to come about at once. It can't happen through slow and gradual processes. Okay, what we see in terms of the amazing design of living organisms, God said he created things very good. They had to start that way at the beginning. And of course, degeneration has been introduced since the fall, but things started off perfect. And these things had to start off perfect and perfectly designed and not built up through evolutionary processes. I yield.
Awesome, SFT. If Jackson, if you want to make a quick response to that, go ahead, man.
Yeah, that's the irreducible complexity argument in the Dover trial, I believe it was. I forget what the full name was, but Michael Behe argued that, and biologists countered with, well, a mouse trap may not be functional if you take it in its intended purpose, if you take a part out of it, but he gave an example. Well, with the spring, it can still be a tie clip. You know, these different things can serve different functions. They're not necessarily built with the goal in mind, the end result. And individual components can still have some function without the whole system in place at once. So that's my answer.
Great. And we'll go to the next question from David Neff. Coming at you, SFT. I have a question for both. Sorry. Both.
He's coming at us both then.
Yeah, that was a facepalm moment. Okay. So what evidence would change your mind? So I guess he's talking about creation for you, SFT, and evolution for you, Jackson.
You can go first if you want, SFT.
Well, I guess we've kind of answered that question earlier.
Yeah, we kind of did.
And what I pointed to, I guess I could point to a number of things, but the biggest thing would be testable predictions. What's the gold standard of science? The gold standard of science is to make testable predictions, future testable predictions. So whoever is making the best testable predictions will essentially win the war, okay? Creation versus evolution. And what we have now in 2021 is genetics, where genetics is the best way to determine ancestry. Because as I've pointed out, fossils are not inherited sperm and egg. And oftentimes you get contradictions in the fossil record. Things in the fossil record are always being overturned. Okay. We see stasis in the fossil record, and we see the fact that convergent evolution is a real problem for universal common ancestry. So if it comes down to genetics and the empirical data, who's making the best testable predictions? We've got Dr. Nathaniel Jensen, who has made testable predictions on the Y chromosome, the mitochondrial DNA. He's making testable predictions on the history of civilization. That, if true, can only be true if young earth creation is true. Okay, he's making testable predictions in terms of mutation rates in people groups where we don't yet know their mutation rate, like the Khoisan peoples. And we have prediction after prediction after prediction on DNA function because we don't assume that what we're looking at in terms of genetics is due to a long history of evolutionary processes. So we don't expect evolutionary leftovers in genomic fossils. Of course, we expect mutations and some degeneration, but not nearly as much as the evolutionists would expect or predict. So we're the ones as young Earth creationists that are making the testable predictions. But if the evolutionists could make better testable predictions that are shown to be successful, then that would be another line of evidence that might convince me that universal ancestry is slightly more plausible than what I see it as now. I know that was long-winded, but yeah, go ahead, Jackson.
All right. Well, what would convince me that evolution is wrong and deep time is wrong? You got to convincingly disprove radiometric dating, because there are many instances where it does work and it's been cross-referenced. But you got to show ice cores are all wrong because they go back 55,000 years, the annual rings, before it gets too compacted to read. But then further dating methods dating to 800,000 years. You got to disprove that there's a glass sponge that was found to be 10,000 years old, older than the Earth is supposed to be. You have to disprove paleomagnetism. You have to disprove sediment cores. You have to disprove just all kinds of things. And but what would convince, if all those were overturned and something was truly found to be thought to be old but proven to be young, that would convince me. And if a transitional fossil or a fossil was found in the wrong place, say a T-Rex was found in the Cambrian, and a Nomalocaris from the Cambrian was found in the Permian, then I would seriously question these time scales and this the whole evolutionary theory. So that's my response.
Gotcha. Thank you for your answer. So you have a question from Health first, and Feeling second. Question for you, Jackson. Why, despite supposedly sharing 99% of our protein coding genes with chimps, do we look so incredibly different?
Well, 99% is a lot, but 1% of what is it, 3 billion or 4 billion base pairs is also a lot. So there's going to be some differences, inevitably. But there are a lot of similarities too, genetically and anatomically. So that's my answer.
Okay.
Please, if I could, I'll respond real quick.
SFT.
What's also problematic too is the fact that the evolutionists will oftentimes say, we're 97, 98, 99% similar genetically to chimpanzees. But it turns out that, for one, less than 2% of our genomes are protein coding. And many, many genetic sequences are not even comparable between humans and chimpanzees. And there are entire gene families that are known in humans and are not even found in chimpanzees. Okay, so when we consider overall size differences, architecture, gaps, copy number variations, a lot of these things that the evolutionists have ignored, we find roughly 400 million DNA differences, single letter differences that separate humans and chimpanzees that, guess what, have to come about, they have to arise and then spread through the populations to the point of what? Fixation. Okay, we've also got, and I'll end it here, we've also got tens of thousands of chromosomal rearrangements that also had to arise, spread, and become fixated as well. Rates of fixation are extremely slow, and most new variants are lost, of course, because they are rare. So the point is, there's a huge waiting time problem to explain how these vast amounts of differences can arise in just a mere 6 million years. Evolution alone has been falsified because of these numbers. The waiting time problem, a new paper came out recently by Dr. Sanford in a secular journal. It has not been answered. It's been read by 10,000 scientists and it's solid, and there's been no real rebuttal other than a couple, YouTube videos or angry bloggers. But I mean, no one takes those seriously. So that's a huge problem for evolutionary theory and ape to man evolution. I yield.
Thank you for your statement there. But Jackson, you want to quickly respond to that? Go ahead, before we go to the next question, because it was addressed to you.
Yeah. What was the question again? I'm sorry.
Let's see here. Why, despite supposedly sharing 99% of our protein coding genes with chimps, do we look so incredibly different?
Oh yeah, I answered that already.
All right, well, so we'll go to the next one. So
Yeah.
this is from CJ Cox, aka the Synagogue. He has a question for you, Jackson.
Okay.
How can humans and Neanderthals mate and bring forth viable offspring if they are not the same species?
Well, there are many cases of hybridization between different species that produce fertile offspring. And like I said, we differ by 202 base pairs, so we are of different species. But Neanderthals were either subsumed into our genome through breeding, or we wiped them out, basically. So that's my answer.
And SFT, if you want to go ahead and respond to that.
Yeah. If I could, to answer the Neanderthal question is, and I'm just looking for some slides here. As Ken Hovind says, it's nice if you ask the questions in the order that I have my slides, then they're easier to find. When you got a million slides, it's difficult. But I found some slides here. So here's the problem. There has been a drastic revision in Neanderthalensis, okay? In 1856, and this is why evolution is harmful, okay? Because in 1856, this is how they depicted Neanderthal. 2015, this is a digital reconstruction. I mean, it's obviously a human. And what we have in terms of evidence, okay, is that they buried their dead, used medicine, okay? They had complex hunting patterns, made complex tools. They dived for shellfish. What we see, once again, just like with Erectus, we see purposeful navigation, okay? And we see that since we have their genes and we actually interbred with them, and the fact that humans and Neanderthals are less different than polar bears and brown bears, obviously they're humans. They're 99.7% similar to humans. Now, of course, that we'd expect them to be different because they are ancient man. They were earlier, and they also, as I showed in a number of sources, they were highly inbred, 40% less fit than modern humans. This is the end stage of the Neanderthal, where they accumulated so many deleterious mutations. Okay, a lot of them coming to fixation, which is why their mitochondrial DNA and nuclear DNA is slightly different than modern humans, but it's because they experienced different environmental conditions. They had a different history. They were the most inbred people on the planet. Their runs of homozygosity are fascinating. So of course they are going to be different, but a lot of the features that we see, okay, we see in people groups today. For example, here, the skull cap, if you found that isolated, the evolutionists would conclude, because oftentimes we find these fossils and these bones in isolation and in fragments. And so they can find a skull cap isolated, and they can conclude this might be from a Neanderthal, even though these same features exist in humans today. I yield.
Very good, SFT. So Jackson, was addressed to you. If you want to say something really quick, go ahead.
Well, I'm not arguing that Neanderthals weren't human-like. They evidently had culture. They had maybe even religion, and they did bury their dead. They had probably instruments. They made fire. But and if they were alive today, we would definitely give them human rights, and as we should. But they weren't genetically human, and I think that's been proven conclusively. And I yield.
All right, so here is our very last question. It's addressed to you, Jackson. So here's what he asked. So, what about where her spine enters the skull? So
The
whatever
foramen,
that means, I don't know.
The foramen magnum. Yeah, it enters. It's kind of intermediate with afarensis, but other Australopithecines have it more forward than backwards. But the shape of the spine also indicates that it was bipedal. It's kind of an S shape like ours, instead of an arch shape like a chimp. So it definitely walked upright, at least most of the time. it was still capable of climbing, evidenced by its arm structure, but it was a biped, and I yield with that.
I'll respond to that too, Praise. So actually, I'll see if I can find the. Okay, so when it comes to and I showed some slides earlier on Lucy and the foramen magnum, and like I said, I got so many slides here. So, but I do have notes based on what he said in his opening. And the fact is, Lucy's foramen magnum lies in much the same position as some apes do today. So to claim that Lucy walked upright is actually misleading. And the best case scenario is probably that Lucy might have had a bit of a unique walk, but not a walk similar in terms of humans where we walk upright. And when we actually, here's something that's interesting, when we actually account for size differences, okay, with Lucy, then a living gorilla and an Australopithecine, its foramen magnum would be nearly identical. Because remember, these Australopithecines, Lucy, for example, was 3 or 4 feet tall. Okay, so a lot of it is based on misinterpretations and erroneous reconstructions because, as I pointed out, we have these isolated bones where oftentimes sexual dimorphism is invoked, as I discussed in my rebuttal. And so you've got the terrestrialists and the arborealists that are emphasizing different bones. And a lot of times those bones they're emphasizing are actually human bones. And so, of course, if you have a human bone that you're misclassifying in the afarensis category, remember, Lee Berger admitted that afarensis is a chimeric species, then, of course, you can look at a human bone and say, look at this. Lucy was kind of human-like. No, that's a human bone. And this is real. You know, creationists aren't making this up. This is in the literature. Paleoanthropologists admit that oftentimes, I mean, it was Ian Tattersall that admitted that habilis is kind of a wastebasket of bones. So I'll yield there.
Well, that concludes the debate, guys. Awesome job.
All right.
Great questions and answers. Super chats. Well, I'm going to have to give a quick shout out to Logical Plausible. Probable. He's having an after show. So head on over there. You're welcome to head over there, both SFT and yeah. So if you guys, and Jackson, if you guys want to do that, go ahead. I'll let SFT conclude it.
Awesome. Well, we're going on 2 hours and 15 minutes. We still got over 50 people in the chat. It's been a lot of fun. This is a great topic, great, respectful, cordial debate with Jackson.
Yeah.
I think he touched on a number of awesome points. The
Yeah,
Q&A was awesome as well. Tons of good questions that we could go back and forth on. And yeah, thanks so much, Jackson. And hopefully we can do this again. Jackson,
thank you.
Thanks for doing this. Any final words from you, Jackson?
Yeah, I just want to say you're going to have to send me the link to the after show. I'm not going to know how to get there. I'm terrible with technology.
No problem.
And, but also, yeah, again, thanks for having me, and thanks for being respectful. And I respect you a lot. You're very knowledgeable. So I hope we can do this again. It was honestly a lot of fun. I thought I would be really nervous, and honestly, I was for like 30 seconds, and then it faded away. And so, yeah, it was fun.
That's usually the case. Leading up to the debate, and then the first few minutes where you're kind of getting into your opening statement, that's where typically you're the most nervous. But then you kind of warm up, especially in a cordial discussion like this. And I'm glad that you brought up some good points and some good arguments that we could touch on and discuss. So I appreciate you making this a fun debate and an engaging debate. And Logical, Plausible, Probable, his after show. Jackson, I'll send you the link because you are invited, and it'll be similar to the link that I sent you here. It's a StreamYard link, and there should be a bunch of people in there and continue the discussion.
All right, yeah, just email it to me and I'll join.
Okay. Well, Praise, thank you so much for moderating. You are the award-winning moderator, just like George is the award-winning co-host. So great job. You moderated a great debate last week with Bill Tavis. You moderated a great debate tonight. So you've got a really strong career in moderating ahead of you, Praise.
Appreciate that. Thank you so much.
All right, guys. SFT is out. Jackson, great debate. Tons of fun. We had like 100. I think we had about 320 people at one point. Human evolution, man. That's one of my favorite topics for sure. Jackson and I debated that several months ago, actually. Human evolution.
I couldn't pick out a winner from that. I think we both kind of countered each other pretty well.
I enjoyed it. I enjoyed that debate. I think we got deep into a lot of the so-called hominids and transitionals. But that's my question to you, Jackson, is how do you feel about, and you may even disagree with this, actually. That's why I don't mind pushback. But I find after examining all the best so-called examples of ape to man evolution, so-called basal forms of humans or primitive forms, as they'll call it, right? They'll look at they'll pick up the most anomalous bone in the fossil record and they'll say, see, this is basal because there's some anomalies when you compare it to extant humans. But from my investigation into this, I've been able to, I believe, sufficiently categorize every so-called transition or example of a basal human as either fully human, degenerated human, fully ape, degenerated ape, or amazingly an artificial species. Like I've got a couple articles in front of me, including a couple papers that talk about how Australopithecus sediba and Homo habilis may indeed be the result of the accidental mixture of ape and human bones, right? So that's what I mean by artificial species, is in that which is called Australopithecus sediba isn't actually a real species. It's a mixture. What are your thoughts on that, Jackson?
Well, there are two good specimens of Australopithecus sediba, so I think it's pretty unlikely that they're artificial species. And habilis might be, and Rudolfensis might be the same species, or they might be an Australopithecus. It really doesn't matter what you call it at that point. But yeah.
Actually, John, if you could share my screen. Can you see my screen, guys?
No, born 100 years late. I only have like 3.
John, is
only
my
3 of them.
Oh, you can see my. Oh, okay.
But the thing about the sediba is they're not really thought to be our direct ancestor. They're more of an offshoot.
Right.
They're the timeline and some of the features doesn't really match with what we would expect from a true human ancestor.
Well, from my experience debating, I'm hearing a little bit of feedback. From my experience debating some pretty militant evolutionists, they'll look to sediba and habilis. For example, Gutsit Gibbon, she's got a video titled something similar to, the perfect transitionals, something like that, or a creationist's worst nightmare. And the two she picked out are Australopithecus sediba and Homo habilis. But what's funny is those
I wouldn't have picked sediba.
Yeah. And those are the two most disputed. Like, for example, even if it wasn't an artificial species, as you pointed out, just in 2018, 2019, there's many paleoanthropologists pointing out that Australopithecus sediba has been ousted as a direct ancestor to the Homo genus, right?
It's more.
But then there's also those right here that I was pointing out. Just to demonstrate what I was saying is true. Human missing link species may be jumble of species. And Ella Bin and Yoel Rack, they've pointed out that Australopithecus sediba is a mixture of Homo and also apes. And also, and last thing I'll say too, and I'll get your thoughts, Jackson. Famous paleoanthropologist Lee Berger, he's actually recently admitted that Australopithecus afarensis, right, Lucy, the famous Lucy species, is a chimeric species. It's the mixture of human and ape bones as well. So Lucy being one of the most favorite so-called examples of human evolution over the years is now even being looked at as a possible chimeric species. So my point is, it doesn't really seem like there's any really strong examples, when if human evolution is true, I feel like there should just be so many examples that it's just undeniable, it's overwhelming, and I just don't think that we really see that. Yeah, go ahead. What are your thoughts, Jackson?
Well, there are a couple other specimens of afarensis in Lucy, like the big man and the Salem child, that are partially complete skeletons, and there are like hundreds of other incomplete just fragments. So I don't think it's a chimeric species, but there's one almost fully complete specimen of Australopithecus prometheus, or which might be an early Africanus, that shows that it was trapped in limestone Sturgeon cave. So we know that all the bones belong to the same individual conclusively. And it's not on our direct lineage, most likely, but it's an offshoot showing the progression, you know.
Right. But that seems to be my biggest problem, is the best so-called examples of a true transition from ape to man, Australopithecine-like ancestors up to Homo habilis, up to Homo erectus, and eventually Homo sapiens. They always consist of such a scattered bone collection, right? Lucy here was originally found in hundreds of pieces, and that's how you can even get chimeric species in the first place, is because, like, for example, when you hear, because you keep saying, you know, they got this many specimens, and, you know, when you hear defenders of evolution say, you know, we've got 400 specimens of Lucy, people think, oh wow, they got 400 full complete skeletons. Right. So, for example, this is an example of what they mean by 400 specimens. It's 400 fragments here. Everybody on the panel can see. So they turn this into that. I mean, it's amazing. From these fragments, they know what Lucy looked like and what Lucy's husband and child looked like.
a lot of assumptions went into putting that together, I think, just like the geologic column theory. You know, don't get me wrong, it's not bad to try to guess things, but whenever you put things together just the way that you think they fit, and they just, you know, happen to almost fit that way, it doesn't really prove anything. It's a theory, don't get me wrong, but it hasn't been observed. I mean, it hasn't gone through a scientific.
I wouldn't even call it a good theory.
It's a hypothesis at best.
Well, they'll say it's a theory, but something way, way before a theory is a hypothesis.
Yeah, I mean, a theory at least has to be tested once, correct? I mean, you kind of hypothesize about it, then you test, observe, repeat, and, you know, form a theory as you go, kind of thing.
For example, if I had a theory, you know, I had an idea that a cow, you know, we know cows can jump. So, I mean, if you feed it enough, train it enough, eventually it'll be able to jump over the moon. we know there's no evidence for that. There's nothing supporting it. So it's always just going to stay as a hypothesis. And that's exactly what we see with human evolution. You know, they have an idea, but it's not supported by the data, and which means it's just going to remain a poor hypothesis. And I'd be curious as to Jackson's thoughts here too, because they'll try and say, well, you've got your Australopithecines, you know, 6 to 8 million years ago, and then, you know, eventually you got Homo habilis, which they'll say is a perfect transition of Australopithecus-like ancestors to the Homo genus. But what we actually see is extensive coexistence of even the earliest Australopithecines and man. And I have a list of examples as to how we know they coexisted. And we find We find, human limb bones, human jaw bones, human hands, foot bones. We find human footprints, human bones, shelters, stone tools, butchered bones, showing that it's humans, obviously the ones doing this butchering, and also humans being buried with Australopithecines. And last thing I'll say, examples of humans eating robust Australopithecines. So my point is, we see such overlap, which is actually what we would expect after the flood and post-Babel, or even pre-Babel, is we would see a lot of this coexistence, right? Yeah, what are your thoughts on that significant coexistence we see, Jackson?
Well, nothing I've read has suggested that humans in their modern forms survived alongside Australopithecines, but maybe Homo habilis or some other early Homo species did. I know Australopithecus anamensis and Australopithecus afarensis inhabited the same area at the same time, but it's inferred that anamensis may be its ancestor. But maybe one population diverged and became afarensis, and one population kind of remained fairly similar in anamensis. Might be one explanation for that, but I'm not familiar with humans living alongside Australopithecines.
Yeah, you find example after example of it. For example, here's a quote from famous paleoanthropologist Richard Leakey. "I see no reason that bands of Homo would not have killed and eaten robust Australopithecines when they could, just as they killed and ate antelopes and other prey animals." There's also a quote from
I know what you're talking about now. The robust Australopithecines, like Paranthropus, they did live alongside like Homo habilis.
Right. But remember, if people like even Lee Berger are admitting that, well, Australopithecus afarensis may be a chimeric species, mixture of Homo and ape bones. Well, that means by definition, humans must have lived alongside or coexisted with the Australopithecine. Now, I know what you're saying is, well, maybe it's a basal form of humans, right? You're saying, well, maybe it's erectus, but that's the point. Is the data that we find pertaining to so-called early humans. I'm trying to pull up the paper here on Homo erectus. I can't find the paper, but I do have an example here that Homo erectus has evidence for purposeful navigation, has evidence for being able to speak, but Homo erectus also has evidence for what's called reductive evolution. And that's where you have these subpopulations isolating themselves. And what results is inbreeding and accelerated genetic degeneration. So they'll pull up these anomalous features, these anomalous bones, and they'll say, see, this is basal to extant humans today. When in fact, no, what we're looking at is something that's fully human, but it's riddled with pathology, it's riddled with disease, it's riddled with degeneration. So where they're saying is, this is basal, this is primitive, it's actually just an example of a fully human people group experiencing reductive evolution, which isn't evidence for obviously ape to man evolution. Yeah, go ahead, Jackson.
Well, we don't have DNA for erectus, but we do for Neanderthals, which aren't our ancestors, they're more our cousins. But we differ by 202 base pairs on the mitochondrial DNA, which indicates there are different species. So I don't think there's a degenerated human or a different human.
If my screen's still shared, you can see the drastic difference in understanding pertaining to Neanderthalensis. 1856, they articulated and represented him as this brutish half ape, half man. But today, for example, in 2015, the reconstructions have drastically changed. And this is based on the fact that now we know that Neanderthals buried their dead, made musical instruments, tools, cosmetics, jewelry, and purses. Their DNA shows that they were human and interbred with us. I mean, we literally have their genetics. And the reason why they are different in terms of their genetics, as in there's a slight divergence, right? Even though they're still 99.7% similar to us, right? Every human today is 99.999% somewhere. Neanderthals, which are an extinct people group, they're 99.7% somewhere. Well, we'd expect their genetics to be slightly divergent because they were the most inbred people group on the planet, actually. And they accumulated a lot of recessive mutations came to the forefront because they existed in small tribes. Therefore, they were highly inbred. And what would result is rapid fixation of new genetic variants. So that explains why they're slightly different in their genetics. But we actually have paleoanthropologists that will admit Homo neanderthalensis should be considered a subspecies of Homo sapiens rather than a separate species. I think the only people that want to say Neanderthals are a sister species or a separate species are those that are so committed to the ape to man evolution story that they'll say, well, sure, according to the modern definition of species, then yes, they would be the same species. But, you know, assumption after assumption pertaining to human evolution, you know, they can't actually be a descendant of Homo sapiens, so therefore they'll put them as a sister species. But the actual genetic data and also the fossil data would just suggest that they're a variant of human, but they are degenerated due to inbreeding. Yeah, go ahead, Jackson.
Well, if they were degenerated due to inbreeding, and all humans started out with 2 individuals, why aren't we all the same amount of inbred?
That's a good question, actually. That would be because, and I think you and I discussed this in our debate as to what the fundamental difference is between our model and your model when it comes to the origin of genetic diversity. For example, how do you explain, from an evolutionary starting point, the origin of most or all genetic variation?
Well, I don't know. I'm honestly don't have a good explanation for it. I know other people do. I'm just not the best representative for some of this subject matter. But,
no, that's fine. That's fine. I'm not referring to like the origin of the first life or the first cell. I'm talking about the DNA variety, the DNA differences that exist within species and across species. I'm just talking about how I'm really hearing some feedback. So I don't know. Hopefully whoever's not talking, just mute. I don't think it's you though, Jackson. So for example, here, if my screen's still shared, it says the ultimate source of all genetic variation is mutation, right? The evolutionary model, they'll say that mutations add genetic diversity, right? Because mutations are adding something that was not previously there. So the evolutionary model looks to mutations as being the source for all genetic variation. And eventually you have processes like recombination, which can scramble up that genetic variation, those DNA differences, creating new varieties and new species. But our model, as a creationist, we look to created nuclear heterozygosity. So what that, it sounds fancy, but all it means is the original created types or kinds, they would have been front loaded with genetic variation. So you look to mutations as being the ultimate source. We actually look to most genetic variation as being not by mutation, but by being the result of design, initial front loading at creation. So that means Adam and Eve would have had no mutations. They would have had no genetic mistakes, no genetic defects at creation. And therefore, there wouldn't have been an inbreeding problem. Yes, there would have had to have been inbreeding the first couple generations, but there'd be no mutations those first few generations to actually come to the forefront and lead to genetic damage. But the evolutionary model, there's a problem for you, Jackson, because you explain genetic variation as being the result of mutation. So yeah, you actually have an inbreeding problem when it comes to the out of Africa population bottleneck, for example, because you have all of these mutations in hidden recessive form that are going to come to the forefront and be manifested, leading to accelerated genetic damage. So that's how we don't have a mutation problem or an inbreeding problem, just to answer your question. Yeah, go ahead, Jackson.
Well, even if Adam and Eve had a perfect genome, their offspring wouldn't. They would be damaged by the sun's rays, or genetic code would be damaged to some extent somehow. But even if it wasn't, You have long after the fall, you have Noah's Ark, which is another severe genetic bottleneck where there are only 3 pairs. So we're back to the same problem of why aren't all humans the same amount of inbred?
Well, it's not a problem because when you
Ask the royal family.
ask the royal family, so you'd have Adam and Eve who would immediately start having kids, and then their kids would start having kids, and so on and so forth. Adam and Eve would have had dozens and dozens of kids. Now, what we see in terms of the biblical bottlenecks, we can actually reflect them on phylogenetic trees. Is we see that's why you see this starburst pattern, right, where you have explosive growth from a center point. We can see this in the mitochondrial DNA and the Y chromosome. Okay, Y chromosome is inherited from our father, mitochondrial DNA from our mother. So the point I'm trying to make is the biblical bottlenecks, they were short. They were 1 or 2 generations max, and they were followed by rapid and exponential population growth. So what that actually means by definition when it comes to bottlenecks is very limited genetic diversity would be lost. And since at creation, you'd have no genetic mutations to come to the forefront, you wouldn't have any genetic damage. And at the flood, you would have, 1 generation followed by rapid and exponential population growth. You wouldn't have the type of mutation accumulation that we see today. But the problem is, Jackson, I'd be curious as to how you answer this. Actually, I should set it up first by asking you, since today all humans are 99.99% similar, right? That means humans have low genetic diversity. This was contradictory to what the evolutionary community expected, right? Because if you're evolving from the Australopithecines up to Homo habilis, up to Erectus, and supposedly Erectus lived on the globe for millions of years, and then eventually Homo sapiens, well, every generation you have new mutations pouring into the next generation. That would be adding diversity. So we today should have high diversity, but how can we have low diversity, would you say, Jackson?
Well, I know some African populations have an incredible amount of diversity. I know that people outside of Africa have a lower amount, and I think that fits with the model of starting in Africa and going elsewhere. But what was I going to say?
But remember,
diversity. Yeah.
Well, remember, when you look at Africans and non-Africans, right? Africans and non-Africans are still human. So overall, whether you look at an African or a non-African, there's still low genetic diversity. I mean, and this goes for the uniparentally inherited DNA compartments as well, like the mitochondrial DNA, Y chromosome. For example, in the mitochondrial DNA, the average amount of differences between any two people is about 20 to 25. Now, the most is about 120, maybe 125. That would be between like me and an African. So yes, Africans have more genetic diversity than non-Africans, but overall, humans in general, both Africans and non-Africans, still have low genetic diversity, especially compared to other species. So my question is, why isn't there a greater amount of genetic diversity if deep time evolution is true?
Well, there was a genetic bottleneck 50 to 100,000 years ago of where the human population got down to a few thousand individuals, they think, based on genetics. And there are animals with lower genetic diversity than we have, and you would think it would be uniform across all species because of the ark. But like cheetahs, they got down to just a few individuals, and they're having all kinds of problems now. Same with Tasmanian devils. I would ask, why aren't all animals suffering severe inbreeding consequences if they all went through a bottleneck at the ark?
Right. Those are good questions. Your first answer was correct. So let me address that. Before I do, I just wanted to point out the source, and then I'm going to stop screen sharing for now. Because in the debate, it was brought up by, I think, Sam Jenkins, that we found human footprints in layers that are millions of years old, that we probably shouldn't find them. Here's 2 examples: the Laetoli footprints, which I'm sure you're aware of, and then more recently, footprints in Crete that were dated to 5.7 million years old. And those that discovered and have examined these footprints, they've admitted that, okay, if these were found today, let's say they were found on a beach today, they would be indistinguishable from human, as in the only conclusion you'd make is they're human. But because they're being found in millions of years worth of strata, then it's this circular reasoning going on. So now they're saying, well, you know, we know humans were not around back then, so therefore they can't be human, and therefore what are they, right? And
yeah, if these are human, this blows our whole thing apart, right? That's
Right.
kind of
exactly. So they can't conclude, well, these are human footprints, even though that's exactly what they like. Here's an example. Here's the Laetoli footprints, right? Inline, big toe.
Yeah.
You know, here's a chip. Here's a human foot. I mean, a modern human footprint perfectly overlaps the Laetoli footprint. Now they've done studies, and I've read the studies where they try. Yeah, go ahead.
It also overlaps with the afarensis foot. They found enough foot bones to basically say that the toe was in line with the other toes. And as for the Crete footprints, they're not perfectly human. They're shaped a little bit differently. They're a little bit broader, a little bit shorter.
I've seen people with some inhuman feet before. I don't know.
Well, here's the thing about that is when it comes to afarensis, what they actually have in terms of what they think is a foot bone or an example of what their feet look like is, okay, it's circular reasoning, is this footprint, okay, that they've just arbitrarily concluded belongs to afarensis, and small little fragments, which many paleoanthropologists have admitted those fragments don't even belong to afarensis. They're just a human. The reason why they look human, the fragments, is because they are human. And this is the paper I wanted to show you earlier. Donald Johansson, he points out, and he is the discoverer of Lucy. He pointed out that his collection suggested that Homo and Australopithecus coexisted as early as 3 million years ago. Now, granted, this Homo could be Homo erectus, but as I've demonstrated, Homo erectus is still fully human, still considered a human, just a degenerated human. Now, they've tried to say that the stride on the Laetoli footprint is slightly divergent from humans today, which is just ridiculous. I mean, there's different strides today. If I'm striding through sand and I've got a limp or my knee's sore or something, I mean, there's so many variations in strides that to conclude that, well, this isn't human, this is, you know, a transitional form of human based on their stride or something like that, to me is rather weak. And Jackson, before you comment on that, because I feel free to do so, your question about the genetic diversity is correct. The evolutionists will say, well, we expected higher genetic diversity. We found that humans have low diversity. Therefore, there must have been some type of near extinction event roughly 70 to maybe 200,000 years ago, right? There's debate over it. And what would have happened was humans at that time would have been downsized or reduced to between 2 and 10,000. So what happens when you have a population of 2 to 10,000 for many, many, many years, maybe even thousands of years, while you have inbreeding, which then reduces levels of genetic diversity. But my problem with that whole model is, this goes back to what we were talking about earlier, Jackson. Okay. We as creationists, we explain the origin of genetic variation as being designed and created, right? But you guys, you explain the origin of genetic variation as being the result of mutations. So this hypothetical population bottleneck, it doesn't work, Jackson, because you have a population of 2 to 10,000 humans that are inbreeding for who knows how many years, thousands of years maybe. You'd have so many negative, deleterious, and disease-causing mutations coming to the forefront that so-called hypothetical bottleneck actually would have driven humans to extinction, rather than a spreading out in all parts of the world where this so-called human population sees dominion over the planet, eventually inventing the space shuttle. You know, this inbreeding event would have been detrimental. I don't think it's realistic. Yeah, go ahead, Jackson. I said a lot there.
Well, biologists think the minimum population size for a viable population is 100 pairs. So 1,000 wouldn't have been the end of the world, but it would have reduced genetic diversity by a lot.
But remember, you've got 2 to 10,000, not for one generation, not for 2 generations, but you'd need hundreds, maybe thousands of generations in order to reduce all of that genetic diversity in the first place. Remember that the human population before the bottleneck would have had high diversity. So now the evolutionary community invokes the bottleneck So it reduces the human population at that time to say between 2 and 10,000. Now they require, and they never give a number because they don't know, but let's say thousands of years worth of inbreeding in order to reduce the levels of diversity. I mean, that population would have been so genetically damaged. It's not just 1 or 2 generations. That's the problem. We see an example of this today, Jackson, as you pointed out, with the cheetahs. Cheetahs are experiencing a genetic bottleneck. They're down to 7,000. Their sperm is degenerate. They're highly degenerate. Conservationists are worried about them. And we're obviously not going to expect this population of 7,000 cheetahs to suddenly spread out into all parts of the world, seizing dominion over the planet, right? They're highly degenerate. And that's an example of what that population of humans would have been like. Yeah, go ahead.
Well, the population, they think, was at one point down to a few individuals, and that's the problem. It's not that it's 7,000 today. It's that it was even lower in the past through probably natural processes, and they just barely survived.
What would have brought the cheetahs down to 3 individuals?
A few individuals. It could have been disease, could have been the ice age, and their habitat nearly got wiped out. No one knows.
It's more so hypothetical, but you're asking about the genetic diversity levels in all animals. Why don't they reflect a flood bottleneck? The thing is, they do.
Yeah.
Because we have, I'm not sure if you're familiar with the newer Rockefeller study on genetic boundaries and the CO1 gene, the barcoding gene. It's in the mitochondria. It's a highly conserved mitochondrial gene called the CO1 gene. It's highly conserved, and there's low genetic diversity in all species. You've probably heard the headline over and over again, Jackson, or maybe anybody on the panel. There's been many articles written on it where it talks about how over 90% of today's species arose or originated at exactly the same time. That's based on this genetic boundary study. So yeah, that makes sense to us because we had a flood bottleneck 4,500 years ago where you have all these archetypes coming out, spreading into all parts of the world. So the diversity levels we see in these highly conserved genes within animal species, they do reflect the flood bottleneck. Yeah, go ahead, Jackson.
Well, I know the paper you're talking about. I don't remember all the details of it, but I remember that it got misinterpreted by one news outlet, and people ran with that. I don't remember all the details.
Well, they said it was misinterpreted because what they did is they took the genetic data and they calibrated it with their assumed deep time evolutionary history, right? So they said, well, the young Earth creationists are misrepresenting this data because, the authors of this paper are saying that the origin of these species goes back 200,000 years ago. But the thing is, when you actually read the paper, they're not basing that off the genetic data. They're basing that off of circular reasoning and calibration. When we only go by the genetic data, we can safely say that this bottleneck, where 90% of today's species arose at the same time, was actually indeed 4,500 years ago. But you have to read through the lines. You have to read through the evolutionary-based assumptions in the paper to come to that conclusion. You have to realize, okay, the date they're getting of 200,000 years, that's not based on genetics. That's actually based on the geological column. So to me, that's not misrepresenting the paper. That's just reading the paper in an unbiased way, where we're not just assuming the conclusions and assuming the evolutionary assumptions. We're looking at the empirical data, and then we're making our conclusions. Go ahead, Jackson.
Well, I haven't read the paper, so I can't really argue against it. So,
no, that's fine. I just wanted to answer your question as to why we don't see genetic diversity levels representing or being reflective of a flood bottleneck 4,500 years ago. And my point is we do because we actually see low diversity in humans, which is what we would expect. God created 2 people, Adam and Eve, 6,000 years ago. That would reduce genetic diversity right off the bat. We know God created populations of animals, so we'd expect a greater diversity within animal populations. That's exactly what we see today. And because of the flood bottleneck 4,500 years ago, we would expect a lot of these highly conserved genes to be reflective of that. And that's also what we find is low diversity in animal populations within the mitochondrial DNA barcoding gene. So, I mean, to me, it's really just fascinating how well the scientific data corroborates the biblical count of of origins. I haven't seen theistic evolutionists deal with it. I haven't seen old Earth creationists deal with it, and I certainly haven't seen atheists deal with it. But I've been talking a lot, so I yield.
Well, my point is the genetic diversity seems to be variable between species. Like the tuatara seems to have a good amount of genetic diversity from what I've read, or maybe not. Maybe I'm thinking of something else. But the cheetah doesn't. Tasmanian devil doesn't. So you would think they would be all identical if they all originated from two animals on the ark.
Well, we wouldn't expect them to all be identical because you have different generation times, different reproductive rates. We have some animal kinds that were brought in 7s as compared to in 2s, depending on clean versus unclean. Larger mammals like elephants are not going to reproduce as quickly as smaller mammals like mice. But overall, the rule is we see greater diversity in animals than we see in humans. Of course, there's always going to be an exception to the rule, but the exception to the rule doesn't overturn the rule. And the example of cheetahs, of course, after the flood, you would have had a couple cheetahs, maybe. No, you wouldn't have cheetahs. You would have had the cheetah archetype, right? You would have had some cat archetype on board the ark, possibly Polarius, if you want to look that up. I think something similar to that. Which would have given rise simply through the shuffling of these front-loaded genetic differences, simply through recombination and gene conversion. You get from one cat archetype off the ark into what we see today: house cats, lions, jaguars, cheetahs. So eventually you would have had a breakaway population. That population would have changed enough where you can consider it a new species. You could say, okay, this is a cheetah, but that cheetah would have faced a different historical and environmental history than the other felids, and therefore the cheetahs might have went into a bottleneck. It's not like a one size rule fits all, right? So yeah, go ahead.
All right. Yeah, I don't really have anything to argue with. I mean, I'm going to go on to the next thing or whatever about human evolution.
Sure. Yeah. No. That's. I think that was a great back and forth. Yeah, go ahead, Dan.
I'm wondering how many specimens they have together. I was trying to look it up earlier of these different types of homos or whatever. Homo erectus, Homo habilis. I haven't seen definitive proof that they've gotten more than like basically that little pile of bones you showed earlier. I was wondering how they determined that type of diversity in the genes and that sort of thing. I was hoping you could elaborate a little bit on that earlier, but I never got around to it. I keep getting kid aggro, so I keep running off and like chasing children around. So.
I feel your pain, brother. No worries. Well, what's interesting is we don't have the genetics. I always point out this fact.
Hey, SFT.
Yeah, go ahead.
SFT real quick. So, born 100 years late, $5 super chat says, SFT, thank you, brother. In my life, I've heard only a few speak about the truth like you do. Jackson, I hope you consider what he is saying.
Well, I
sure,
appreciate
everything.
I appreciate the kind words. Born 100 years late. I find this topic of ancestry fascinating, so I've done my best to look at both sides, and I'm just not very convinced that the evolutionary side. Yeah, go ahead.
Do you want to explain this to Andrew for about the 50,000th time? He doesn't
I've got
understand.
A whole playlist of debates with Andrew just because he's still talking a big game.
He still doesn't understand heterozygosity in a front-loaded genome. He does not understand what that means. It's really amazing.
It's good practice, though, if you keep practicing doing it. And
yeah. I've only written several books on it, Andrew, and had complete debates on it. Created heterozygosity just means, in simple terms, a state of DNA diversity. Homozygosity means a state of no DNA diversity. So the original created kinds would have obviously been created in a state of DNA diversity. For example, the Bible says, be fruitful and multiply. Now, would God have wanted that to be carried out through cloning? Because if the original created kinds were created genetically homogeneous with no DNA diversity, that's exactly what would have occurred. Cloning. Okay, there's no DNA diversity. built in. So for God to design organisms with the front-loaded capacity for change, for diversity, that makes more sense of God's command to be fruitful and multiply. That means the DNA differences we see for the most part between species within species, we would predict that since they're the result of initial design, they should be functional, beneficial, and useful. And these predictions have been in print for years and years and years. And as the years go on, John can speak to this, we find paper after paper of just that. DNA sequences, DNA elements that we once thought were non-functional evolutionary leftovers, they're actually showing and proving to be functional and highly beneficial to healthy life processes in the cell of all life. I mean, that's a fulfilled prediction right there. Yeah, go ahead.
Go ahead, me.
Oh, I don't know. I guess
it's
anybody
a
that
very,
wants to comment.
very good point. Very good point. So real quick, so ladies and gentlemen, Patrick Alexander is the first to subscribe to the new Twitch channel. So if anybody wants to head on over to Twitch and sub to the channel, feel free to do so. Link is in the side chat.
That a boy, Patrick.
But yeah, man, this whole conversation of like, I really don't grasp why so many Evo atheists have such a hard time grasping that premise in relation to how it would, in many instances, account for what they think is proof of evolution from a phylogenetic perspective. It's like, if you have divergence and deletion of, you had their pre-existing capacity, you go down a level, and you now don't necessarily have the previous version accessible, it's going to look like a hierarchy.
Exactly.
Like, this is not that complicated. And this is what I talk about in relation to like computer software all the freaking time, and nobody seems to grasp. Although last night or the night before, whenever it was, with Deepak, who was he's a machine learning artificial intelligence developer, he understood exactly what I was talking about when I was talking about something as simple as user permissions and, you know, the hierarchies that are created in that logic in basically every software application that exists. And developers understand this. Now, some of them who are the hardcore atheists don't want to accept that the same logic applies to genetics or could apply to genetics, but it doesn't mean that it's not real. And
right, like how would you explain like
apply.
Yeah, like how would you explain like symbiotic relationships and things like that? I didn't mean to cut you off, dude. My bad.
No, that's a great. Actually, John, a pull up Andrew Desperate Cummings worthless argument that we've demolished a million times there, where he says recombination does not lead. Okay, so recombination does not lead to this diversity when there are only two alleles at one loci. Okay, which is a genetic location, and two individuals to start with can't happen. John, just out of curiosity, how many loci do you think exist in, let's say, the human genome?
Oh, no idea, but it's a lot.
A lot. Okay, so I'm going to teach Andrew here, who I've had several debates with, same with John, basic science. Okay, it goes back to this idea of just in basic genetics, big A, big B, little b. Okay, so the point is, Adam and Eve would have been created with millions of DNA positions, where what they would have is allelic diversity. They'd have capital A, lowercase a, capital B, lowercase b. Okay, you wouldn't just do this in two spots within the genome. I mean, you would have this kind of heterozygosity in millions of DNA positions that exist. Let me repeat that, Andrew. You'd have DNA heterozygosity in millions of DNA positions that exist. Okay, it's erroneous to say an allele is just two or more genetic variants when you don't actually elaborate on the fact that this is DNA variety at a specific loci. Now, because you have millions of genetic locations in the genome, you then have pretty well an unlimited potential to produce all sorts of variety, even in a single generation. Think about Adam and Eve. They got millions of spots in their genome that are heterozygous, big A, big B, little b. In one generation of recombination, where you scramble up those DNA differences, John, what are you going to get? You're going to get amazing variety, unbelievable variety. The evolutionists, they don't believe you can get variety quick because remember, they need millions and millions of years to build up this allelic diversity in the first place because they look to mutations, right? Well, if that allelic diversity is already built in from the start, then all it takes is 1 or 2 generations to produce variety. This goes back to my debate with Sloppy Saint a couple of weeks ago with John here on the channel. I mean, I must have tried to explain that to him 100 different ways. He just wouldn't get it because he kept trying to say, oh, no, you can't explain all the species today in a young earth creation model. And I'm like, yeah, if you assume that the origin of all DNA varieties mutations, of course not. You need millions of years to build up the variety. But if it's built in from the start, you get visible distinctiveness in 1 generation. Does that make sense, guys?
Yeah, it makes sense to me. It's just like and if anybody else wants to weigh in, feel free. But I mean, I just don't get why this premise seems so difficult to comprehend. I really wonder if Evo atheism prevents people from actually thinking and doing an analysis of anything that actually contradicts their standard talking points. And that's just
they can't.
The more I interact with people and observe the responses and like the apparent inability to grasp concepts, like what you're talking about, it's like they think, this contradicts evolution. I will not compute, cannot compute. They go into complete like system meltdown, red spiral, or spiral death. And
like Andrew right now, pull up his next comment where he says, you're not addressing what I said. Put it up. This guy who just is incapable of addressing. Yeah, right there. Okay. Andrew, again, listen. Okay. Cause I know most of it goes over your head. And Andrew still hasn't been able to, for one, make better predictions than the young earth creation model. 2, he's been incapable of addressing the phylogenetic data from the mitochondrial DNA and the Y chromosome. Okay, Andrew, as I've pointed out, when we start from the genomic position definition of an allele, okay, rather than your gene unit definition, I've even pointed this out to Joshua Stramidas, and I noticed in some of his later interviews, I think he's accepted correction without admitting it because I've noticed he's also been using the genomic position definition of an allele now as well. Okay, so let's put this into perspective. Let's take one single gene, right, John? Just one gene. Okay, well, we know that one single gene usually spans roughly thousands of nucleotides. Okay, and then you have these variants. Okay, these single nucleotide variants, you have them distributed throughout that gene. Okay, for example, let's say at 90 of the nucleotides within the gene, you have single nucleotide variants. Okay, well, that means every single one of those 90 variants, okay, that means they would have existed in a heterozygous state in each of the individuals of the pairs, let's say, brought on board the ark. And then obviously the creation event where you have more than just pairs. I mean, just from one single gene, you have a tremendous potential for allelic diversity. I mean, the question is, why don't we see even more variety today? You know,
well,
this
the other thing,
argument's
and the other thing, something else that has to be addressed, and I know this will go right over Jackson, not you, Andrew's head, is the variability that is, we know beyond doubt is possible from the regulatory perspective. So the caterpillar to butterfly, right? We can take on an entirely different form, not just a kind of different form, an entirely different form can happen. The genome actually doesn't change. Regulation of it does. And in fact, lots of it like melt, like literally melt and disappear. Anyway, my point is that we know beyond doubt from observed evidence that from a regulatory expression variance of gene expression through variety of regulation can dramatically change things in real time. So to somehow argue that, the gene is, the only thing that could have cause a change would be, allele variant, and somehow say that's the only thing that you have to account for in terms of potential variation of species and like body plans, all that kind of stuff, is like, to me, it's a very, very small observation frame of reference in terms of like what is actually necessary from a big picture perspective. And I think because actually looking at it from a bigger perspective, the right now, so good grief, Andrew's continuing to be dumb. Do you want to address that, SFT?
Yeah, he's misrepresenting because he's not getting it. Okay, let's take Adam and Eve, for example, okay? Just Adam and Eve at creation. So let's say they're created genetically heterozygous. Well, what does this mean? This means that they would be a mix of capital and what? And lowercase letters. Capital A, lowercase a, capital B, lowercase b. Okay, that means they were probably something like a middle brown skin. Okay. And therefore, just from that mix of capital and lowercase letters all throughout the genome at millions of DNA positions, DNA spots within the genome, they have pretty well unlimited potential to produce every shade of skin that exists on this planet today. And this is the same for the animal kinds. Okay, you bring on a cat archetype, a dog archetype, a bear archetype, you name it. All of those front-loaded DNA differences, those front-loaded functional DNA differences can transcribe and explain all the different species we see today, all the different species of bears, all the different species of felids, canids. And a prediction of this model is DNA function. And we now have over 80% of the genome. We have evidence that over 80% of the genome is active. Now, we don't know what every DNA sequence does, and that's why we have PhD young Earth creationists like Nathaniel Jensen, for example, who is making very specific predictions, saying, okay, through mutagenesis, we're going to figure out that this is important to the genome, this DNA sequence, therefore it's going to lead to this disease, or, this gene here is not truly neutral, it's actually multifunctional, and therefore multifunctionality explains a lot of the hierarchical patterns, so on and so. I mean, you could read these papers, you can read these predictions, but these critics don't do so. It's just looking really bad for them at this point. John, did that make sense, brother?
Yeah, I mean, I totally follow what you're saying, but let's stop trying to.
Yeah. We've given enough time to Andrew, so we'll let other people jump in.
I know.
Yeah, I was supposed to say he's just over here derailing. It looks like that's kind of funny.
Yeah, Slavic. You can come back in, brother. Sorry about he was waiting while we were ranting. I don't know, Jackson. I think you'd come off mute there for a second. Did you have something you wanted to insert?
No, not really. I'm not really into genetics that deep, so I'm not the one to argue that position, really.
Well, Jackson, let me ask you a question. What would be your favorite fossil specimen in terms of what you believe is a good example of like a transitional or basal-like human?
A human? Let me think. Probably my favorite fossil, not that it's the best ancestor, but my favorite fossil was the Littlefoot specimen, the Australopithecus prometheus. It's the most complete.
Obviously, I have some comments to make on that, but I don't want to. Anyone else want to engage the fossil evidence with Jackson, because that's more a specialty? We'll kind of get off the genetics. I can engage him further, but I know we've been engaging quite a bit on this. So, anyone else have any thoughts or?
I actually got a quick question. A lot of this stuff is way over my head, but I always wondered about how in the world did they know how far away we are from the genetics of like the super early humans? I mean, I'm assuming it's because they compare us to apes, just because, I mean, nobody has the genetic code of, really old humans, whatever they're called.
Yeah, exactly. That's a great question. I was going to point this out earlier, but I've often made the point that when we're trying to answer this important question of ancestry, I usually label it as separate ancestry or universal ancestry, right? Then we need to look to genetics. We need to look to traits, because that's what's inherited in sperm and egg. That's how we're going to answer the question of ancestry. But yeah, a lot of these bones found in the dirt, a lot of these fossil specimens, we don't actually have their genetics. So oftentimes there's a lot of inference being made simply based on morphology, simply based on a collection of bone fragments. And that's why the fossil record itself involves a lot of fancy storytelling. The story is oftentimes changing. That's why when we got the Neanderthal genetics, we quickly realized, wow, I mean, we interbred with them. We literally have their genetics. They're human, you know, but we don't have the genetics of the Australopithecines. We don't have the genetics of most of these so-called examples of human evolution. So yeah, that's a good question. It's a lot of inference. Also looking to the chimpanzee genome, comparing chimpanzee genetics with human genetics, comparing human genetics with gorilla genetics, so on and so forth. But yeah, they don't have the genetics of most of these fossil specimens.
So would it be fair to say it's like completely guessing at, you know, well, because we have similar genetics code to, let's say, an animal, well, then therefore we are related. It's literally just guessing because you have an assumption that we came from them. If we didn't have that assumption that we came from them, then you can just be like, you know, like the Ford car. I heard this, I don't know where I heard this, but like they have different models. Does not mean that they're all evolving to a newer and better model. It's just that different, you know, one designer designed a bunch of them. So if they don't hold to that dogmatic view that we're all evolving, well, at least, you know, different ape, we were apes earlier and now we're a little bit more non-apes. If they didn't hold to that view, the genetic information wouldn't be pointing to that. Is that correct?
Exactly. I like the way Kent puts it, since it was Kent versus Jackson today. He says, you know, just because the lug nuts of a Pontiac are interchangeable with that of a Chevy doesn't mean that they evolved from a skateboard millions of years ago, right? It demonstrates a common designer, just to put it simply. Jackson, I saw that you unmuted yourself. Go ahead.
Oh, I was just going to say it's getting pretty late over here. Well, not over here, but just for me. So I'm probably going to head out. That's all right with you guys.
Of course. Yeah, Jackson, you've been busy tonight. You had a 2-hour debate and now a 2-hour after show. You know, you're a champ. So I appreciate it. If you want, you can have some final thoughts, final words before you get out of here. But it's up to you.
No, I just thought it went good. I mean, I enjoyed it, win or lose. I haven't decided if I won or lost yet. I'll have to watch it back. I'm probably going to do that here in a minute and go to bed.
Yeah, it's pretty late here as well. I got to go make sure everybody's still in their bed too. You did a great job, Jackson. I didn't mean to steal your. I'm gonna get off too.
No, thanks. All right. Well, see you guys later.
Hey, blessings, Jackson. Great debate. Dano as well. I was gonna say before he left, I liked his profile picture there. I thought that was funny. So Jackson, yeah, I'll shoot you an email sometime. And again, great debate, tons of fun.
Okay.
I appreciate it.
Yeah, it was fun. All right, I'll see you guys later.
Appreciate it, Jackson. Have a good one, man. I just stepped away for a second. Appreciate you joining the after show.
So just to kind of double check here. So most of this genetic, you know, the stuff they talk about, or geneticists, at least the ones that are talking about evolution, they're literally making up stories because they have an assumption. It's not like it's hard science, even though they talk about it like it's hard science. Am I understanding this correctly?
Absolutely. Yeah, they don't. You'll be fascinated by this paper that I've been invoking in almost every debate for 3 years. Nobody ever addresses it. John, maybe share my screen just real quick. The genetic data where they build their stories doesn't even help them at all. Because for one, they're finding out that when we actually consider the entirety of the chimpanzee and human genome, we're really only about 88% similar genetically. And that translates into like 400 million DNA differences. Okay, so what we have is a huge number of fixation events. Fixed in genetics just means to get stuck in place. Okay, so we need a huge multitude of fixation events since the split about 6 million years ago between humans and chimpanzees. We know fixation for the most part is relatively slow. So what there is something called a waiting time problem to even get enough fixation events since the split between humans and chimps for human evolution to even be true. But more so recently, when you can see the paper here, chimpanzee, this is a secular paper, this isn't Young Earth Creation. Chimpanzee and human Y chromosomes are remarkably divergent in structure and gene content. So here's what's interesting, is the Y chromosome is non-recombining DNA, okay? We only inherit that DNA compartment from our father. With the nuclear DNA, there's recombination, right? So it recombines, it scrambles, a little more messy. So the Y chromosome should be relatively more stable. So when they sequenced the chimpanzee Y chromosome and human Y chromosome and compared it, they were shocked. They were shocked that, notice here, more than 30% of the chimp Y chromosome lacks an alignable counterpart on the human Y chromosome and vice versa. Page's team found that chimp Y chromosome has only 2/3 as many distinct genes or gene families as the human Y chromosome, and only 47% as many protein coding elements as humans. So point is, the Y chromosome between humans and chimpanzees is only about 35% the same. It's hugely different, massively divergent. Too many differences to explain in just 6 million years since the split. Because remember, you got to consider every single human Y chromosome on the planet is nearly identical. That's how we can build these Y chromosome family trees, and we can trace our last Y chromosomal ancestor back to just 4,500 years ago, because there's low variation. But then the Y chromosome between humans and chimps is only about 35% the same. I mean, how are they going to explain that? Point is, that's exactly what we would expect from a creationist. But it's the genetics that is actually contradictory to the ape to man evolutionary story. So even the genetics is not on their side. The genetics themselves, the genetic data is refuting them. So if they go to the fossil record, okay, they're using fossils and bones to build a story that's not consistent with the genetics. The genetics comes first. That's the direct way to answer this question of ancestry. So they want to overturn the genetic data with their bones found in the dirt, their bone fragments. You see how circular and you see how unscientific that is?
Yeah, I didn't know this part, but this is beautiful. I mean, It's like a nail in the coffin.
Oh, it is. It is. I mean, I've debated PhDs on this, and I've brought it up to them, and you know, their best explanation is chimpanzees have had a wild sex life from generation to generation, where they have multiple partners, and therefore, you know, the sperm in their genetics on the male side is doing some crazy things enough to explain the divergence. But you know, this is all just a guessing game, all a hypothesis. They even admit this is a hypothesis. Put it this way. Okay, they say that our closest cousin is the chimpanzee, okay? It turns out that our Y chromosome as a human is more similar to the gorilla Y chromosome than it is to the chimpanzee Y chromosome. Well, that ruins their hierarchy. It should be humans and chimpanzees that are closer. Why in the world do we have the gorilla closer to us? And they'll say, well, it's the chimpanzees, the odd man out, because of their interesting polygamous way of mating all throughout history. You know, just stories, of course, to explain the data. But I mean, there's another nail in the coffin, right?
Yeah, but so if I understood you correctly, the Y does not really recombine correctly. Is that correct?
That's right. Yeah, it's what's called by genetics uniparentally inherited DNA compartment. Just means that it's only inherited for example, we got our Y chromosome from our father. We got our mitochondrial DNA from our mother. So there's two non-recombining DNA compartments.
Okay, yeah. So technically, it doesn't matter how many partners they would have. It wouldn't really change the Y, and you know, so their theory wouldn't really hold water.
It may degenerate it a little bit more. Yeah, I mean, there wouldn't be any recombination as a factor here. But they could also say, so we know that the Y chromosome actually mutates really fast. So here's a technical paper from Dr. Nathaniel Jensen. He's a Harvard graduate. He works at Answers in Genesis. And he looked at high quality mutation rate studies on the Y chromosome, and he realized that the Y chromosome mutates really fast, way more fast, like about 1 per generation. So this is the title of the paper. He demonstrated that the Y chromosome mutation rate and the number of DNA differences in the Y chromosome suggests a 4,500 year history, which is perfect because that brings us back to Noah, our last Y chromosomal ancestor. So the evolutionists will say, well, okay, the Y chromosome mutates fast. So that explains why the chimpanzee and human Y chromosome is so different. When in fact, it actually makes the problem worse for them. Because think about this. The Y chromosome mutates really fast, right? But when we actually examine Y chromosome diversity worldwide, we've realized that the Y chromosome, no matter what population you look at, it's the same. I mean, there's very low variation, very low differences in the human Y chromosome. So how in the world do you have a very quickly mutating Y chromosome that's also nearly identical worldwide? It should be different. There should be divergent people groups. There should be more diversity. Well, you want to know why? It's because it's young. It only goes back 4,500 years. So although it's fast, there's only been 4,500 years of a fast mutation rate. Does that make sense? It has to be young if it's fast and there's low variation. Because if it went back a couple 100,000 years, well, that would be a couple 100,000 years where we have mutations accumulating in the Y chromosome, which means the Y chromosome should have high diversity. So that actually doesn't help them either. That actually makes the problem worse. So like you said, it's nail after nail after nail in the coffin for the evolutionists.
Yeah, so it's interesting because even if that was the case, it would not just have to, like, a certain line of chimps have to be changed. They would have to have a similar, like pretty much one person's completely changed or one, animal changed, and everybody followed that. Everybody else died out, right? Because if it wasn't, it would still, they'd be closer to us than,
Oh yeah,
35%.
Oh yeah, it should be way. I mean, our Y chromosome is only like half the size of the chimpanzee Y chromosome. The architecture, gene content, totally different. For example, Call Me Emo in the chat says what I just said. He's an evolutionist. So here's his rescue device. He says, actually, it's the chimpanzee's Y chromosome that is significantly different. He says mainly due to losses. And that's what I said earlier, right? Is that's their story. It's the chimpanzees, the odd man out. And everybody else's Y chromosome is normal. When no, that's contradictory. The human Y chromosome mutates really fast. So if what Call Me Emo is saying is true, okay, that it's only the chimpanzee Y chromosome that's doing some wild things enough to make it so different, then we should find highly divergent people groups on this planet where the Y chromosomes are quite divergent. Okay, but no, we find one singular Y chromosome worldwide. I think there's about maybe 300 at most. It would be the max differences between any two people. So if you have a mutation rate in the Y chromosome that's 1 per generation, you can easily explain the maximum diversity in the Y chromosome in just 4,500 years. If what Call Me Emo is saying is true, we should find much more divergence in human Y chromosomes, and we don't find that. So every one of their stories and rescue devices is actually refuted by the empirical data. Like we can make a prediction. Like we can look to their rescue device and say, okay, that'll lead to a prediction. Let's see if it's true. And we find out it's false every single time.
Yeah, and another thing that you guys were talking about was the specifics of, like speciation, let's say it that way, how you can have one animal that, breaks down into, like, let's say there was a pre-cheetah, and now that is broke down into different cats. And you can technically explain it by the genetic code degenerating quickly. Yet we don't see that in humans. It's literally staying pretty similar if you look at it in that sense.
Right. Yeah. It's very similar. Right here. All human beings are 99.9% similar, identical in their genetic makeup. I mean, isn't that fascinating? Apparently, we've been evolving for millions of years, and yet we're 99.9% the same. And I was pointing out to Jackson, I was trying to find, I've got thousands of slides, so sometimes it's hard to find the exact paper, but here we go. inbred Neanderthals left humans a genetic burden. Remember, he was talking about how, Neanderthals are slightly different than us in their DNA. Well, of course, they were the most inbred people group on the planet. Their rungs of homozygosity are amazing. And the systems and the environments that they lived in this cold glacial type of ecosystem where the environment changed their genetics. The environment actually changed their morphology. That's why they're built the way they are, because they're adapted to that cold environment. So of course we predict them to be slightly different, but overall they're 99.7% the same to us. Meanwhile, chimpanzees are 88% the same. Call me emo in the chat. Look at this. He's going all the way to rats. So his rescue device now is this. Look, he says, Standing, do I have to bring up those rats again? So he's pointing out that there's rat species within the same genus that have massive differences, right? Well, when you look to rats and mice, just have a look at their reproductive rates, have a look at their generation times. I mean, they can recover populations quickly. Kind of like rabbits, they'll say breed like rabbits, right? But we're talking about humans. And he wants to, on the phylogenetic tree, he wants to go all the way down to rats and show that the Y chromosome is doing some pretty crazy things there. He's missing the point. Yeah, I admit the Y chromosome is mutating really fast. But you have to answer the question, then why is the Y chromosome so similar in every human male on the planet? If it's as wild as they say, and it mutates as fast as we know, then it should be, I think this makes sense on the panel, right? If we're accumulating that many mutations, then if we really do go back a couple 100,000 years, it shouldn't be as similar as it is. But no, it's very much similar because the Y chromosome's only a few thousand years old. So that's really,
that makes perfect sense.
All right, and welcome everyone to Standing for Truth Ministries. We have a good one today, a live debate between Jackson Roe and Standing for Truth himself, Donny. So this one's going to be good. The debate is on, is all life related through common ancestry? And just to let everybody know that there is going to be an after show on Logical, Probable, Possible's channel after this. He will have a link probably in the side chat. I think I just see it right now. So that'll be good. The debate format is going to be a 17-minute opening with a 10-minute rebuttal each person, with a 35-minute discussion and a 5-minute closing, followed by audience Q&A. So make sure to either tag myself or Donny or Standing for Truth. We'll see them on the side coming through, and we will gather them up. So don't forget to do that, because it is an indeterminate time that we will try to go and have questions get answered until we burn out, of course. So without further ado, I'm going to have our debaters give an introduction about themselves and why you guys are here, and feel free to take it away. We will pass it over to the channel owner himself. Wham, where are you in this corner?
Thanks for the opening there, Matt, our award-winning moderator. So I appreciate it. Matt was just here last week debating Jackson. I was moderating. Now I'm debating Jackson and Matt's moderating. So I appreciate that. And yeah, my name's Donny, also known as Standing for Truth. Usually I'm the one sitting here moderating. But tonight I am debating one of my favorite topics, which I know this is also Jackson's favorite topic, and I guess his specialty. So I'm looking forward to this. This is my formal debate number 95. If we were to count all of the impromptu debates that take place on people's channel, like Logical, Plausible, Probable, all the impromptu open mic debates, I'm probably at like 200 or something. But I love, these types of sophisticated discussions. Critical thinking is important. I've written numerous books on this topic, so I'm looking forward to this. You guys can check our website, standingfortruthministries.com, where we've got endless content, including, articles, books, videos, lectures, so on and so forth. So, yeah, enough for me. Thanks for doing this, Jackson.
Thanks for having me again. I'm pretty popular on this channel lately. Yeah, anyway, I crossed 400 subscribers, so it's a big deal.
Nice.
I mean, not really, but And kind of unrelated, something that does affect me though. They're passing it doesn't affect you at all, Donny. You're in Canada, but they're passing a bill that effectively bans all exotic animals from traveling interstate or being imported into the country. So anything past a leopard gecko, you're basically not going to be able to get anymore. So it passed the House. It's going to pass the Senate. It's pretty much a disaster. So call your senators if you can. I mean, well, not that ever works, but yeah, that's pretty much all I have to say. I just wanted to say that. Just do something.
All right. All right. If you guys are ready, then who is going first on this one? I'll get the timer.
I guess it'll be Jackson, since he's the affirmative tonight.
Okay.
As he would say yes to the debate thesis. So, Jackson, if you want to kick us off, my good man.
Go ahead and share my screen. Let me see.
And whatever time's not used, as we always do, Matt will throw it into the audience Q&A.
All right, can you guys see it?
Yes.
Yep.
Okay. This is the graph that Ken Hovind always uses to make fun of the idea of common ancestry. So I thought I'd just use it just to play into it. But yes, everything is technically related. That doesn't mean everything is closely related. Like the bird and the octopus, they're not very closely related, but they are distantly related by several 100 million years. Sounds crazy on its face, but it's true. Here's a phylogenetic tree of mammals, tracking several genes, and it makes a tree and a phylogeny that you wouldn't expect if they weren't all related somehow. It's just a very big coincidence that God would create something like this, especially created in 6 days, and have it all track like this. It doesn't make sense. Here's the overall tree of life, phylogeny of life: eukaryotes, bacteria, archaea, and they're all linked together, distantly, of course, but they are all linked. Now, here's a funny example. Hippos and whales are the two closest living relatives to each other, and artiodactyls are more distantly related. And whales came from artiodactyl ancestors. That's proven in the fossil record, like Pachycetus, an artiodactyl, but with a skull very much like a whale. And there's a clear progression, like Ambulocetus. Those are one of the ones everyone knows about. And there's Myocetus, which is a fairly recent discovery. And there's Georgocetus, which is basically just Basilosaurus with legs, more developed legs. This is Durodon, a basilosaurid whale. You can see the skulls are very similar throughout the progression. So has some hind limbs, but they're basically useless, definitely useless for walking for their intended purpose. They have heterodont teeth, unlike modern whales, molars, premolars, canines, incisors. They don't have a melon on their head. They have an intermediate blowhole, kind of halfway up the snout. They have a smaller brain, a lot of primitive features. So this in itself would prove that whales are from land animals. Even without the walking whales, this thing would do it, I think. Skipping right over to something totally unrelated, well, distantly related, of course. That's the argument. But turtles. This is what the turtle came from, a diapsid reptile with broadened ribs like this. Then it progressed gradually to something like Odontochelys, which basically had half a shell. It had a plastron, but no carapace. It had thickened ribs on its back, but no carapace, no true carapace. Skipping again to earlier in the the tree. This is Parmastega, which is a tetrapodomorph fish land animal thing. It's the best way to explain that to the layman. This is, of course, Tiktaalik, probably the most famous one on the list. It was predicted to be found exactly where it was found in Devonian strata, exactly where it was found on the place on Earth, Ellesmere Island, in the Arctic Circle. They went to the Arctic Circle expecting to find something like this, and lo and behold, they did, which is a pretty big coincidence. This is Elpistostegi, which is basically Tiktaalik 2.0. It has the beginnings of digits in its fins. This is Acanthostega, the next iteration. There's a little bit of a gap between the two. It's yet to be found. But this is Acanthostega. It has internal gills and fin rays and 8 digits. It's still technically a fish, but it looks very much like a tetrapod. This is Ichthyostega. In the center is the actual skeleton and a diagram or a CT scan. I can't really tell. But you can see this is still technically a fish with internal gills and tail fin rays. So very strange shaped fish. But it's got 7 digits, 3 digits forming into 1 digit. So a transitional structure. Then going back in time again, I don't have these in order. This is Bicellum brazeri, about 1 billion years old. The first known multicellular organism. It has 2 types of differentiated cells. Then we skip ahead a little bit to the Cambrian, one of the first chordates, Hycuictis. And the Silurian, one of the first jawed fish, maxillate placoderm, had premaxilla, maxilla, and a dentary bone, the first fish to have that feature, the first modern jaw. There are 4 species of those that are known. Now skipping way ahead, again, they're out of order. This is Australopithecus prometheus, the Littlefoot specimen. This was found almost completely articulated in Sturtfontein Cave, South Africa. It was encased in limestone. It took years to fully drill it out. But you can tell from the femur structure, the hip structure, the spine structure, the rib structure, the knee structure, the ankle bone. Everything about it says that it walked upright. This is the big man fossil, which is not very complete, but it's complete enough. It's an Australopithecus afarensis, the same as Lucy. Only this one is 5 feet tall. Again, has the knee, which creationists argue about Lucy's knee a lot. Well, problem solved. We found another one. And another one, in fact, on this one, Australopithecus afarensis. This is a juvenile specimen. Skipping back in time again. This is the mammal progression. In the early Permian, you had pelycosaur, synapsids, like these. This is Dimetrodon. Then you skip ahead to therapsids. In the late Permian, this is a Gorgonopsian therapsid. I forget what it's called. This is a cynodont of the mostly Triassic. More mammal-like reptile, but still a reptile. I already talked about this, but this is kind of limb evolution. This is Elpistostega. You see digits forming here. Acanthostega with the 8 digits. And Ichthyostega with the 7 digits. Now this one's one of my favorite lines, the theropod bird line. This is Anchiornis. I actually have a canvas of this one behind me. You'll see in a minute. 160 million years old. Here's what it actually looked like. They actually can tell the color of the feathers from fossilized melanosomes in the fossil. This is Eosinopteryx, another 160 million year old one, predates Archaeopteryx by 10 million years. Kai Hong, actually older, 161 million years. Aurornis, 160 million. This is Protopteryx, about 130 million years old. Now, clearly at this point, it's more bird than anything else, but it still has 3 unfused digits, teeth, and its feathers are much more primitive in structure. Another way you can tell things are all related to each other, chickens can still produce teeth. Dolphins can still produce hind limbs, atavisms. Here's something about more about whale evolution. They can tell those walking whales are whales because of the very diagnostic ear structure, not shared with anything else on the planet. This is an Indo-Hyas skull next to a raccoon skull. On the surface, they look kind of similar, but the teeth and, of course, the auditory bulla give it away. This in the middle is a pronghorn. Next to the walking whales, I'll just say walking whales. I don't want to say their names. It's too complicated. But they are clearly artiodactyls based on their ankle joints. Here's Basilosaurus, which everyone agrees is a whale, even big creationist groups like Answers in Genesis, but still has the same kind of ankle joints, kind of fused though, down to 3 digits each. So it's kind of wasting away as time goes on. This is a seal inner ear structure. I bring this up because I saw a talk or read an article once from David Minton, which I believe he recently died, and I don't want to speak ill of the dead. He seemed like a nice guy. But he was very wrong when he said that seals and these walking whales are related. He said they were pinnipeds, and that just doesn't make any sense. This is about single-celled algae. When introduced a paramecium, a predator, 50 weeks, 700 generations, 2 of 5 experimental populations developed multicellular life, a multicellular kind of colony. And they weren't just colonies, they formed a cell membrane, and they reproduced by kind of budding, I guess is the best way to put it, instead of cell division like these would in the single celled. So we kind of saw multicellular life evolve independently. And they stayed that way for 4 years. They ran the experiment that long. Here's a manatee ancestor, Pasosiren, a quadruped. And there's a line of those like the whales. They're just harder to find photos of. So here's a diagram of them. Here's some basic genetic evidence. I'll just read the titles for the sake of time. Reconstructing the genome of the most recent common ancestor of flowering plants. Last common ancestor of green algae and land plants. Ancestor of animals, fungi. Determining the divergence time of the major kingdoms of living organisms with a protein clock. Primate molecular divergence dates. So all this stuff is well known. I won't really go into it here because I'm not going to read all that and waste time. But yeah. One last thing. I got a bunch of stuff for rebuttals later, hopefully. I think that's where we're really going to get into it. This is just kind of breaking the ice. If everything was specially created, we would see weird animals like this that don't fit anywhere, genetically or anatomically. Like, what is this thing? Is it an amphibian or a bird or a mammal? I don't know. What's this? Or that? Or this thing? They wouldn't fit anywhere. But everything on Earth does fit in the tree, so to speak. And let's see what else I got. I think that's good for now. I'll end it by showing you this. It's a fossil whale vertebra I have. It's 5 million years old, so it's still technically an anatomically modern whale, but it's kind of cool. Thought I would share that. All right.
All right. Good one. You went 12 minutes. So we will add those extra to the Q&A or debate part. All right. And whenever you are ready, let me know and I will start the clock. And you are muted. Don't forget.
Yeah, just getting everything ready here. Let me see. And I'll be screen sharing in 1 second. We've got 17 minutes. So let me share a screen here real quick. And okay, let me start my timer. I appreciate that opening, Jackson. This is going to be fun. Okay, here we go. 17 minutes. Thank you for doing this debate, Jackson. I have written, as I pointed out, numerous books on this topic and look forward to a profitable discussion on it specifically. Here are some of the books I have written. So tonight we are debating the question, is all life related through common ancestry? Jackson here tonight has the burden of proof, as he is in the affirmative. He must provide convincing evidence that not only dogs and wolves are related, but that dogs, wolves, banana plants, and whales are related through common ancestry. Does Jackson believe he is related to a mosquito and a strawberry? The answer is yes, as he demonstrated in his opening statement there. And it is his burden tonight to demonstrate this. Agnostic lines of evidence will not help Jackson win this debate. He spent his entire opening going over agnostic or non-discriminatory lines of evidence. What I mean by that is the sky is blue and the earth is round. Okay, that is not evidence for either creation or evolution, as in that can't help us differentiate between the models, since both evolution and creation can explain why the sky is blue and the earth is round. Jackson must present differentiating lines of evidence that can distinguish between both universal ancestry and separate ancestry. Jackson must explain how universal common ancestry and its proposed mechanisms can account for the four-dimensional code. The genome is not what evolution expected. The examples of mutations we have are not the types required for evolution to advance. Evolution has to explain how the four-dimensional genome with multiple overlapping codes and chock full of meta-information came about. Genetic degeneration is a fatal problem to Jackson's position tonight. What is genetic entropy? It is the genetic degeneration of living things. Genetic entropy is the systematic breakdown of the internal biological information systems that make life alive. Genetic entropy results from genetic mutations, which are typographical errors in the programming of life's instruction manuals. Essentially, evolutionists like Jackson look to mutations as being the source of all genetic variation that natural selection can act upon. Unfortunately for Jackson, natural selection is very limited. It is a fine-tuning mechanism that keeps a species as strong as it can be. It doesn't create anything new. It only selects from what is already there. And mutations, the so-called source of new genetic variation, systematically erode the information that encodes life's many, many essential functions. The human mutation rate is roughly 100 new mutations per person per generation. The 8 mutation rate is similar. Almost none of these mutations are beneficial. The new ENCODE findings reveal that most of the human genome is functional. How does Jackson address this? I'd like him to address this in his rebuttal. Hence, most random changes in the genome must be deleterious. Even if 90% of the genome is perfectly neutral junk, which is no longer feasible, there would still be about 10 harmful mutations arising in every person, every generation. Even the most fit individuals are still more mutant than their parents. So even intense selection against the less fit cannot stop mutation accumulation, because most deleterious mutations cannot be selected away. They will accumulate continuously from one generation to the next. Now, we understand genetic entropy on a personal level, but since mutations are passed on from generation to generation, there is also genetic entropy on a population level. Why can't natural selection filter out all the bad mutations? There are 2 reasons. There are too many bad mutations continuously pouring into the population, and B, most bad mutations are too subtle to be selectively removed. Epigenetic mutations will consistently magnify this problem. We also have polyconstrained DNA, the fact that most DNA sequences are polyfunctional and so must also be polyconstrained. This means that DNA sequences have meaning on several different levels, and each level of meaning limits possible future change. For example, imagine a sentence which has a very specific message in its normal form, but with an equally coherent message when read backwards. Now, let's suppose that it also has a third message when reading every other letter, and a fourth message when a simple encryption program is used to translate it. Such a message would be polyfunctional and polyconstrained. We know that misspellings in a normal sentence will not normally improve the message, but at least this would be possible. However, a polyconstrained message is fascinating in that it cannot realistically be improved. It can only degenerate. We also know that the evolutionists look to beneficial mutations to supposedly counterbalance the damage done by the accumulating genetic load. Unfortunately, most mutations are reductive. They are rare. And as you can see here, most so-called beneficial mutations that they can point to are degrading to genetic functionality. That is a problem for Jackson here, unfortunately. The arguments that have been put forth against genetic entropy, I'd be curious to see what arguments Jackson here looks to have been systematically analyzed. For example, here on the slide, we've got junk DNA, mutation count mechanism, truncation selection, synergistic epistasis. They've all been looked at and they've all been falsified. As you can see here, the human germline mutation rate, 100 de novo mutations, our future genetic load. Human mutation rate revealed, and you can look at these paper yourselves. Right here, slightly deleterious mutations. This isn't young Earth creationists making this up. Okay, they you see in the literature from population geneticists, very slightly deleterious mutations. We are degenerating every single generation. It seems unlikely that any mutation is truly neutral in the sense that it has no effect on fitness. All mutations must have some effect on fitness. Nearly neutral mutations, again, it seems unlikely that any mutation is truly neutral. The question is, these mutations are unselectable. They're unselectable. So what type of selection can Jackson here present us today that can stop the degeneration process? Because selection can't see most mutations. We know mutations degrade genetic functionality and information content, and mutations are primarily why we die. And as we pointed out earlier, there is genetic degeneration on a population level. Now, what's interesting is Jackson's opening statement is he spent a lot of time on what's called reconstruction in phylogenetic systematics. Okay, and a lot of this involves imagination. Most of Jackson's arguments involve just that. For example, fossil reconstructions and interpretations that Jackson looked to in his opening statement, and these so-called transitional forms in the fossil record, quote unquote, universal common descent is void of an adequate mechanism. As we have seen, most mutations are invisible to selection. They build up and degenerate. Natural selection is limited. It doesn't create anything new. And mutations in natural selection are simply not up to the task to take a single-celled like ancestor into a whale over billions of years. Without an adequate mechanism, any historical reconstruction lacks explanatory power and is therefore not to be viewed as a correct reconstruction. Jackson's reconstruction interpretation of the data, as seen in his opening statement here, including the fossil record data that he spent a great deal of time on, is incorrect. Jackson assumes universal common descent, and because of this presupposition, he imposes it onto the genetic data. This is not a conclusion drawn from the genetic data. Remember, genetics is the best way to answer the question of ancestry. Okay, genes and traits are what is inherited, sperm and egg. Not a fossil, which Jackson looked to, not a bone, not a reconstruction, not a picture. We saw a lot of pictures in his opening statement. Not geography, no, genes and traits. And genetics has refuted universal ancestry. Genetics has actually confirmed separate ancestry, and we will get into this a little bit more later. Okay, let's get into some of the most common lines of evidence for universal ancestry and demonstrate why they are not good evidence for fish-to-fisherman evolution, or specifically why they don't help Jackson meet his burden of proof tonight. Notice on the slide here, we have transitional forms that Jackson spent some time looking at, homology, and nested hierarchies. Okay, we have nested hierarchies here. And let's examine each one here. Homology. The biological phenomena that we've explored parallel the properties of family trees, but they also have strong parallels to another concept. In other words, a competing explanation exists for all the evidence we've just discussed. This competing explanation becomes clear with an example. Okay, so we know humans design in homologous patterns. Okay, take the sedan as an example. Whether sedans are manufactured by automakers based in Asia, North America, or Europe, all share a common set of features. You won't find cars without wheels, engines, doors, or windshields. Despite the geographic distance separating their locations, sedans made by Honda, Chevrolet, and Mercedes have four wheels, doors on the side, windshields in the front, brake lights in the back, and a host of other shared characteristics. Now, the reason for this similarity is not common ancestry. Honda, Chevys, and Mercedes did not evolve from a common sedan ancestor in the Arctic. Rather, because these cars were designed for similar functions, these purposes put constraints on the final product. We can go on and on about homology. It's exactly what we'd expect given the design perspective. Same thing with shared embryological patterns. We could extend this logic to the manufacturing process where we would see similarities in blueprints. In fact, it would be not until the end stages of these modes of transportation that we see the largest differences. Okay, same thing with nested hierarchical patterns. Under the young earth creation model, God designed the entire universe, including the various kinds of biological life that exist in it. And we would expect to find that life fits a design pattern. Since humans are made in God's image, we can get a sense for what kinds of design patterns God may have used by examining the patterns that result from human designs. For example, design means of transportation easily fit a relative hierarchical pattern. This fact is unequivocal. Sedans resemble SUVs more than they resemble tractor trailers, and all three vehicles have more in common than do sedans and amphibious assault vehicles. The latter two vehicles have more in common with one another than with submarines, and this simple pattern matches the type of hierarchy that we see in biology. Again, relative hierarchies are characteristics of design. Of course, we would expect more similarity in genetics, morphology, anatomy, and physiology between a human and a chimpanzee than between a human and a dog, or between a human and a banana. It's exactly what we'd expect. Transitional fossils that he looked to. Okay, these parallels between design and biology extend even further. In our discussion of the deeper fossil layers, which Jackson spent some time on, we observed the presence of fossils that seem to blend the features of two different categories of creatures. Some products of human design also seem to span two categories. As we've observed, boats and tanks would fall into entirely different classes. Yet vehicles exist that blend the features of these two very different types of transportation. For example, amphibious vehicles resist classification. They're the perfect example of a design transitional form, designed for the transitional environment between water and land. In fact, if you were following my classification scheme for vehicles, you might have been mentally protesting. This is a source from Dr. Nathaniel Jensen, where he points out that we have an increasing number of hard to classify vehicles. For example, crossover vehicles, where the distinction between cars and SUVs has become blurred. What is currently labeled a car and labeled an SUV is now a matter of debate. These are designed. Okay, these did not evolve. These were designed for a purpose. We see the Jeep Gladiator here, the Dodge Journey, the Ford Flex, an amphibious assault vehicle. To the evolutionists, this is the perfect example of a so-called transitional form. Notice the slide. We have a unicycle, a bicycle, a tricycle, and a four-wheeler. I could predict, because Jackson talked about predictions with Tiktaalik and so on and so forth, I could predict that since we have unicycles and four-wheelers, there must be evidence for a tricycle and a bicycle out there. And it just so turns out that's exactly what we find. This is not evidence that the unicycle evolved into the four-wheeler. Rather, this is evidence of design. Each design is suited for what it was meant to do. Vestigial organs and structures overturned. What we see is function, function, function. The chromosome 2 fusion has been completely overturned through genetic related data. There's a huge problem that I want Jackson here to address, because we have these very large satellite sequences at the ends of the chromosomes of chimpanzees and apes. Okay, but what we find is that these large ape-specific satellite sequences that should be seen flanking the reputed fusion site on both sides, they are not there. They are not there. They are absent. This is a very serious problem that I would like Jackson to address if there really was a chromosome 2 fusion related to human evolution. And you can find more about this in the Ventura paper. So as we can see here, pseudogenes overturn the so-called genetic identity of human to chimpanzee. Actually reflects, when you consider all genetic regions, only about 88% similarity, which is 400 million DNA differences. There's a huge waiting time problem to fixate that many DNA differences. In my last couple minutes here, I'm just going to cover a few lines of evidence that suggest separate ancestry. Even though the burden of proof today is on Jackson to demonstrate that whales and banana plants are related through common ancestry, agnostic lines of evidence are not going to do it. But there are differentiating lines of evidence that actually support the separate ancestry model. For example, we have a genetic boundary study that was done showing clear genetic boundaries in the CO1 gene through these barcoding studies, and they demonstrated that over 90% of all species arose at the exact same time. The only historical record we have of a recent sudden origin of the diversity of life with distinct boundaries is in the opening chapters of the book of Genesis. The Bible's account is vindicated by science once again. We have the Y chromosome data, where we know the Y chromosome is mutating incredibly fast. Dr. Nathaniel Jensen has discovered that the Y chromosome really only goes back 4,500 years to Y chromosomal Noah, and there's been subsequent papers going over the predictions that are derived from that model. Mitochondrial Eve, the mother of us all, we know that the mitochondrial DNA also mutates fast. There's incredibly low variation. And if we look at a phylogenetic tree, for example, here we see that perfect starburst pattern. Explosive growth from a center point. We only see a few mutations, low variation. We see in some of these haplogroups, for example, HVR, we have some people picking up more mutations than their cousin in the same amount of time. That destroys the evolutionary molecular clock. This is exactly what we'd expect given the separate ancestry model. How does Jackson address this? How does Jackson address the evidence from orphan genes, these taxonomically restricted genes that are exactly what the separate ancestry model would suggest? Time flies by. I've only got 30 seconds left here, but we could go on and on and on about the evidence for separate ancestry. This is a debate on universal ancestry, so the burden of proof is on Jackson. But I can tell you, we could spend hours and hours going over the amazing lines of evidence that confirm separate ancestry, and there's 17 minutes, so I will call it quits there. Thank you so much.
All right, Jackson, if you wanted to share screen, go ahead, and I will start whenever you are ready.
I'll share screen here in a minute. I got some notes first. First, you said mutations can't make new information. Well, there's duplication mutations, which can work with existing stuff, copy it, and then it changes through further mutations. Then there's de novo mutations, which I know you expressed you don't believe in those, but we can get into that later. I've got some notes on that. Then there's horizontal gene transfer from one organism to another, which is not novel in itself, but it's novel to that new organism that it's getting from. Now, genetic entropy. I've done a lot of work on genetic entropy recently. I'm doing some calculations. I'm trying to equate, for example, let me find it. I got a ton of notes. Like influenza B, it's existed for 4,000 years. It has 14,500 nucleotides, one mutation per cycle. Sometimes some sources say 2, but I said 1 for the sake of argument. It replicates every 6 hours. So you take the amount of human nucleotides, divide that by 14,500, divide that by 100 human mutations, comes out to 4,000 something. 4,000 years times 365, that comes to 1.4 million times 4, because 6 goes into 24 4 times. Comes out to 5.8 million. Multiply 5.8 million times 1 times 4,000, comes out to 23 billion. What am I talking about? Some people are probably lost. I was explaining this to my family, and they didn't know what I was talking about. But I'm equating mutation rates of viruses with the mutation rate of humans and trying to see when genetic entropy takes hold. According to this, it would take humans 23 billion years to suffer some sort of genetic entropy. Viruses mutate very fast, replicate very fast, and they don't have the benefit of recombination that sexual organisms do. Now, we can get more into that later. Let's see, functional genome. See, you actually, a long time ago, I actually wrote this down somewhere. The functional genome, you said in the side chat of one thing, we were having sort of a little debate, Donny. You said evolution should be selecting out non-functional DNA, and that would disprove evolution. Then you said functional DNA proves that evolution is not real. So it's kind of a contradiction. You can't have your cake and eat it too there. A functional genome, I don't see how it would disprove evolution. I don't see how a totally not, 90% non-functional genome would support it necessarily either. Now, John Sanford, he's worked on this mutation rate. He's equated, he said H1N1 went extinct from slow error catastrophe, he said in his paper. He didn't use the word genetic entropy, but that doesn't really make any sense. Like I said, the influenza B has existed for 4,000 years and has a fairly rapid mutation rate, and it hasn't suffered any ill effects. And a question I have about genetic entropy, which Donny can answer at some point: at what mutation rate over what period of time with what genome size will result in extinction by genetic entropy? No one can answer that question as far as I can tell. Now, I will share my screen while I exit out of it. Hang on a second. Give me a minute. Sorry. You know what? I'll just tell you for now. I will pull it up, but save time. Georgia Purdom said in a slideshow, she had a slide that said there are no beneficial mutations. And below that, she said there are beneficial outcomes to mutations in certain environments, which is just saying there are beneficial mutations, because a beneficial mutation is not going to be beneficial everywhere. A beneficial mutation that lets you breathe oxygen isn't going to be helpful if you're in an atmosphere made of methane. Slightly deleterious mutations, I don't think they're much of a problem. Neutral mutations or near neutral mutations are needed for creating diversity in the genome. Can Jackson provide his calculations for critique? Someone says, yeah, I can later. Now, the walking whales I gave an example of, they're not just tricycles and bikes. They're like, how does it have a whale's ear and an artiodactyl's ankle? There's not really a great explanation for that. You can say, and did you find the unicycle and the strata below the bike, and then find the four-wheeler above the bike and the strata, and radiometrically date all those? I don't think so. Chromosome 2, you mentioned, I think something about probably the telomeres being too short. The telomeres fusion is drastically reduced to like a few hundred copies, which is usually much greater than that. I forget the number. But yeah, there's still obviously telomeres. It's still obviously a fusion. There's a degenerated centromere as well. You said 90% of all life on Earth arose at once. There's a problem with that. It doesn't really say they arose at once. It says they suffered some kind of genetic bottleneck at once, about 100,000 to 200,000 years ago, according to genetics. So that doesn't really fit in the creationist model. Chimpanzee Y chromosome and human Y chromosome. Y chromosome is at the forefront of reproduction. So it's going to go through more selection, especially with the Pan genus, the chimpanzees. They multiple males will mate with a female. So the most virile male has the most dominant sperm will pass on his genes. That's kind of how that works. Now, Jensen, you mentioned Jensen. One of his papers, I'm not sure if it was the one you brought up, but he used mitochondrial DNA and nuclear DNA, I think it was. I may be wrong on that. To set a new kind of creationist molecular clock, but it was contingent on it either being mtDNA or nuclear DNA. And it turns out it was the wrong one, so it nullified his whole paper. Now, let's see. The mitochondrial DNA mutation rate, Jeanson also estimated, and it's totally different from any secular literature or any literature on the mitochondrial mutation rate ever done. It's 35 times faster than what everyone else says that the mitochondrial mutation rate is. The mitochondrial mutation rate is like 3,600 years for one mutation. And he said it's 35 times faster. I forget how I came to that conclusion, but it was a limited study, a small sample size or something like that. And he uses an unrooted tree. So a phylogenetic tree will be like a three-dimensional kind of graph. He uses it flat. He shows relatedness, but he doesn't show ancestry. So it's kind of a misleading graph. I don't know if he does that on purpose or not. I don't think he does. And the last thing I've written down is orphan genes. Like there are different genes that aren't shared between us and chimpanzees. Well, there are fruit. We talked about this before too, in another. We kind of had this debate already, Donny. But there are orphan genes in different species and even different populations of fruit flies. Now, I think we would all say that fruit flies are related to other fruit flies. So orphan genes aren't really a good line of evidence to disprove common ancestry. That's all I have written down. We can get more into it later, obviously, but yeah, I'll hand it over to Donny.
All right, pretty good. 9 minutes.
Okay.
All right, let me get my timer going here. Time flies by in these openings and rebuttals, don't they, Jackson?
Yeah, they do.
Quick sip of water. All right. I've got 10 minutes on the clock starting right now. And I've got quite a bit of notes based on everything you said, Jackson, and we can do some serious discussion as well in the discussion portion. So Jackson spent a lot of time on phylogenetics. I did address a lot of this in my opening statement, but the reason why everything is connected is because life exists on the same planet. We are made of the same DNA, RNA, proteins. We breathe oxygen. We eat similar foods. Therefore, similarities in DNA, morphology, anatomy, and physiology is expected by the creation model. Does Jackson propose God should have used different building blocks for each form of life? DNA, RNA, and proteins for some, and then not for others? This argument doesn't really make any sense. And it's also a misrepresentation of the creation model that suggests God created us in his image. Therefore, there should be something about us that reflects the divine. And we've predicted that humans will design things in a way that can give us an idea or sense into how God designed things. And it just so turns out that humans build in homologous patterns. They build so-called transitional forms, right? These nice, interesting mosaics. And they also build in nested hierarchical patterns. Jackson talked about whales. He talked a lot about so-called transitional forms and fossils. He looked to the fish to tetrapod series. Tiktaalik stated that evolution has made fulfilled predictions given the fossil record. Well, Tiktaalik failed miserably because they found tetrapod foot tracks or footprints earlier than Tiktaalik in Poland. Okay, so Tiktaalik wasn't transitioning into anything. We see significant overlap. We see geographical mismatches. And again, Jackson is presupposing that evolution happened. And so this is a conclusion or interpretation that's forced on the genetic data, which is the direct lines of evidence. Okay, his interpretations are wrong given the direct lines of evidence. He talked about the human evolution related candidates, the hominids. He discussed Afarensis, Prometheus, and others. He also discussed the mammal-like reptiles. And he looked to a lot of specific features in morphology and anatomy, such as ankle joints, ear structure, and so on. The problem with Jackson's opening is that we don't have the genetics for most or even all of his examples. Today, we know that there oftentimes exists more variation within the same species than across species. This means without genetics and breeding tests, we can't always tell the difference between homology and convergent evolution. I want to ask Jackson that question. If you have a bone found in the dirt and you don't have any genetics or breeding tests, how can you tell the difference between homology and convergent evolution? Anytime convergent evolution is invoked, it is an admittance by the evolutionists to the lack of uniqueness in the universal phylogenetic tree. So that is a problem. And that's why genetics is the best way to determine ancestry. Not a bone, not a fossil. A fossil is not inherited sperm and egg. If every single dog type today was buried in a flood and dug up thousands of years from now, without the genetics and breeding tests, we could make all sorts of erroneous interpretations or fanciful stories that may or not be true. Okay. The problem with Jackson's understanding of genetic entropy is the fact that, because he said that these neutral mutations build up and they act as a hidden reservoir, essentially, of genetic diversity that natural selection can eventually act upon for future novelty in terms of large-scale evolution. But here's the thing. These neutral mutations are not actually neutral. They're effectively neutral. I showed a number of quotes demonstrating that these mutations are only slightly deleterious. It's like a one single spelling mistake in a book, let's say the size of an encyclopedia, okay, or rust on a car. These build up. On their own, they're inconsequential, but over time, the individual mistakes accumulate, just like in a book or on a car with rust marks. In the genome, these individual mutations build up, they degenerate, and the message is eventually lost. Okay, it's not the individual mistakes, it's the accumulation of these individual mistakes. So Jackson wants to assert that the accumulation of these individual mistakes will eventually be called upon or acted upon for evolutionary advantage, when in fact the buildup of these mistakes actually degenerate. Okay, it's going the wrong way. The change is downhill, it's not uphill. We know that selection involves the death of individuals, so selection is extremely limited. His mechanisms are limited. He doesn't have a mechanism. Therefore, don't tell me that it happened if you can't tell me how it happened. You can't select away an entire population, since that would result in immediate extinction. Selection can only ever slow down the degeneration process. It can never solve the degeneration problem. And I like the way Dr. Robert Carter has pointed out that if our species can't persist for millions of years into the future based on mutation accumulation, which puts shelf lives on genomes, that means our species has not persisted for millions of years into the past. Okay, as a matter of fact, if we look at humans in the fossil record, since Jackson likes to look to the fossil record, we see no evidence of hundreds of thousands of years of human existence. Okay, given the evolutionary model, we can assume that there were about 1 million Homo erectus living across Eurasia, Africa. I mean, they handled a massive distribution. Okay, if we look at their average generation time, let's say it was about 20 or 30 years. That means about every 20 years, you have another 1 million dead bodies. And so after 1 million years, we have over 20 billion dead people. This is 20 billion people, 20 billion Homo erectus, of which we have no evidence ever existed. Okay, we know that Erectus was a sailor. They were a sophisticated people group. They were not primitive, so therefore they would have had campsites, villages, fire. We should have a ton of evidence for their existence, and yet we do not. In my last 4 minutes here, I do want to share screen real quick because I want to address some of his specific examples. And let's see here. I'm sweating. We got a lot of points to address. So the Australopithecines that he focused on, okay? Australopithecus literally means southern ape. As the name suggests, the Australopiths have the overall body plan of an ordinary ape. Their anatomy indicates they were best suited for life in the trees. It's not surprising that they should share most of their features with living African apes today. They're around 3 to 4 feet tall with long arms and short legs, ape hands and feet with long curved fingers and toes, opposable thumb-like toes, ape size and ape-shaped skulls, funnel-shaped rib cages with high-placed shoulders for suspensory behavior. They also exhibited their own unique morphology, which is what we'd expect in the pre-flood world and the immediate post-flood world. Okay, the Australopithecines that are found are typically post-flood. Here's the thing. In this post-flood world today, this is only a snapshot of the overall diversity that ever existed on the planet. So we're only getting a snapshot. Look at how many rhino species today exist. 5. Look at the fossil record. We've got a massive variation of just the rhino type in general. I mean, rhinos with huge horns, middle-sized horns, no horns, small horns. There is a greater amount of diversity in the pre-flood world. So what we see in terms of Jackson's examples here are just examples of pre or post flood genetic diversity, and he wants to line it up any way he wants. With that amount of diversity, you can make any story. You can make any story from that. We can get into the ordering of the fossils a little bit later if he wants to, but I do want to touch a little bit on his other examples in the last 2 minutes. So the therapsids, mammal-like reptiles, for example, I had a paleontologist on, Dr. Matthew McLean, and he did a presentation showing discontinuities rather than continuities in the therapsids as well as Dinosauria. So we actually see genetic boundaries and about boundaries in the fossil record, including created kinds of the therapsids. We see that evolutionists use selective data using the fossil record selectively. I talked about geographical mismatches that we find in the fossil record as well. Another good point, similarity does not equate relationship. I need to know from the fossil record, okay, how can Jackson differentiate between homology and also convergent evolution? And we have example after example of convergent evolution here that essentially is just a rescue device. Okay, we could go into all the whale evolution examples. Pakicetus, there's been a drastic revision with Pakicetus, Ambulocetus as well. We know that the so-called vestigial hind legs are utilized and functional for reproductive purposes. Okay, the presumed transitional forms, including Pakicetus, Ambulocetus, and Rhodocetus, are highly questionable. The claim that these are all whale precursors is based more on speculation and wishful thinking. Remember, a lot of this fossil record data, guys, is highly fragmented. Again, we don't have the genetics and the complex adaptations that would be required to convert a land mammal to a fully aquatic whale are unimaginable. Even according to the evolutionist time frame, there's a huge waiting time problem in terms of fixating. Fixating just means to get stuck in place. These amount of beneficial mutations for this type of evolutionary transition. I've got water to land refutations as well. Tiktaalik, again, failed prediction, but unfortunately I'm out of time. So why don't we end it there, and then we can get into a little bit more detail.
Awesome, man. You were on the timer better than me. That's good.
1 second to spare.
No problem. And just a reminder to the audience while you get ready is that I would like to tell everybody to tag me if you can, or just throw a question in the side chat and I will compile them for everybody. And Standing for Truth, if you want to play a clip for a commercial, let everybody know. It's okay if not. You've probably already gone. But what's going on is we have a lot of debates coming up. Oh, thank you for
drank too much coffee. That's what's going on.
All right. Here we got some questions coming in already. So I'm posting them in the side for you as they come. This is great. Thank you, everyone. These questions are I didn't have any yet, so this is really good. All right. Here we go. Thank you, everyone. And we have Donny working on a couple things right now. One of the things that's keeping him busy is the production of a new book coming out. And as you would imagine, trying to do debates and write at the same time is really hard. So he will be quite constrained. I don't know. I think he's taking debates, but another question for Jackson. Here we go. From SpongeBob himself. Thank you, SpongeBob. Love the name, too. And thanks, George Bond. I got your question, and I got the question above that.
It looks like, Jackson, you won in terms of bladder endurance. So you took an hour. I had to go for 20 seconds. You win, at least in terms of that battle.
Yeah, I won the bladder battle, that's for sure.
And now we're good for 35 minutes. So whatever you want to talk about, we got a lot of points here to discuss. So
yeah, I got a ton of notes. You said this is the open discussion, right?
Yes.
Okay.
Open discussion now.
Therapsid kinds. Could you be more specific about what that means?
Sure. Yeah. If I screen share here, okay. So we have baraminologists, for example, Dr. Todd Wood, Dr. Kurt Wise, Dr. Matthew McLean. are a few that come to mind, and they have done studies and analyses using fossil data, morphology, anatomy. And what they do is they take a number of traits and characteristics and they look for continuities or discontinuities. Now, the created kind would reflect a continuous, right, where you have a group that has continuity within it, but outside of it, you have discontinuity. And that's what they've discovered, specifically Dr. Matthew McLean, when it comes to the therapsids, that there's enough discontinuity in the fossil examples or specimens that we can safely say that there were distinct created kinds of the therapsids. They've also done studies on Dinosauria and the in humans, where we have, this is the last thing I'll say, where we have the separate ancestry model of humans, the Homo group, ape-like group, or the Australopithecines, and then the artificial species wastebasket taxon. So this all comes down to, you know, the phylogeny challenge, or where can we separate ancestry. We've done just that in a number of groups, including humans, where we know that for the most part, we can categorize the fossil examples as either fully human, fully ape, degenerate ape, or wastebasket taxon, as I believe we see with some of the afarensis examples, but especially with habilis and sediba. Go ahead, Jackson.
All right. You're talking like what is it called? Dinocephalian and Gorgonopsian therapsids, like those are different kinds, maybe.
Yeah,
Something
you
like
can
that.
You can read the examples or watch the presentation, but here's a couple of the papers here that you can, you know, they're technical and long. But this one is specifically with the Dinosauria, baromonological and multivariate patterns, the proceedings of the International Conference of Creation. And he goes into the amount of characters and taxa and characteristics that they looked into. And they go through just how many possible created kinds there are in these fossil groups. But let me kind of turn this around on you, because even baromonology, when it comes to the fossil data, I think is also highly flawed, as in it's restricted. Again, it's genetics, genes, traits, and genetics is what's inherited sperm and egg, not a fossil, not a bone, not geography. So my question to you is, and I think this is the problem we also face when it comes to baromonology in just strictly fossil data. How can we tell the difference if we don't have genetics or breeding tests, Jackson? How can we tell the difference between homology or convergent evolution? Because today we know there oftentimes exists more variation or more diversity within the same species than across species. We see that with Canis domesticus, Canis lupus, Canis coyote, so on and so forth. So how can you differentiate that without genetics and breeding tests?
Like, let's take, for example, the platypus bill and the duck bill. On the surface, they look kind of similar, you know. Would you agree with that?
Yes.
Yeah. Well, when you get down to it, they're not very similar. The platypus bill is electroreceptive. The duck bill isn't. The platypus bill is leathery. The duck bill is solid. The bone structure is totally different of a platypus. I mean, there's convergent in the sense they're kind of similar and they kind of serve a similar function in some cases, but you can always tell them apart. There's not like a full duck bill on a platypus.
But we have platypus today, right?
Yeah, that's right.
So we can do breeding tests. We have their genetics. We have the genetics of the platypus.
Right.
Okay. So what about in the fossil record, since all the examples, correct me if I'm wrong, all the examples that you presented in your opening statement were from the fossil record and therefore had no genetics associated. Like, for example, Tiktaalik or from the fish to tetrapod series, do we have any of their genetics at all?
No, we can't. That's a testament to how old they are.
And that's,
I disagree with that one.
And that's the problem that I have. For example, we have we could do 6 pages worth of examples of convergent evolution. Similar physical mechanisms are used to transmit sound waves from the ear to the brain in both vertebrates and certain insects. So we have these highly complex features having to evolve sometimes 30 times, like when it comes to the eye. I mean, how many times apparently did the eye have to evolve? So the question is, if we don't have the genetics, if we don't have breeding test capabilities in the fossil record, how are you going to be able to tell the difference? Just using a fossil example, the difference between convergent evolution, right? The evolution of these features independently, but yet they're still similar, okay, but not related, at least recent relationship, homologous. How do you tell the difference just looking at a fossil?
Well, it would just come down to anatomy, and maybe you're right. Sometimes maybe they would get something mixed up, but with I've cataloged like 2,500 transitional species or genera actually. The odds are that one of them at least is actually a transitional fossil, if evolution is true, which I know is the kind of a false premise in your view. But
well, here's a problem I have. Oh, I'm sorry. Go ahead, Jackson.
I want to address the tetrapod thing. You mentioned Tiktaalik.
Yeah.
There were footprints found 10 million years earlier than that, but that doesn't necessarily mean Tiktaalik is not on the tree. It's a branch of the tree. That's all it means. And the foot, let me share my screen real quick.
Yeah.
The foot of the tetrapod that was found looks very similar to something like Ichthyostega. This was the footprints. These are the footprints. And this is Ichthyostega. So we have a very primitive foot with multiple extra fingers, so to speak. So it's not like we're finding a deer print, 10 million years before we find Tiktaalik.
So with these tetrapod foot tracks, okay, so here's a little more detail on it for the audience sake too. In 2010, it was reported that a well-preserved and securely dated landwalker tetrapod tracks from an ancient Polish marine tidal flat were found that were supposedly 397 million years old. Since these tracks would have been made before any of the creatures discussed in this article, and that would be the examples you look to, Ichthyostega, Acanthostega, including Tiktaalik. Right. The problem is, according to your model, right, Tiktaalik would be on the tree somewhere as a so-called transitional form,
but not necessarily the ancestor.
Right, not necessarily the ancestor. But remember, if we're comparing models and we're looking at agnostic lines of evidence versus lines of evidence that can differentiate between the models, this is not a problem for the creation model. Because again, we expect interesting mosaics, as I demonstrated in my opening statement. Okay, Tiktaalik being just that. We could look to Archaeopteryx. We can look to a platypus today. We can look to a seal, all sorts of different creatures. The problem is, for your specific model, okay, we've got tetrapod foot tracks before Tiktaalik.
Mm-hmm.
So one could say from the creationist perspective is we've got another interesting creature, just like we see all throughout the fossil record, just like we see today. It doesn't have to be interpreted as something that's transitioning into a tetra. If the tetrapod foot tracks are already there, pre-existing Tiktaalik, how's that a fulfilled prediction?
Well, here's the thing. Lobe-finned fish and stegocephalians are a very diverse group. We know about probably 100 species of both combined, and there are probably hundreds more we don't know about. They were distributed from the stegocephalians, like Tiktaalik. They were distributed from Africa, Europe, Russia, Greenland, Canada, and it's not likely that we found the exact line that led up to what we know today. And
when it comes to the lobe-finned go ahead, take your time.
I remembered. Lobe-finned fish evolved in the Silurian period. If you find a stegocephalian footprint before that, then there's a major problem. But there is, like you said, the fossil record's not complete. And that's what I would chalk that up to. But we haven't even found what made the track. The skeleton of what made the track.
So I would say the evolutionist has faith that hopefully in the future they find more examples in order that the fossil record can become more complete. But when it comes to these lobe-finned fishes that you're referring to, we see that they first appear suddenly in the beginning of the Devonian rocks, along with many other types of fish with no evolutionary precursors. But the point that I want to make here especially is that we have a coelacanth. Okay, the oldest coelacanth fossil is dated by evolutionists at about 408 million years ago. Right? While the coelacanth was originally thought to have been extinct, we now know that it's alive and well and adapted to live deep in the ocean. So lo and behold, today we have an example of a lobe-finned fish that at one point I believe was used, or maybe even still being used, as an index fossil, but today is a coelacanth evolving into anything other than a lobe-finned fish, Jackson, in your opinion, the coelacanth we have today, which I know there's some differences between the coelacanth today and the coelacanth in the fossil record, which we would say is the pre-flood world. That's expected, of course. We believe in change. But yeah, is the coelacanth today evolving into anything novel? Anything that's going to walk on land? Go ahead. Take your time.
No, it's not. And there are basically two clades of lobe-finned fish. Coelacanth is in the clade that's not as similar to tetrapods as the tetrapodomorph fish were. Like Gogonassus had a very tetrapod-like skull. It had spiracles for breathing air. Coelacanth doesn't have anything like that. Its teeth aren't anything similar to tetrapods. Its skull isn't really similar. It doesn't have spiracles. So it's a lobe-finned fish, but it wasn't really ever considered to be an ancestor. And their fossils have been found not only early, but they've also been found in like the Cretaceous and leading up. So there's a fossil history of them. They're not just in the Devonian and then now they're present, and there's no trace back in time, so to speak.
I would say, to at least conclude this one topic, because I do want to move to a couple other things you said. I think that the ultimate death knell here, just like in the article, for this story from fish to tetrapod evolution is what we were discussing earlier, these terrestrial tetrapod tracks that were found in rocks dated older than when any of these so-called transitional forms supposedly existed, because we, from the Cretaceous model, we would predict overlap, we would predict, and we see this in the fossil record, we would predict that the pre-flood world was a lot more diverse. And as I indicated in my opening statement, our model would suggest that we should be able to get a sense or an idea into how God designed the biological world and biological systems based on the way we design. And my question to you is, if you're familiar with these interesting crossover vehicles that seem to resist classification, and please, Jackson, don't say that these are not biological organisms, they don't reproduce. Listen, adding reproduction is even more of a problem for large-scale evolution, okay? So our model would suggest we should be able to get a sense for how God designed the biological world based on the way we design things, and we design interesting mosaics as well. We design what you would say is the perfect transitional form, as in your lines of evidence are agnostic to the debate, because what we see in the fossil record, what we see today is also reflected in the design world based on the way human engineers design. My question to you would be, with these crossover SUVs, I mean, is this right here, is this a Jeep or a truck? Is this an SUV or a van? Is this Dodge Journey an SUV or a van? Can you classify these for me, Jackson?
Well, if there is a God and he is guiding this process, this is actually a good analogy, because cars didn't just suddenly become cars. They were wheeling around carts not 1,000 years ago, and they technically evolved by the creator to these higher forms. So you could say that God is guiding the process in that sense, but I think God could be more creative if he wanted to be. I think he could put something out of a nested hierarchy or something that doesn't fit in the phylogeny. I think, he could do it the way you described, but why would he? I would be as creative as I would want to be if I was creating something like that.
Well, we do see a sort of descent with modification with cars. Let's say you go to the earliest Chevy Cruze and the newest Chevy Cruze in 2022, but each version, each model was designed, right? You could say there's an evolution in
not
the
necessarily saying
designs.
they weren't designed per se, but
now here's the thing: we as humans, we're just catching up in terms of design and complexity. We're just catching up to God, who understood this since the beginning. I mean, that's why we find these interesting mosaics. That's why we find that creatures have this built-in capacity to adapt based on environmental conditions and stimuli and stressors. For example, epigenetics, this extra layer of incredibly complex information where you have millions and millions of genetic switches just waiting to be turned on. Or through mutation, sometimes these switches are broken or permanently shut off, which temporarily usually has a type of advantage, even though it's overall reductive. So, I mean, the point is, God, his design in the biological world is a billion times more complicated and complex than us. I mean, today in 2022, we're just starting to design these types of modes of transportation or computer systems or cell phones. I mean, look at your cell phone. It has so many built-in capabilities where you get all these different apps, and it's all front-loaded into your cell phone device, things that God already thought of, knew of, and designed the biological world with. Like, how do you, from an evolutionary perspective, explain through evolutionary mechanisms like selection, mutation, the forward-thinking mechanisms?
Well, here's a good analogy.
Yeah,
the phone. Sure, the phone is there, and you could say it's front-loaded, but it's getting new updates all the time. You know, like new genetic information. I know you guys aren't big on the concept of new genetic information, but like de novo gene birth. But I've got some notes here if you want me to read them about that.
Sure.
There was a study on stickleback fish on ancient genes, novel genes, and newer genes. They can tell the difference between them because the younger genes are expressed less broadly and at lower levels. 38% of ancient genes were in all populations, while 17% of the new genes were. New genes were less CG depleted than cytosine guanine depleted than ancient genes. Younger genes were often gonad specific. Young genes are expressed in one sex and one gonad. New genes tend to overlap with copy number variations, etc. So, how would you explain that away? That
now, are you saying that these are de novo genes, the origin of like orphan genes?
Right.
Okay, so is this like an example, like the fish fly example that I showed? And here, I'll go to the orphan gene examples here. Is this like the, for example, they've done tests on fruit flies, and they've realized that in water fleas, so here's an example of the water fleas that contain many orphan genes that are only expressed under specific sets of environmental conditions and stresses related to water availability. So the point is, you can have examples of fish, you can have examples of water fleas, different types of creatures that have sets of orphan genes that are not expressed, but then bring in a stressor or put them in a different environment, and these orphan genes that are already there, they're pre-existing in the genome of the living organism, now they're expressed. They're being expressed. They're just like pseudogenes, for example. Many non-coding RNA genes like pseudogenes are only expressed under certain conditions, which means the researchers there analyzing this data, they're not actually seeing the de novo formation of a new gene, a highly essential and functional gene. Okay, no, they're just seeing the expression of a pre-existing gene. I mean, how is that explaining the evolution or the creation of the gene itself? Go ahead.
Well, it's just a gene made from a non-coding section of DNA, and they can. I don't know if they've ever seen a de novo gene being born, but they can tell young from old. And that's a pretty good indication that they're some of them formed more recently than others.
See, what they look to is either gene duplication. They'll look to, you get a duplicated region of the genome, and then the duplicated region undergoes what's called neo-functionalization. You get a series of hopefully beneficial mutations, and then you get a new function out of it. For one, there's a waiting time problem for that. We've never seen it. Yeah, as you're putting forward, they're sequencing these genomes, analyzing these genomes, looking at similarities. They're seeing a lot of orphan genes now being expressed, trying to say that, this is an example of a new orphan gene popping up. They oftentimes see the loss of orphan genes. We know speciation events, okay, variations within the same type. We know that these oftentimes occur due to reductions in genetic variability, allelic variability, shifts in heterozygosity to homozygosity. So by definition, you are going to lose variation. You are going to lose orphan genes. That doesn't explain the de novo origin of these orphan genes. And notice here, orphan genes are being demonstrated to have key roles in a variety of complex genetic and biochemical pathways in a variety of creatures across the spectrum of life. So this idea of de novo gene synthesis, this is the last thing I'll say, and I'll get your response, is it's circular. Okay, the evolutionists, the naturalists, they see these taxonomically restricted and essential genes, which is exactly what we would predict from the creation model. They see these, so they think, well, how did these come about? They must have come about through evolutionary processes. So they must have come about through non-coding areas of our genome, right? De novo gene synthesis. This is circular because all they're saying is, well, it couldn't have been created that way or designed that way. So why do orphan genes exist in the mind of the evolutionists? Is, well, because of de novo gene synthesis. But then why is there a process called de novo gene synthesis? Well, because of orphan genes. You see how circular that is? Instead of just admitting, no, these are fully integrated into the information systems of life. They're highly functional, highly essential. They couldn't have evolved. We haven't seen them evolve in the lab. Okay, observing the expression of a gene that's not been expressed, observing the loss of a gene, that's not explaining the origin of a gene. It's much more likely to lose a gene or that a gene will be broken down through a series of mutations than for a gene to be built up. I said a lot there, but yeah, what are your thoughts? How do you get around the circularity of de novo gene synthesis? And do we have any evidence of this gene duplication and neo-functionalization in terms of orphan genes? Go ahead, take your time.
Well, I'm not saying that epigenetics isn't a thing, or that orphan genes or pseudogenes don't have a function. Oftentimes they do. What was the rest you said? It's kind of a lot.
Well, how do you get around the circularity of de novo gene synthesis? We don't observe it today in a lab.
Right. They haven't observed it, I don't think, but they can infer it. They're not just saying, well, orphan genes exist, therefore they must have been created out of nothing. I think there's more to it than that. I mean, they're seeing signs that some genes are younger than others based on the cytosine, guanine, what's the word? I forget. But
right, they're just comparing
and they're
similar
more specific, the younger ones.
Yeah, they're comparing similar sequences, right? For example, the whole point, especially for the audience, they call them orphan genes because they have no ancestral sequences in other creatures that they evolved from. So they're looking at similarities in what the evolutionists believe is shared relationship, right? And they're hoping that, well, there's some similarities here. So maybe this is the precursor to the orphan gene. Maybe this is the precursor in the non-coding regions of the genome. Point is, it's non-empirical. They're not actually seeing, just like endogenous retroviruses, for example. Can you present me with a technical paper of a non-functional endogenous retrovirus or any type of retrotransposon that has gone from something that is non-functional to something incredibly and critically functional in the genome? For example, we know ERVs are functional in determining cell type, cell stress responses. ERVs are functional in the embryo. I mean, do you have any example of this being observed in the lab, this type of co-option?
I would have to do more reading on that. I'm not, I don't really have an answer to that one. But again, what was the first part? I keep losing track.
Well, I'll just break it down like this. How do you get around the circularity? I understand you believe that de novo gene synthesis explains the existence of orphan genes. You believe that. but we haven't observed it. It's not empirical. It's inference, and I don't even think it's a good inference. How do you get around the circularity of that?
Oh, you're talking about ERVs. That's what I was going to talk about.
Right. Okay, go ahead.
5 minutes.
Sorry to change the subject, but ERVs, you say, I think your model is that viruses came from the DNA, and that's why they're endogenous retroviruses. They didn't graft themselves into it. They came out of it, right? Is that right?
Yeah, the question when it comes to ERVs and other classes of retrotransposons, including these ALU sequences, is are these created units of DNA function? Or are they the result of past viral infections millions and millions of years ago? Now, I've shown numerous papers. For example, here's a bunch. Scientists identify new beneficial function of endogenous retroviruses in immune response. There's another one that has to do with regulation. The ALUs influence alternative splicing. Okay, regulatory activities of transposable elements. The pervasive retrotransposons that populate the genome have a powerful capacity to influence genes and chromatin. So on and so forth. Retroviral promoters in the human genome. So the point is, the prediction of the creation model is these were designed this way. There's obviously been rogue elements, mutations, things like that. Viruses in general are good, right? Healthy. They regulate the amount of bacterial species in our genome, for example. So that is the question, and we find function. And so I think the evidence best fits with the creation model. Yeah, go ahead. That's how we would explain like ERVs and retrotransposons. They're initially created, designed that way.
Yeah, but I think I've heard you say that viruses came from those segments of DNA. Is that right, or am I misrepresenting you?
Because, for example, I asked you this question, I think maybe last week, since a virus requires a host to replicate, right? Mm-hmm. What came first, the virus or the viral-like element or the host?
Right, the host probably would have come first.
Okay, and that's exactly what we're saying. And there's a model put forth by creationist PhDs. I wonder if I can find it here, but I got a million things here. So where is it? Okay, so the creation model. It is possible to reinterpret retroviruses. Okay, accordingly, retroviruses could have originated from within the host genome. Okay, you admitted that the host would have predated the virus. Okay, that's exactly what the variation-inducing genetic element hypothesis put forth by creationists suggests. Okay, accordingly, retroviruses could have originated from within the host genome instead of invading it from outside. This had to have been the case in any scenario since viruses are dependent upon host cells. This means that the cell's genome had to predate the viruses. You can read all this yourself. The point is, we find confirmation of this model and hypothesis based on the function that we find in paper after paper after paper when it comes to ALU sequences, endogenous retroviruses, all different types and classes of retrotransposons. So for your model and your explanation to have any validity, you'd have to show me how it's possible for endogenous retroviruses to go from non-functional to something extremely functional. Does that make any sense? And take your time, Jackson.
Yeah, well, here's the thing about your model. You couldn't make a virus out of the ERV most of the time if you wanted to, because it's so fragmented. They're fragments of a viral genome. They're not the whole viral genome. So you couldn't really, that wouldn't really work most of the time anyway. Maybe there's some hole, but from what I've read, it's pretty fragmentary.
Are you saying that the only parts of the ERV are functional?
No, I'm saying only parts of the ERV viral genome is there, like compared to viruses. I'm not saying it's only partially functional. I'm saying it's there. Some of them do have functions. I'm saying you couldn't make a virus from that if you wanted to.
See, here's the thing. We have just viruses in general. We have more viruses in and on our body than we have cells, and we have 100 trillion cells. I mean, that's amazing to think about, right?
Yeah.
And what are the viruses there for? They're there for beneficial purposes. They help regulate the amount of bacteria and bacterial species that we have in and on our bodies. They're good. Okay, when people hear the word virus, they think of something bad. Only very few viruses are actually bad and harmful, and usually it's when a virus or a viral-like element leaves its original host or its original environment, right? So the viruses that were created or that exist in our body that are beneficial, if they were to jump to another species and that other species can't regulate it, it can become harmful to that species. And oftentimes viral-like elements can break off in our genome and become rogue elements. And that's what we're saying happened with these endogenous retroviruses and these transposable elements. Like within the host genome, within these ERVs, you've got like the GAG and POL genes, which are functional to help with variation, things like this. But I don't want to spend all the time on this. I'll give you the last word on that. And then I definitely want to ask you a question that has to do with the rooted versus unrooted tree, because you made that argument in your rebuttal. So go ahead, take your time on the ERVs, have the last word on that specific topic, and then we'll move to this.
I'm kind of ERV'd out. Plus, I was reading the side chat, so I kind of lost track, honestly. We can go to this point you're about to make.
Okay, so you made the claim that, well, this is an unrooted tree. Mm-hmm. It should be rooted, right? But here's the problem, Jackson, is whether it's rooted or unrooted, that doesn't change the data. Okay, we still know that the mutation rate in the mitochondrial DNA, as well as in the Y chromosome, is incredibly fast. We know that there's incredibly low variation in both mitochondrial DNA and Y chromosome. Every single Y chromosome on the planet, male Y chromosome, is nearly identical. And you can take every single Y chromosome, build a family tree, and that family tree reveals that we have all descended from one single Y chromosomal ancestor in the not so distant past, okay? And the same thing goes with the mitochondrial DNA. The reason why Dr. Robert Carter, represents worldwide mitochondrial DNA diversity in an unrooted tree is because it gives a natural reflection or a natural reading or representation of the data. It allows the branches to flow out naturally, and that's why we see explosive growth from a center point. So why do you believe this whole unrooted versus rooted tree is an issue? Go ahead, take your time.
Well, the first part of what you said, I'm trying to remember, but I should have been taking notes. Repeat yourself a little bit.
It was the unrooted tree part.
Yeah, I know that. But
well, the first thing I said, right here. Here's an example. I like to, show the audience, show you all my sources. The human Y chromosome exhibits surprisingly low levels of genetic diversity.
Yeah, that's what I was going to say. Everything I've ever read about mitochondrial mutation rates say it's incredibly slow. It's like one for every 3,600 years. It was just one kind of creationist experiment that said it was faster.
Well, the latest mutation rate studies, for example, one in the D loop region, the control region of the mitochondrial DNA, they discovered it's about 0.5 per generation. Even if you went with 0.2 per generation, that's still too fast. You can go to the Parsons study, which demonstrated that if you were to take that mutation rate back, mitochondrial leave would go just 6,000 years. A high observed substitution rate in the human mitochondrial DNA control region. It's pretty well established that the mutation rate, we can do pedigree stuff. We can take great grandmother, grandmother, mother and daughter, and we can sequence their genomes and we can find DNA differences and we can determine a mutation rate, the pedigree based mutation rate. What you're talking about is the phylogenetic rate, which calibrates the known or empirical rate from pedigree-based studies. The empirical method, it calibrates the data, to the geological column, to evolutionary-based assumptions. But I pointed out, I think I had time in my opening statement, is the evolutionists can't make those assumptions. Because if you notice on this tree, go to the most common haplogroup in Europe, the HVR haplogroup. Notice the differing sizes of the branches. Well, what that means is people in the same time that go back to a common ancestor, they're picking up more mutations than their cousins. That means you can't make assumptions based on a straightforward molecular clock because this is chaotic. This is just DNA differences, mutations over a lot of time. And you can't then make assumptions that the evolutionists do about when humans and chimpanzees split from a common ancestor, let's say between 7 and, 13 million years. Okay, because what they'll do is they'll look at the amount of DNA differences that separate humans and chimps, and then they'll derive a mutation rate, and that mutation rate is very slow. But that's ignoring the observed mutation rate from pedigree-based studies is very fast. I said a lot there, but go ahead, Jackson.
Well, you mentioned they're correlating it with the geologic column. I don't understand how they're doing that.
Well, because if you, for example, we put out a video recently, Biblical Creation Basics, on mitochondrial DNA, and I think it was PBS. They put out a video where they admitted that they calibrate the data with what they believe is evolutionary history through the geological column. They'll oftentimes calibrate it with archaeology, right? They're not looking at the empirical method. They're not looking at known mutation rates from pedigree-based studies. No, they are calibrating it with their assumed history in the geological column. But here's the thing, it's the geological column that is in question. So that's circular. You can't refute the young earth creation model by calibrating the data with your geological column assumptions or your assumptions behind the human and chimpanzee split. That's circular. That's what we're debating here, is the existence of deep time. So we as creationists, we're just looking at the known mutation rate and we're like, wow, the known mutation rate through pedigree-based studies is fast. And we know there's extremely low genetic variation in the mitochondrial DNA. For example, here's a question I'll ask, and then please just take as much time as you need. I've been talking a lot here, Jackson. I don't want to dominate. So just take as much time as you want to address it. This is only a reflection of a few hundred generations, because on average, you got 20 to 25 DNA differences separating any two people in the mitochondrial DNA. Max, you've got maybe 120 if we were to include your longest lines like the Africans. So with a fast known mutation rate, without any calibration, without any evolutionary assumptions, how do you get hundreds of thousands of years out of this? Go ahead.
I don't know, but I'm not sure that they calibrate it the way you're saying. I know that like some things do line up, like the Australopithecus seems to line up about the time when the chimpanzee human split happened. But yeah, I don't have much of a response to that. But
okay, that's
other than to say that when it's rooted, it looks a lot different.
Well, see, here's the thing. They root it, though, based on evolutionary assumptions. Where they're picking the root is based on evolutionary assumptions. So it doesn't give a natural reading of the data. Whether we root it or unroot it, doesn't change the data itself. It doesn't change the mutation rate, doesn't change the diversity, the low variation, doesn't change anything like that. Unrooting it just gives us a natural reading or representation of the data. But we'll leave that up to the audience. With our last few minutes here, Jackson, I do want to ask you, because this is another thing you brought up in your rebuttal. And I think we do need a few extra minutes in discussion time because there's so many points brought up and we're discussing most of them. This is good. Your explanation for the chimpanzee and human Y chromosomes, the reason why they're so different, they're about 70% the same, okay, to the audience. But when you actually consider overall size differences, the fact that the Y chromosomes between humans and chimpanzees are only about half the size, when you consider overall gene content and architecture, it's actually only about 35% the same. So 70% the same, but when you consider everything, it's only about 35% the same. Okay, not to mention that the human male Y chromosomes have incredibly low variation and the mutation rate is fast. So how do you explain this data? And can you reiterate your argument in the rebuttal?
Yeah, the Y chromosome is a kind of the front line of reproduction and fitness, obviously. So it's going to be subject to mutations. And apparently, to answer another thing about the size difference, apparently chimpanzees suffered a lot of gene death in the Y chromosome and a lot of substitution. And yeah, I mean, again, the pan genus relies on a lot of males inseminating one female, and the fittest sperm inseminates the egg, or fertilizes the egg, rather. So that's kind of a reiteration of the argument.
Okay, so
kind of an answer.
So here's the thing. I've debated numerous people including PhDs, Dr. Stephan Frello, I've debated, I mean, Gutzit Gibbon numerous times, Dr. Ron Garrett, they never have an answer to this. I think this line of evidence, the fact that humans, the chimpanzee and human Y chromosomes are remarkably divergent in structure and gene content, I think this alone falsifies universal common ancestry, because there's no answer. There's just so stories, a lot of imagination in terms of the answer to this. And I've seen this with people like Gutzit Gibbon. They just dodge, duck, dip, dive, and dodge. I'm not saying that you're dodging, but here's the problem is the Y chromosome and the mitochondrial DNA, they are our uniparentally inherited DNA compartments. They're non-recombining, okay? In the Y chromosome, for the most part, they're non-recombining. That means they should be the most stable. That's actually why we look to the uniparentally DNA compartments, inherited compartments, to build these family trees that go back to one Y chromosomal ancestor, one mitochondrial DNA ancestor, who, by the way, are nothing like the chimpanzee. Nothing like the chimpanzee. They should have been slightly similar to the chimpanzee. But here's the thing. Because there's no recombination, they should be more stable. And yet, the Y chromosome was the most different between humans and chimpanzees. As a matter of fact, and address this one, the human Y chromosome and the gorilla Y chromosome is more similar than the human Y chromosome is to the chimpanzee Y chromosome. But wait a minute. The evolutionary story is that humans and chimpanzees split from a common ancestor 6 to 12 million years ago. And we have a more distant common ancestor with the gorilla. Why is the human Y chromosome more similar to the gorilla Y chromosome? And this is the last thing I'll say, and then you can yield, because this argument that the chimpanzee is kind of the odd man out and there's a lot of sperm competition, and therefore there's been more change and loss in terms of degeneration in the chimpanzee Y chromosome. They're the odd man out kind of thing. This is all nice. This is all cute. But without any numbers, without any data, without anything empirical, it's just a story. And Dr. Rob Carter points this out. And then I want you to take as much time as you need to respond. What are the implications of this new information? First, for the evolutionists, the Y chromosomes must be evolving much more quickly than anyone imagined previously. They are now going to have to apply. Notice this, Jackson. So when you respond, I want you to give me this data. I want an empirical answer. They are now going to have to apply, and this is something none of my critics have ever done, including the Gutsy Gibbons of the world, have to apply mathematical models to try to demonstrate how a sequence can change extremely rapidly. You can go with your chimpanzee sperm competition. Go with it. Show me how mathematically, empirically, we can get wholesale rearrangements of significant parts and the evolution of entirely new gene families in a relatively short amount of time, yet stay homogeneous. This is important, yet stay homogeneous within a species. Go ahead, take your time, Jackson.
All right, that was a lot. But first I'll say the gorilla and chimp and human Y chromosomes, I think the reason that the gorilla would be appear more close to us is because the chimp has gone through so much change. And with gorillas, they have a male-dominated society and multiple females, so there's not as much sperm competition. So that might give the appearance the Y chromosome of a gorilla looks like it's closer to us than the chimp is, because the gorilla is our next closest relative.
But are you done?
I forget the last part. I'm on a lot of gabapentin, so it's hard to remember stuff.
No worries, no worries. You're doing great. This is an awesome discussion. But here's the thing, Jackson. The evolutionists are always asking for data that contradicts phylogenetics, nested hierarchical patterns. Here we have a Y chromosome, which should be more stable. That's way more different, okay, in the humans and chimpanzees than between humans and gorillas. And again, the last thing I asked you for your response is, I don't want a story. Like, I know you believe that in terms of the chimpanzee, they're the odd man out, right? But as Dr. Robert Carter pointed out, you have to demonstrate this through empirical data. You have to apply this explanation, which hasn't been done, because the last paper I read on it, they admitted this is just a hypothesis. So they believe that this explains the incredible divergence between chimpanzee and human Y chromosome. But without the application of a mathematical model to demonstrate how these sequences in the Y chromosome can become so different, it's just a story. So go ahead. What are your thoughts on that?
Honestly, I have no answer, but I will look into it. And, you know, I've been doing a lot of math with this virus stuff. I'll look into that, honestly. So, yeah, I mean, I'm not a geneticist. I just got into genetics recently, doing some research off and on. Been doing a lot of viral research for this genetic entropy stuff. So I don't have a good answer, honestly.
No, that's okay.
The honest answer, yeah.
But I do want to say this, Jackson, before Matt jumps in. The debate is all life related through common ancestry. I think the genetic data, which is the best way to determine ancestry. Okay, we don't even have to go as far as whales and pine trees. The evolutionists like R and Ra, they always say, oh, these creations, they just want to go like, you know, the two furthest forms of life on the, tree of life and kind of make fun of it. No, let's go as close to home as possible then. Humans and chimpanzees. And guess what? The evolutionists didn't predict this amount of difference. They didn't predict that the human Y and the gorilla Y would be more similar than the human Y and the chimpanzee Y. It makes sense from our position because all genetic data suggests separate ancestry. God created Adam and Eve independently from every other form of life. We have a confirmed expectation and prediction here. We've got the Y chromosome that fits perfectly with our model. We don't have to make up any fanciful stories. It's the evolutionist that has the problem. And I can assure you, Jackson, when you look into it, there's been no sophisticated mathematical modeling being done. So if you find out that there's been no sophisticated mathematical modeling being done in terms of this Y chromosome problem, what's your conclusion going to be?
It's going to be that, well, first of all, let me ask you this. Why would we be similar to gorillas at all? I mean, well,
here's the thing. We are all made of DNA, RNA, protein, right?
Sure, but
DNA,
why are we
RNA,
so similar to the things that appear to have, through the fossil record and other means?
Well, what's more similar? What's more similar? A hatchback and a sedan, or a hatchback and a boat?
Right, but they don't have a DNA code that says they're very similar.
Well, they have a blueprint. They have underlying materials that are used, to design and engineer and build the products. But here's the thing: adding DNA, adding reproduction, that's just adding more complexity. Imagine we could do that.
Sure, it's very
Imagine we could do that. But my point is, what? To just answer what is more? Why is a sedan more similar to an SUV than a sedan is to a skateboard?
I don't know how to answer that, honestly. I mean, I'm kind of confused by it.
Well, because there's just more similarities in the way that the sedan is designed and the SUV is designed than between the sedan and the skateboard. By definition, when God created life, he had to create, Jackson, some creatures that are more similar to other creatures. For example, humans and chimpanzees, humans and gorillas, humans and bonobos, humans and...
But why do we have to do that?
Well, if we go back to our model at the beginning, do you remember what I said? The Bible says that we are made in the image of God. Therefore, there should be something that reflects us, that reflects the divine. About us, that reflects the divine, if we're made in his image. So we've formulated a hypothesis or a model that suggests, okay, if we really are made in the image of God, maybe we can get a sense for how God designed the biological world based on the way we design things. And it just so happened to turn out, Jackson, that we build in homologous patterns. We build in nested hierarchical patterns. We build interesting transitional forms. And that's exactly what we see in the biological world. How do you explain that? It matched up so perfectly.
Well, I would just say that God created us in the same category as the filthy apes. I wouldn't have done that as God.
Well, wait a minute. But here's the thing. If chimpanzees didn't exist, Jackson, then the evolutionists would just say, well, humans and gorillas, look how similar they are.
Well, that would be the next closest common ancestor.
Yeah. And then remove gorillas. Oh, well, the evolutionists look at, humans and bonobos. Look how similar, and then remove bonobos. Well, look at humans and lemurs. Remove lemurs. Look at humans and, the old world monkeys or something. So it's never going to be good enough for the evolutionists. Are you trying to say that humans, answer this question, are you trying to say that humans should be equally distant in morphology, anatomy, genetics, and physiology, equally distant, okay, from all forms of life? You have humans, and then everything else is equally distant. I mean, dogs, a banana plant, a chimpanzee, it's just equally distant to all three of those. Go ahead.
Well, not necessarily just humans. I think a lot of things should be distant from a lot of other things, and they're just not when you look right at it.
I would say it doesn't. I would say it's not very sophisticated thinking to say that if God designed the biological world, that we wouldn't expect more similarity between humans and chimpanzees, even before we had genetic sequencing technologies. Just stand back and look at the outward appearance of the organism. How many similarities do we have with the chimpanzee? You can easily predict, sure, in the DNA, in the genotype, we're going to expect more similarity between humans and chimpanzees than between humans and a dog. Just stand back and look at the outward appearance. But here's the thing. Did evolutionists, answer this question, did evolutionists make a prediction as to how similar genetically humans and chimpanzees would be before we sequence their genomes?
Well, it used a little bit of circular reasoning there, too, to say, well, chimpanzees look similar to us. That means they must have DNA. And if we say, well, the DNA looks similar to us, that means they must look similar to us. That's kind of some circular reasoning there as well. But
how's that?
What?
I'm sorry. Go ahead. Answer the second question. Yeah, go ahead.
Again, I forget the second question. Will you repeat it?
Did the evolutionary community make a prediction on just how much similarity there would be in the genetics between humans and chimpanzees before we discovered the similarity?
I'm not sure if they did or not.
I don't think they did. Now, I could be corrected on that, but I'm pretty sure they didn't make a prediction, which means it's not evidence for evolution. They never made a prediction like, well, humans and chimpanzees, they're going to be 99% similar, 97% similar, 95%. They never made a prediction. We sequenced their genomes. It turned out we're pretty similar, at least more similar with the chimpanzee than we are to
anything else. Yeah.
Now, if we didn't have these similarities, if we didn't have these nested hierarchical patterns, how could we ever advance in, let's say, the medical industry? I mean, when we're doing experiments, when we're looking for cures, when we're studying the biological world and finding out things about this amazing planet that we live on, if we didn't have these similarities in DNA, RNA, protein, so on and so forth, how would we ever advance? I mean, what do we use mice for, Jackson? I mean, don't we use...
I use them to feed snakes.
Right. But without the similarities in even mice, and the evolutionists will say, well, the reason why humans and chimpanzees share more than humans and mice is because, humans and mice have a much more distant common ancestor. That's where they look to the DNA differences. But I would say the lack of uniqueness displayed in things like incomplete lineage sorting, where you can look at different genes and come up with totally different trees, convergent evolution, loss of traits evolution, these types of things that demonstrate a lack of uniqueness in the phylogenetic tree. It's nothing but a rescue device and a story from the evolutionists when it comes to the inconsistencies. They rarely want to focus on the inconsistencies. Jackson, have the last word. It looks like we've already gone 15 minutes over the discussion time. Have the last word in the discussion, my good man. I said a lot there.
I'll just say that was a lot, but I'll just say you're basically saying everything was created with DNA hierarchies and anatomical hierarchies for future medical research, looking forward thousands of years. And I don't find that a very good explanation, but I mean, we all have our own conclusions, so.
You can have the last word, Jackson.
That's it.
All right, then. We are going to jump into our closing statements, which are about 5 minutes long, and then go right into Q&A. So, who is going first on the closing statement?
I guess it's
Looks like
me.
you
You
are.
zoomed in on me.
Yep, looks like you're up on this one. Okay, 5 minutes
All right.
whenever you're ready.
Good debate, good discussion. I love doing this stuff. I forget 90% of what I say during the debate, so I don't know how I did at all. I might have done terribly, might have done great. I have no idea because, again, I'm on a lot of Gavin. That's the joke I like to use. But yeah, Donny and I are friends, and nothing will change that, I don't think. But I do say the evidence points to common ancestry. And, you know, there are a lot of stories about, well, cars are built in hierarchy, so they must have been created. Well, why would God, in his infinite wisdom and infinite creativity, need to do that or want to do that? Surely he would choose at least 2 separate lineages and kind of build from there. Why would he just do all one? So, yeah, good debate. I'll be in the after show. And again, contact your senators about that exotic animal ban. Let's not let that pass, but not like contacting your senator ever does anything. So I'll hand it back to Donny.
All right, Jackson. And if you have a link to what you just said there at the very end, you could post that for the audience below so we can let them know.
Yeah.
Take a sign. All right, and Donny, when you are ready.
All right, let me get my timer here. 5 minutes. And I appreciate that, Jackson. We are friends. We are good friends, and I appreciate this profitable discussion. And I always appreciate how cordial and respectful you are. And I appreciate you asking the good questions. So, okay, I am going to start my timer here and share screen. So, again, universal ancestry or separate ancestry. Jackson's burden of proof, the burden of proof for all the evolutionists. Don't show us variation within a kind. Okay, and you can define a kind as all those organisms that go back to an ancestor. And you know, we've done that with Dinosauria. We've shown created groups. We've also done that with especially with humans. The Australopithecines, there's a separate ancestry model, but it's their burden of proof to demonstrate that not only dogs and wolves are related, but that dogs, wolves, and banana plants are related. Okay, homology is not going to do it. Nested hierarchical patterns is not going to do it. Transitional forms is not going to do it. And the evidence, the best differentiating lines of evidence that we discussed today, and this is my 95th debate, and I've had debates on all sorts of topics, but these main questions that I have regarding the Y chromosome data, we didn't even get into the hat map data. I didn't have time to get into the linkage block data that suggests separate ancestry, because we have these massive linkage blocks that have no evidence of recombination gene conversion. I mean, there's some, but not nearly enough. If we have recombination every generation, let's say, okay, you're scrambling up the DNA variants for hundreds of thousands to millions of years, then why is there still so many large linkage blocks in the human genome? There should have been much more scrambling if human evolution were true. If we actually go back a couple 100,000 years. I've asked that question to PhDs. I've asked that question to your most militant evolutionists, and they never have an answer. I didn't see any answer to the satellite DNA problem from the Ventura paper when it comes to the chromosome 2 fusion. And when it comes to the chromosome 2 fusion, if there was no fusion, then guess what? There was no human evolution. And he talked about the centromere and the telomeres, and no, that's wrong. That's wrong. However, both the alleged fusion and centromere sites fail the test when analyzed closely. Not only are these alleged evolutionary signatures very small in size and muddled, degenerate, but both of them are found inside what? Functional genes. The alleged fusions site functions as a genetic switch called a promoter inside a long non-coding RNA gene, and the so-called fossil centromere, which can't be found, Jackson, contains both coding and non-coding sequence inside a large protein coding gene. Combined together, these data destroy the evolutionary hypothesis. And again, the question from the Ventura paper, in the fact that chimpanzee and gorilla chromosomes differ from human chromosomes by the presence of large blocks of subterminal heterochromatin thought to be composed primarily of arrays of tandem satellite sequences. Listen, the evidence refutes a chromosome 2 fusion. If there was one, then based on the satellite DNA observations that we have here, at best, you have a human-specific fusion, which can best be explained by the creation model because we have two bottlenecks. We have the bottleneck at the flood. We also have a mini bottleneck at the Tower of Babel. So that'll, I mean, we could just go as close to home. Pseudogenes, functional DNA elements necessary to sustain healthy life processes in the cell, hierarchies. And last thing I'll say, why did God create this way? Well, he created this way for functional purposes. And this is exactly what we see. We predict that, see, you got the universal ancestry model that suggests that the majority of the genome is junk, consists of genomic fossils, consists of evolutionary leftovers. And yes, if that's exactly what we found, that would be reflective of universal ancestry, but that's not what we find. Over 80% of the human genome is actively involved in at least one or more biochemical reactions associated with gene regulation in at least one cell type. Function after function, gene expression, regulation, function for pseudogenes, endogenous retroviruses, functional redundancy, these third position codon. The third position codon variation we now know is involved in information flow. It's not just there to tolerate error. They're there to help slow and speed the process. ALU elements, co-option doesn't work. What we're seeing is confirmation of the separate ancestry model. We predict function. The evolutionary model predicts junk, evolutionary leftovers and genomic fossils, and we don't find that. So why is there homology? Why is there nested hierarchical patterns? Why do we find interesting mosaics? Because they were designed that way for a purpose, for a functional reason, and that's exactly what we find. And we have people like Dr. Nathaniel Jensen who've made testable predictions based on this model of created heterozygosity that suggests most of the genome is functional. Most of the DNA elements in the genome, like pseudogenes, ALU sequences, and ERVs, are functional, and that's exactly what we find, contradictory to the universal common ancestry model. And the proponents of universal common ancestry are not stepping up to the plate and making better testable predictions. So
Time.
why, and how much time do I got?
Time.
There you go. And thank you, Jackson. I appreciate the debate. Tons of fun. 5 minutes flies by. So,
okay, everyone. We are now heading into the questions. So, Jackson, these are like 90% for you. So I'm sorry.
Yeah. Sorry.
But we are going to jump into the super chats first. So the first one is going to be from Logical, Probable, Plausible. Thank you for the first super chat, directing everybody into your after show, which I will remind everybody again later. And then to Slew, I hope I got that name right. And it is for Jackson. It says, what explains the giraffe's neck better? Darwinian or Darwinianism or Lamarckism? And why would, I guess we could just start with that. It's a two-part question.
Yeah. I'm not good at remembering questions, as you can remember. But like I already forgot what that one was. No, Lamarckism doesn't work. It's not a thing. So it would be like neo-Darwinism, just a gradual process. It's not, the giraffe stretches its neck, so the next generation has a longer neck. That's basically Lamarckism. So that's not how it works.
And the second part, why would an organism change to begin with?
To adapt to a new environment.
All right.
Yeah.
Yeah, it was just a short question.
Yeah.
Next one was from Logical, Probable, Plausible. I guess the question would be for you, Standing. Will Joel Duff join the after show?
There's a good question. Yeah, let's see if Joel Duff has any answers to these problems that have not yet been answered. Let's get an answer. And maybe some better testable predictions, since Joel Duff likes to attack models from people like, Dr. Nathaniel Jensen and Dr. Sanford. But he's a nice guy. So yeah, let's have him join the after show.
All righty. Next question. Question for Roe. Anthony 1701. Man, he's really old. What is the best beneficial mutation you know of without any deleterious effects? And how will this mutation work against other mutations without being bred out after a few generations?
Like the gene that in the Linsky experiment that allowed E. coli to metabolize citrate was a duplication mutation. And how it would balance out with the other ones, I don't know. But yeah, they do add information, and so.
Okay, good.
Matt, let me respond.
Yes.
So when it comes to beneficial mutations, okay, they do occur, but there's a few problems with that. And the problems are that they occur infrequently. They're rare. I think it's about 1 in a million they've discovered from the Linsky experiment. And when they do occur, they're usually reductive and functionally compromising to the organism. Even a lot of their best beneficial mutations, it just turns out that it's a gene that's broken and permanently shut off. And there's a waiting time problem when it comes to these beneficial mutations becoming actually fixated in a population, because there's a lot of genetic noise. Oftentimes selection doesn't even see them. And when it comes to a lot of these beneficial mutations, like I believe the major one seen in the Linsky experiment, all we're seeing is rearrangement of pre-existing regulatory system. Okay. But that's not going to drive a fish to fisherman or bacteria-like biologist, a bacteria-like organism into a biologist. I mean, if you look at sickle cell anemia, it's got, I guess, a bit of an advantage, even though malaria is bad, sickle cell anemia is bad, and it's all due to a broken gene, broken protein, broken cell. How's that ever going to counterbalance the damage done by this low impact, nearly neutral deleterious mutation accumulation? So the point is, beneficial mutations, although they happen, they're never going to drive large scale evolution. I yield.
Okay, I guess that will bring us into the next question again.
Well, I think if Jackson wants the last word, because it's his.
Not sorry. We can go to the next question.
It's on the same topic. So, if whales went from fully terrestrial Pachycetus, or I'm not sure what he actually said.
Pachycetus.
Yeah, Pachycetus, to fully aquatic Basilosaurus. In 200,000 generations. How can't E. coli evolve into literally anything at all? Well over 65,000 generations.
Well, I don't know how many generations it took for whales, but bacteria are reproducing at the rate they've always reproduced relative to them. I'm not sure if that makes sense. It makes sense to me in my head, but I'm not sure that translates out. So you're not going to just pop out a frog from some bacteria. That's basically my answer.
I'll respond, Matt. A little bit of share screen. It's funny because Linsky's bacterial populations, we've got thousands and thousands and thousands of generations of bacteria, and all we ever get is bacteria producing more bacteria, but the more bacteria is degenerated bacteria. So we got bacteria producing bacteria in these experiments, fruit flies producing fruit flies in the many thousands of fruit fly experiments. But those that believe in universal common ancestry want to say that fruit flies, bacteria, and strawberries are related. And that's why they can't meet that burden of proof, because that's not what we see empirically. When it comes to Linsky's experiment, I find it funny because Linsky, as you can see here from Dr. John Sanford, although a very trivial adaptation happened, optimal growth on a given medium, his bacteria shrank in genome size. The functional genome decreased. Evidently, the more rapid growth was largely accomplished through genetic degeneration. Many useful genes not essential in that artificial environment were apparently lost. When transferred to a natural environment, those highly degenerated bacteria would essentially be dead on arrival. So we have bacteria that are losing genes short term for adaptive purposes, but what it actually is long-term degeneration. And the last thing I want to say in terms of the whale evolution is, even according to the evolutionist time frame, okay, millions of years is not enough time to convert a land mammal to a fully aquatic mammal. I mean, how does Jackson explain empirically these types of novel adaptations, emergence of blowhole and musculature and nerve control, emergence of ball vertebrate, modification of the eye for underwater vision, change in birthing process where fetus is delivered in breech position, ability to nurse young underwater, origin of blubber for temperature insulation. So, I mean, the origin of tail flukes and musculature. Again, this goes back to Jackson's opening statement and the opening statement of most well-studied evolutionists is, don't tell me that it happened if you can't tell me how it happened. If you can't tell me how it happened, then you're just imposing your presupposition on the genetic data, which is the empirical, direct way of determining ancestry. I yield.
Well, a lot of the physiological features were already present by Pagasitis, but not all of them, obviously. But like the birthing thing, Myocetus was found with a fetus inside it. It indicated it was still getting birth on land, even though it was semi-aquatic. And tail flukes, Paragasitis had a compressed tail caudal vertebra that would have given it like a kind of beaver-like tail, kind of an intermediate tail fluke. So that's all.
All right. Next question is going to be for you, Donny. It is, I think that the strength of Jackson's model is the ability to predict where fossils will be found. How does Standing for Truth explain their ability to predict where a specific fossil animal will be found?
Sure. Good question. Jackson and I discussed that at great length in the discussion portion. And again, Jackson, I want to thank you for a sophisticated discussion. We talked about so many different topics and subjects, and I think this was one of my favorite debates. So
sorry, my memory isn't that great, or it would have been a better debate.
That's okay. And you know what? Even the greatest memories can't always remember every single point that's being brought up in a discussion such as this, a highly technical discussion. So here's the thing that I find funny is they can't accurately predict the series of so-called unnecessary Here's the thing. We would need thousands and thousands and thousands of so-called transitional forms. Okay. If evolution really were true, that we've got a single-celled-like ancestor billions of years ago, evolving into a multi-celled ancestor, into a fish, amphibian, reptile, mammal, okay, eventually a man, something as sophisticated and complex as a man, one single cell, more complicated than the space shuttle. Not to mention that single-celled ancestor billions of years ago would have had to come from non-living chemicals, which we have no evidence for, which is why the design diversity hypothesis is most reasonable and most plausible. Now, this predictions, no, they're failed predictions. Even the Tiktaalik example, and I understand that they can force fit it into the story. Well, it's still on the tree. It's not the direct ancestor. Okay, sure. You got a population of this creature called Tiktaalik. You don't know if that's just an evolutionary dead end, an interesting mosaic. You don't know that evolved into anything. You don't know that species survived far enough into the future to actually evolve into a land-dwelling tetrapod. No, what we find here is tetrapod foot tracks before Tiktaalik. That means we have tetrapods living before Tiktaalik, which is best understood from our model because that's what we believe is in the pre-flood world. We have overlap. We have different ecosystems, right? We have logical zonation. This is our model. When we apply the creationist global flood model of progressive burial by ecological zonation, the presence of these unusual fish-like fossils that we talked about that Jackson presented makes perfect sense since these creatures were buried in the flood layers shortly before most coastal land plants appear in the Carboniferous. It becomes apparent that they likely inhabited coastal waters based on their various unique adaptations and their place of burial in the flood strata. When it comes to the evidence, the fossils say no to tetrapod evolution. No, the predictions fail. They're not good. Anyways, go ahead. Jackson, you can respond if you want.
Well, this doesn't just apply to transitional fossils. It applies to pretty much any fossil. I mean, where are you going to find red lichen trilobites in the Cambrian, without exception. Where are you going to find other species of trilobites? Up until the Permian and never past it, without exception. Where are you going to find Gorgonopsian therapsids? In the Permian. Where are you going to find cynodonts most of the time? The Triassic. Where are you going to find archosaurs most of the time? The Triassic. Where are you going to find, there are 100 more examples I could go on about, but that's basically the answer.
Sure. I can have a final response. So again, that's assuming, and the places where you can actually find that sequence, okay, Precambrian, Cambrian, up to the Jurassic, Triassic, so on and so forth, Permian, okay, is less than 1%. Less than 1% of the Earth's strata. The entire Earth is where you find the so-called layers in that sequence, which means and you still don't get all the necessary strata that would have to be there if the geological column as portrayed in the textbooks is true. So that's why here we go. Does the geological column exist? I've got hundreds of slides here. So you can read through this where it shows, secondly, those locations where it has been possible to assign all 10 periods represent less than 0.4% of the Earth's surface, or 1% if the ocean basins are included. It does not engender confidence in the reality of the geological column when it is absent 99% of the time. So the point is, most of those layers were formed at the exact same time. We have amazing evidence for the flood. For example, the cold slabs that were predicted from catastrophic plate tectonics that are found deep within Earth's crust that should have melted if tectonic forces put them down there millions of years ago. The fact that they're still cool and they haven't yet either melted or warmed up to the surrounding material is fascinating evidence of the global flood and rapid subduction, and therefore rapid burial during the flood, ecological zonation. Remember, this is a burial during the flood. As the floodwaters inundated the continents, the shallow marine invertebrates were first swept from the pre-flood ocean floors and buried on the continents in rapid succession. After the waters rose over the continents, they progressively encountered different ecological zones at different elevations, which were inundated in rapid succession. So sure, you got different ecosystems, let's say with the trilobites, which we wouldn't expect to be living, let's say, with humans. We see different elevations in Genesis. We know that there are rivers flowing out of Eden, and we know rivers flow downhill. So we have different elevations, different ecosystems, humans and dinosaurs living in different systems. Okay, so the evolutionists, of course, are always going back to that assumption that the geological column exists. I think the question was for Jackson, so he can respond however he wants, but I want him to address, especially.
No, that was for you.
Oh, it was? Oh,
that was yours.
So,
yep.
Okay. I'll yield there.
All right. Question is then for Jackson. What would be an alternative explanation for the diversity of life if both creation and evolution turn out to be wrong?
That's a good question. I guess it would be a different God. That would be my best guess, but I don't know.
Donny, what would you say?
So if creation and evolution wasn't true.
Right. Is there a third option?
I guess, either we're created or not, created or evolved, or I guess there's, a percentage of people out there that just say we're not really here. Maybe this is the Matrix. Maybe this is some kind of computer simulation. So, who knows?
Right. All right. Question for Donny this time from DMC. What did animals eat after the flood if the earth was covered in salt water?
If the earth was covered in salt water after the flood, what did they eat? Well, it depends if the pre-flood world was fresh water and then gradually became saltier from the flood itself, the fountains of the great deep breaking open, leading and resulting into the perfect chemical composition of the water to form things like limestone, so on and so forth. So as the oceans gradually became saltier, we also know that the pre-flood world had incredible diversity in terms of plants and organisms and just fauna. So we would have plenty of plants that were uprooted, right? And then replanted either during the flood or after the flood, where you'd have tons of plants to eat, tons of fish. I don't see it being a problem if the salt water is just going to gradually accumulate during the flood and then after the flood. So there'd be plenty of plant material to eat, which would not be a problem for a lot of the pre-flood animals that were brought on board the ark. Those archetypes, many of them would have been vegetarian. Plus Noah would have brought a ton of different types of food onto the ark for the animals themselves. So I don't see there being a problem.
Well, I see it as a problem because basically the whole world is leveled and all the vegetation would be destroyed. I don't see how a tree is going to get picked up and replanted somewhere else, which is kind of an oversimplification of what you said. But yeah, they get off the boat. There's nothing. It's a barren landscape. Maybe there's some dead plant material, badly decayed after a year. Just mats of goo to eat, maybe. But the herbivores don't have that problem because they're going to get eaten by the carnivores anyway. So, I mean, we kind of have a major problem.
Well, I guess I get the last word. So again, Noah could have brought on, which makes sense. I mean, he would have been incredibly smart. He had 100 years to build the ark. He would have brought on fodder animals, like, tortoises are a good fodder animal, for example. He could have brought on dried meats. Okay, tons of food to eat during the flood year, and also food that can be eaten afterwards. And we see it in floods today. Of course, if you have plants being ripped up, trees being ripped up, they can be rooted again after the fact. You'd have plenty of dead animals, plenty of food to eat. When it comes to the carnivores, apparently eating the herbivores, I think that has to do with a lot of the migration patterns, where you have more of the predatory animals driving a lot of the non-predatory animals like kangaroos further and further. And that's where you see them getting stuck in places like Australia. You'd have land bridges where you'd have a post-flood ice age, where you'd have the water levels are not raised as high because you put all that water into the ice caps. Then you can pretty much walk anywhere on the earth. Once the ice caps melt, a lot of these animals that were driven, non-predatory animals that were driven to different places in the earth, they'd be trapped to where they are. But the carnivory wouldn't be the same as it is today then, especially if a lot of the animals were still used to a vegetarian diet. A lot of the habits of carnivores today would have occurred sometime after the flood through epigenetic means, epigenetic switches that can be turned on when a lot of these non-carnivores become carnivores. So I think we're looking at animals today, what they eat today, and assuming that's exactly how it was in the pre-flood world and in the immediate post-flood world. So I don't know. I yield there.
All right. Next question for Jackson. In the book Phosphate Rocks of Carolina, it was stated unequivocally that human bones and arrows were found buried in the same rock layer as dinosaurs. Jackson, how do you refute? If you've even heard of the book, of course.
I've never heard of this idea, so I don't know how to refute it.
Okay. Have you, Donny, have you heard of
Can you repeat the question?
Yeah, it's about the phosphate beds of South Carolina. In the book Phosphate Rocks of Carolina, it states that human bones found with dinosaur.
Right. Yeah, that's a good argument too. So there are some anomalous examples like the Ashley phosphate bed where you have human bones mixed with dinosaur bones. Okay, so we have examples of that, which I believe the evolutionists would say like there's maybe some overthrusting going on. Of course, they've got a rescue device, but again, when it comes to ecological zonation, and I'll screen share here real quick just for the audience, whether or not we found humans and dinosaurs together. I mean, it's amazing that it seems like that's what we find in the Ashley phosphate bed. When it comes to ecological zonation, we wouldn't necessarily expect have humans and this is what I wanted to go to. Humans and dinosaurs living in the same areas. In Genesis chapter 2, this is kind of what I hinted at earlier. The river coming out of the Garden of Eden is described as dividing into 4 rivers, which may imply the Garden of Eden, with its fruit trees and other angiosperms, mammals, and man, was at a high point geographically. The rivers flowing downhill to the lowland, swampy plains bordering the shorelines where the gymnosperms grew and the dinosaurs lived. This would explain why we don't find human and dinosaur fossils remain together in the geological record. Dinosaurs and gymnosperms only fossilize together, and angiosperms only fossilize with mammals, and man higher in the record, separate from the dinosaurs and gymnosperms. This is the same thing with flyers too, like pterodons, pterodactyls, birds, bats, different ecosystems. Just because they fly doesn't mean that we'd expect them to all live in the same ecosystem. Same thing with the ocean, sharks, whales, so on and so forth. But I think the Ashley phosphate bed example is bonus on top of the creation model. We wouldn't necessarily expect it or predict it because of ecological zonation, but the fact that we do find them together is incredibly contradictory to the evolutionary base model and the geological column. We can give Jackson the last word if he wants.
Yeah, I don't really get the ecologic zonation thing, because it's assuming that everything's buried immediately and stays there. In a turbulent global flood that lasts a year, I would think things would get mixed around pretty badly. That's all I have to say.
All right. We're going up on about 2.5 hours, so I'll blow through some of the questions. Norman Bates wanted to know, do you think humans could ever evolve into leprechauns?
No.
It would be pretty cool if they could. If they had a pot of gold. Of course, they would devalue gold. So, but no, I don't think so.
I think we see humans today accumulating 100 new mutations per person per generation. So it's funny. You could make a ton of funny questions, derive a lot of funny questions based on the evolutionary model, since according to the evolutionists, humans came from non-human-like ancestors, which ultimately go back to a single-celled-like ancestor billions of years ago. So, I mean, are there really limits? Maybe they'll say there's physical limits. For example, can we get a dog the size of a whale or a dog the size of a flea? I mean, there's animals that exist that are the size of a whale and the size of a flea. They're called whales and fleas. But to take a single cell like ancestor into a whale, why are there limits according to evolution? But we know, based on the empirical data, humans are accumulating deleterious mutations. They're not evolving into anything other than humans. As a matter of fact, there's a shelf life on humans, which is why our hope is in Christ. That's why we want Jackson today to believe on the Lord Jesus Christ, because genetic entropy is a reality. There's been no rebuttal. All the best rebuttals have been systematically analyzed and falsified. And our hope is in Christ, and our hope is in heaven. So, I yield.
Well, I don't think that genetic entropy and Christ are, necessarily needed together. But I think genetic entropy is not the greatest idea. I mean, I've been doing a lot of research on that myself, and I'll provide the numbers later, I guess. But if viruses with high mutation rates aren't going through some kind of genetic entropy, I don't see how we're going through it in 6,000 years from the patriarchs to now.
Are viruses alive?
They're made of genetic material. So, I mean, Sanford himself used H1N1 as an example for genetic entropy. So
now here's the thing. We talked about viruses a lot during the discussion. If viruses are created DNA elements, they started in the host rather than predated the host. Okay. But genetic entropy is written for longer lived animals, for example, which we know have higher mutation rates. Why would we then look to viruses, which exist in us, and then viral like elements as well, to refute the concept of genetic entropy?
I don't know. They're just made of genetic material, and they're replicating and they're mutating very quickly. I think it would apply.
We've done thousands and thousands and thousands of experiments, numerical simulations, all realistic parameters, different types of selection, different types of amounts of beneficial mutations, different population sizes. And what we always get, and these are documented, is degeneration. Selection can only ever slow down the process and not stop it or solve it. And what they've put forth in terms of truncation selection, synergistic epistasis, mutation count mechanism, they've been looked at, documented, analyzed. You can read papers on it and articles on it. They've been falsified, but you want to look to viruses. Doesn't really make much sense. It sounds like you just don't want to believe it. And I'm not trying to be rude. It just sounds to me like how come all of these evolutionary population geneticists like Lynch, Kondrashov, Kimura, how come they never say, well, there's no point in, in looking into the mutation accumulation problem because, wow, viruses exist. How come we don't see them saying that? I mean, they recognize it's a real problem. Kondrashov said, why are we not extinct or dead 100 times over? How do you respond to that?
Yeah, he did say that. I forget why he said that. I read part of that. Yeah, I mean, I don't have a great response to you. Again, it's just to say that viruses are genetic material, and I think they're a good analog.
It's okay. We can use that for the after show. If you're going, that is.
Yeah, I'm going.
All right. Thanks,
the
John.
Continues all night.
Yeah, thanks for the super chat, John. Just a reminder, that's where that's going to be right now as we wrap this up. So, Donny, if you wanted to let everybody know about future debates that are coming up, and you can close it down.
John's like, this is the Q&A, not a further debate. Yeah, this has been a long one, a detailed one, a technical. I'm exhausted. Jackson, I'm sure you are too. I'll probably get a sandwich in, or I don't know, another coffee or something. This was a technical, fun debate. Lots of good subjects and topics discussed. Good audience Q&A. The next debate that we have is Jackson Roe. You better get some rest, my good man.
Yeah, I need to do some more research on that. Also,
we'll try to not
make some slides.
We'll try to not make it yet. So that's going to be fun. You and Professor McQueen. And then right after that, the big round 2 between David Neff and Dr. Dino. And all I can say is go have a look at the upcoming live stream section because we got about 40 more debates. So, Matt, great job moderating. I appreciate it.
Except for the end there when it turned into a second debate.
Matt, you're on mute, brother.
Whoops. I'm just saying good job. Couldn't even get through it. Thanks.
You did a fantastic job. There we go. 2.5 hours, man. It didn't feel like that though, Jackson. Tons of great points discussed. I hope the audience had a good time. Looks like we still have 90 people. So, we kept everybody's attention. Thank you for preparing and bringing some good arguments that we can discuss, Jackson. So, guys, we'll see you over at the after show. Logical, plausible, as Matt calls it, possible.
I can't remember
Logical, plausible, probables channel. We'll see you there for another epic after show. I'll probably join in about 45 minutes. I got to get some food and take a little break here. So,
and some coffee.
Yeah, guys, God bless. Thanks for tuning in. We'll see you in a couple days. Standing for Truth is out.